you can get an answer for this. So most people have known Einstein's equation E equal mc squared. Very simple, very few letters. But that describes the energy and matter conversion with all the electricity you have here, and they all come from that. So here can also be described by mathematical equations. Here, Benoit-Mandelbrot. So z n plus 1 equals z squared n plus c. Very simple formula, or simplify even here, z reaches z squared plus c. Benoit-Mandelbrot. He spent more than 20 years at IBM to develop the single formula. And it's called a fractal. So what is fractal? It's a self-similarity at a multiple scale. So some people have seen those kind artistic images. Very beautiful image. And turned out, if you amplify one image here, you amplify tens of times, 100 times, they look almost the same inside. Down the bottom part here is called self-similarity at multiple scale. It's a square, big square, small square, and so on for small square. So that's a fractal, can be described by the mathematical formula. So if you have an iPhone or any kind of mobile phone in your pocket, and 20 years ago or 15 years ago, phone has an antenna, right? Well, people too young, you don't know. So what's happened to antenna? Disappeared. Turned out they all have the antenna inside your phone to receive the signal. The fractal is only pattern for the antenna to be invisible. That's the power of mathematics. Okay, so ask questions, what have in common? Turned out something be very useful. Now it's a hundreds of billion dollar business annually. Okay. Here is the man who invented the equation, 20 years dedicated at IBM. And MIT tried to recruit him, Harvard tried to recruit him. In the end, he turned down all this because it involved the teaching and the committees and so on and so forth. IBM left him alone. And he could do this. So I visited him, and he visited me. So these two images, the look at the art, had the exhibit in the Guggenheim Museum in New York. Now, how many of you have watched the movie Star Wars? So all the Star Wars, the mountains, the rivers, the lakes, and images are generated by fractals, not real. It's all done by his mathematical formula. So save Hollywood hundreds of billion dollars just done in the computers. So it turned out fractal has a lot of uses for medicines. So he wrote many books. Turned out fractal is useful in physics, in electrical engineering, in stock market, and in medicine. So the heart rate, and it's also fractal. So here is a simple examples how fractal look like. It's a first iteration, third and fourth. Go back here, 1, 2, 3, 4. 4 iterations. So each time it's a similar scale. It's a different scale, but a similar pattern. You can go this. So it's a little unreal. That's just right. And here it's a fractal in triangle form. And here, finally, it's a self-similar dynamics. It's a 300 minutes, 30 minutes, and 3 minutes. Earthquake is also very similar. The heart rate is very similar too. We have doctors sitting here, so he would know. Here is the universe. Its scale is billions light years. Billions light years. Here is a neuron synapse. You have heard Ed Boyden yesterday gave a talk, neuron synapse. The scale is 100 micron. So it's how many trillions difference the scale. But look at the same, similar pattern, right? Without taking this scale, you actually don't know which one you're which. Mandelbrot gave a talk in 2003 at a meeting without a scale bar there. So ask the audience to guess. People guess this and that, so on. Finally, he put a scale bar there, and everybody was wrong. So here is, again, the universe. If you look at from billions light years, this local universe look very similar. Very similar to that, and similar to that. So each one of them start. It's not a planet. It's a galaxy. You can see it's a galaxy. Finally, it's our home, the Milky Way. The Milky Way from Earth will look like that. You go to Grand Canyon or go to New Hampshire, go to this place and no light, you can see galaxy like that. Finally, you come to our home. If you look at stars in the dark skies, so many stars, you go closer, you still see more stars, just getting closer. This cloud, again, you cannot describe this phenomenon by conventional geometry, what you learn in high school. You cannot describe this. How can you make which triangle, which circle? You cannot describe this. But that single formula by Mandelbrot, and then led to explain everything about the nature. Okay? 20 years hard work. The wave is also fractal. So is the light flash. You can even see it's the first iterations, second iterations, fourth iteration, fifth iteration, and so on. Okay. This is the inspiration for your phone antenna. Okay. And many other things. Look, this is Brazil's, the Amazon River. You can really see first iteration, second iteration, third, fourth, and fifth, and so on and so forth. Yep. Here's the river in Greenland, the same way. I'm from China, from Sichuan province. Look, this is a mountain range. The mountain range here, taken from the satellite, is very similar to this frostbite on the window of your car. Yeah, but this is hundreds of thousands kilometers it's taking this. And the frost on the car is only how many inches. This is a desert, the same way, have same fractal pattern. The mountains is the same, big and small. When you amplify going closer, it's more details can be seen. I sent this before. You can see the cracks. It's cracks here, big cracks, small cracks, small crack. It's all in fractal pattern. So imagine one single mathematical formula describe everything, unify everything. So observe what the nature phenomenon can lead to many interesting things. Observation is the key. So people say to see, but not observe. Happened to me. I come to work, and there's so many streets on both sides. When you're driving, you don't bother to look what street is, and because you don't observe, you only see it. So to observe, that's a part of science, part of engineering, and make discoveries. So there's a waterfall, the fractal pattern. This is actually taken from my home, from the window of my house. You can see all the fractal patterns. The top, you can see the winter, and it's similar under the roots. Okay, so I've shown you so many different things. There's tree bark. So something so ordinary, and people just don't think about it. But if you think deeply, like a Mandelbrot, and he connect all the seemingly unconnected things into one thing and describe a simple formula. Okay, so the same thing to Einstein uses to describe a single formula with E = mc squared. Okay, so this is a piece of stone from Himalayas. This shows a fractal pattern, there's self-similarity at a multiple scale. The seashell, you can see the seashell system, big one, small one, so on and so forth. And if you cut open, the same way. Yeah? So mainly, I just want to get a student to pay attention to your everyday life, everyday things. This is corals. That's actually they're in my office, those ones. And there's this beautiful this plant with this water droplet on it. This are actual plants. This is one with the leaf. Beautiful fractals. Just look like the frostbite on your window. The leaf of the tree. This balcony, the photo was taken in supermarket by myself. Or maybe the trees and also took this picture. And look at the snowflake, a single snowflake, and a fractal pattern as well. Here is the frostbite on the window. The same, very similar to the Himalayas, the mountain range. The soap bubbles and the doctor's famous chart. The circulation system of your own body and the lung. Okay, again, all the fractals. You have your fractal yourself walking around. Now, the question is, would any be thing useful? I just mentioned to you earlier. Turned out it's billions, hundreds of billions dollar business. There's people file patents for this, design new antennas. And people make antennas, the real, and put in your phone here. Okay? Those are simulations, and this is actual phone. Open up your phone. This is the lens. So most people don't realize we have a camera has a lens, glass lens. But when you have a phone, this camera is so small. What's happened here, right? Turned out that's a lens, a photon collector. Again, it's a fractal pattern. So it can have 20 megapixels, beautiful pictures, come from the photon collectors. This is my own research. When Mandelbrot visited my office, I explained to my research. He said, this is fractal. It is fractal. This my own research shows the nanoscale, this hydrogel fibers. This discovery led to a company that was $1.2 billion from serendipity discovery. You can see fractal use everywhere. So we're eating Jello, we're eating some gelatin stuff. Why is mostly the water holds that thing, right? Turned out all the things has a fractal pattern if you look on the microscope. So people don't realize it's because 90%, or sometimes in this case, it's 99% is water. But water don't flow away, then form a gel type material. This material is so useful for wound healing, for surgical wound healing, for the heart wound healing, for heart surgery, liver surgery, kidney surgery, and many other surgical use. This turned out this material because of fractal pattern. First can also maintain some art or useful things. And this is stock market here. So different days, and 1 year, and 6 months, 1 month, and 1 day. Look at this. That's a Mandelbrot wrote a book about the stock market. This is the temperature, right? Bottom part is heart rate. Yeah. Okay. So my question is, can you find other things that have fractal pattern and useful? I gave a similar talk in China, and someone, academician, Chinese Academy of Sciences, he said, Chinese bureaucrat system. You have a central government, you have a provincial government, you have a county government, city government, down to something. True, fractal. Yeah. Okay. Now, let me tell you something else now. So this is a media lab. You are in one of the buildings. The building was designed by I.M. Pei. In 1985, the media lab was opened. I'm glad you saw this earlier, the video. When they designed the building in 1985, there's one of the building at MIT has so much called wasted space because no rooms. Now you can see also when you come upstairs here, it's a lot of empty space. Turned out this waste space is kind of general inspiration. On the first floor here is the piano, grand piano, Bosendorfer piano people play in there. Very useful. And turned out that kind of design has inspired so many hotels and other company buildings similar. This is building you are in. You are right in this building. Okay, now tell you a little bit about my research and my heroes. So the person, 4 people here, one is Alexander Rich. He's my mentor. And he has been at MIT since 1957 and passed away 2015. 58 years here. He come to work every day and until almost last couple months before he went to MGH. And he told me when he was your age, 16 years old, he also tried to work in the lab. He loved it. And then people later even pay him. He said, wow, I love to do what I do and get paid. So I want to do this for the rest of my life. You get a job, you don't get a job. You get something, get paid to play. That's what the exact media lab people have this pleasure. It's not called a position, not called a job. It's really get paid to play. Lego company here, Lego toy company, pays media lab. Yeah, Swatch, I have a Swatch company, pays media lab. Motorola, earlier companies, and so on. Numerous companies, we saw the television, 90 companies then funded media lab research. So you have heard earlier they invented so many different things. People take it for granted. It's a crazy idea the student had, and it becomes reality. Yeah. So the house on the Francis Crick and Jim Watson, that's one. How many have heard those 2 people? Heard those people? Francis Crick, Jim Watson. Those 2 people did a model of DNA double helix. Then we obtained the data from Rosalind Franklin and made a model. It was that model before February 28th, 1953. People did not understand how genetics work. Why you look like your parents? Why is a cow produce a young cow, and horse produce horse, and broccoli produce broccoli? Why? People try to understand that. Before the 28th of February 28, 1953, people had no clue. That day, the February 28th, 1953, everything become understood of genetics, a single day difference. Because as Francis Crick said, they discovered the secret of life. is a double helix structure. Explain all the genetics. Okay, that has generated trillions dollars economy. So all the biotech companies around here, and all the healthcare hospitals in Boston area around the globe, had a lot to do with the discovery of DNA double helix. So Leslie Ogle, you may have heard yesterday, who is one of the pioneers studying the origin of life. And Jack Shostak, he gave a talk yesterday and was very much inspired by Leslie Ogle. The four people are giant icons of modern molecular biology. In religion, there are a lot of icons. But in science, we also have icons. There are four of them here. The picture on the right is the house of Francis Crick. The yellow helix is not a DNA double helix. He put this up before he discovered the DNA double helix. He was working on the alpha helix of protein. Turned out that alpha helix is so ubiquitous in proteins. Everything in protein has one with the fundamental structure, alpha helix. The person who discovered this, Linus Pauling, he won a Nobel Prize for not only discover alpha helix, but many other things. Professor Linus Pauling was at Caltech. Very poor, grew up very poor in East Oregon, eventually become giant in science. Went to Europe, brought quantum mechanics to US, and applied to chemistry. Before quantum mechanics was applied to chemistry, was called imperial science. Once applied quantum mechanics into chemistry, become exact science. Okay, so the alpha helix of protein is directly relevant for my own research. That picture was taken, first picture was taken in 1953, 1955 in Francis Crick's house, in that house. And this is taken, I took the picture, 1993, and in Paris. Look at the ties they have, 3 of them. It's an RNA tie club. It's only 20 ties on the planet, in the world. Watson could not wear because that went in the archive in Cold Spring Harbor. He was not allowed to bring it out, to wear this. So the tie RNA tie club. And to talk about the interesting phenomena, you can go Google RNA tie club. It's composed by physicists, engineers, chemists, and biologists. They started the club by George Gamow, the astrophysicist. He also coined a word called Big Bang theory. He also proposed the DNA codon, the genetic code is 3-letter code. Purely by calculation, mathematics, is minimal you need it for 20 amino acids. And 4 times 4 is only 16, not enough. So he said must be 3 would be minimal. Could be 4, could be 5, but 3 is the minimal. Turned out experimental evidence indeed verified it's 3. Actually, Professor Ben-Knodin has a theory why it be 3. So he can tell you a story later. If 3 is minimal needed, but why is 3 needed? He had done experiment and showing why it is 3. This is Nanger. He gave a talk, inspirational talk yesterday. But look what he said here. He said in school, you are judged by the question you answer. That's your exams, your homework, right? You either get an A or get a B because the question you answer. Or you don't get a pass because you answer wrong. But he said, in real life, you're judged by the question you ask. So yesterday, he was sitting up here. I asked him to repeat this one question he said. It's true. So ask questions, constantly ask questions. That leads to a new frontier of research. So Molecular Frontier gave one prize called the Molecular Frontier Inquiry Prize. I encourage you to submit the questions through your teachers. And this first prize given to people who have never accomplished anything. But it's through ask questions. The questions leads to research. Question leads to answers. If you ask big questions, you get big answers. You ask small questions, you get small answers, right? You ask where to go for lunch. Well, that's a small question. You ask question, what is the salt? What is molecule of salt? That's a big question. By the way, that question is asked by one of Molecular Frontier students. What is molecule of salt? That's actually very profound. It's asked by students for no more than 18 years old. Adults don't ask this kind of question anymore, right? So this is myself, and that's why I told you earlier, I invented this discovered totally by serendipity discovery. Working on the yeast, discover protein, and protein has a small repeating peptides. I developed this material, a fractal material, eventually become product and useful for human surgical and wound healing applications. Now, I will ask another question to all of you here. So, what do they have in common? Again, right? Keep asking questions. The watch, the Gucci bag, Swiss Army knife, and the porcelain plates, dinner plates, chocolate, and the sunglasses. What do they have in common? Think about it. It's not so obvious. It's one word describes this. Design. That is design. The watch is designed, right? Gucci bag designed. And the Swiss, all the things are designed, even including chocolate. The design, very precision. Design means you have absolute control. So what the final product comes. Okay? So you can design, you can become name brand. iPhone is designed. iPhone has a design team. So there's a school, Rhode Island School of Design. Industrial design and what design media lab people here is for design. So design something visible, design something invisible. So what I'm doing research now is design something invisible, but could be also useful. Here is Leonardo da Vinci said, simplicity is ultimate sophistication. First I saw this quote is from John Murray Lin. 25 years ago, he gave a talk, had this quote, come from someone from Florence. Well, professor there, Brignani from Florence, that's his home. Here is, I work on 8 years to design a code. So this is a summary, the code, impact of the code. Morse code there, making that telegraph communications simple. It's one side you can know the code, the other side 1,000 miles away. If you have the same code, you can decipher the code. This generates billions of dollars industry, telecommunications. Second code, computer code, it's so ubiquitous, people even forgot about this. So if you know how to do coding, right? In high school, learn some coding, your life will be much better off than without knowing coding. That generates the business with the wealth for humanity is incalculable. Third, the genetic code is so important for all life, not only from bacteria, viruses, for human being, plants, and so on and so forth. That's why you get infected by virus, because virus use the same code as you. That's also the reason when you eat a chicken, you do not become a chicken. true. You can eat a fish, eat a hamburger, and so on. You do not become cow, you become fish, because you use the same genetic code. When you eat it, you digest the protein, making individual amino acids, and use again to build yourself. Just like building a house with the bricks. Individual brick can be used building walls, building houses, building streets, and so on. Individual pieces, that's coded in biology by the genetic code. It's a simple code, 64, as I said, proposed by this astronomer. So finally, I invented the QTY code. 8 years to invent the code. The code is something, once you invent this, you can design the protein molecule so precisely according to your desire. So what we want to do, there's a class protein called the G protein coupled receptors, which is not so important. It's membrane protein. The membrane protein here embedded in the membrane is membrane protein is called a cell communication device, just like a mobile phone. If somebody takes your mobile phone away, you cannot communicate with a friend. You cannot texting, cannot make phone calls, cannot find somebody. So the membrane proteins have the same function. By the way, 1/3 of your genes coded for membrane protein is not important, 1/3. Yeah? And every other medicine you take, every other one, this regardless prescribed by doctor or by the over-the-counter, is acting on membrane protein. So, but it's so difficult to study them. And again, you can say mainly alpha helices is on the house of Francis Crick. But if you take them out, you need a called surfactant or detergent. Laundry detergent keeps them not falling apart, but difficult. So I want to design a different protein, take them out, put into water without a detergent. If people say that's crazy, it is crazy, but crazy ideas, crazy is better sometimes, as long as you can find something to do. So here are the student postdocs who did the work. One of them, by the way, is from Linköping, Lothar Tickler. I had 5 Swedish postdocs in my lab. And others are visiting professor from Shanghai, and Dr. Han Qiuyu from Shanghai, from Fudan University, and Ray Qin, Dr. Ray Qin is here somewhere. Finally, Michael Skursky, this student here. Michael student have said one word about him. And he's a student so unusual that it's a crazy idea MIT Media Lab accept him to become PhD student. He totally learned on his own from 6 years old. He didn't go to elementary school, didn't go to kindergarten, no elementary school, no junior high school, no junior high, no high school, no university degree. He has 0 degree paper. But Media Lab accept him to be a PhD student because he's a genius. Yeah. He has been on his spare time and worked on astronomy, worked on nuclear physics, worked on protein folding, protein design with me and with Andreas. And so it's a truly amazing individual and very young. Came here at age 22 without any degree. So MIT Media Lab accepted him to become a PhD student. So a degree is a piece of paper, but the capability, what he can do, is something different. That's why he knows how to do coding. Yeah? Learn some coding is very good. Here is a code or DNA code. DNA code is simple. The TCAG, once you write on one part, you immediately know other side. That was Francis Crick, James Watson discovered the code. Okay, generated trillions of dollars economy, probably 10% of the economy worldwide is through this discovery. Here is hemoglobin. Hemoglobin molecule, again, for the alpha helices. I should have used the alpha helix picture. And this protein has 80% alpha helix discovered, and the crystal structure designed by Max Prout. And this protein is one of the most water-soluble protein known. 30% in your red blood cells. 30%. That's why it can breathe. It can go transport oxygen in and out. It's transport oxygen gets in, get out carbon dioxide out. So this protein is 80% alpha helix. There's another protein here. Look very much like it. This protein, the membrane protein, is totally insoluble in water. So nature has made something water soluble, look like something look like it, but water insoluble. So my question then was how to convert this one, how to convert this one to this one. That was the question, yeah? So in textbook by Carl Brandin and Banks' friend, and in the textbook on page 17 has this figure. So this figure shows the green part is all the amino acids are not water soluble. And the red and blue are water soluble. The structure, they are identical. Like your hand, with the naked hand, or you have a pair of very thin doctor's glove. So looks exactly the same. But one is hydrophilic with your hand, one the other hand hydrophobic, one with the rubber gloves on. The shape is the same. So that's the key for, again, this idea that people have seen for how many, 50, 60 years, but to see, not to observe. So be observe, okay? Here, again, this is another one. The crystal structure of individual amino acids, look, some of them are very, very similar. One is not hydrophobic, the T, other one is hydrophobic, V. The shape is indistinguishable. In fact, the protein tRNA synthetase making charge, the amino acid, making mistakes. 1 per 200 or 1 per 400 making mistakes. You can say bottom first 6 of those also quite similar as the shape. Okay, is this kind of idea looking at the details? Devil is in details, or God is in details, right? So this kind of comparison, you can see that's how to generate the code. So another details is following. This red is oxygen, blue is hydrogen. On the left part, the single side chain of amino acid can form 4 hydrogen bonds, 4 water molecules. So make this amino acids soluble. Other one, you can form 3 hydrogen bonds, also soluble. But if you take oxygen away, the blue and the red one on the link to the green, the whole thing becomes insoluble. So that's how we got the idea to change this. So Karl Gajicek, he said it's important to explore to do things others ignore, but that will become important in 10 to 20 years. I told Mandelbrot spent 20 years working on this, right? Well, his formula. So I only spent 8 years. So I don't think it's necessary. So these questions is not so important for the high school student. We can escape this. So after 8 years, we published a single paper. So if I've been in other universities, in Shanghai, in Beijing, in China, in Hong Kong, in Europe, I would have been fired. Because 8 years to not publish a paper is a death sentence in academia. Publish or perish. Yeah? But Media Lab here allow us to explore something, ideas other people cannot afford. So idea is simple. Here you can see the shapes are very similar. Now we take all the green part, change into blue. So water insoluble on the right part, the green one, become water soluble, total based on the shape, the amino acid shape. Okay? So we used 4 different proteins to as guinea pigs. If you only do 1 protein, nobody believe you. If you do 2, maybe say maybe. If buy 3 or 4, then people cannot say maybe anymore. Just buy a lottery ticket. You buy 1 lottery ticket, right? You win lottery, he said, wow, right? But sometimes you don't buy. You buy 2nd lottery, you win again. What's the chance? You buy 3, you win again. Buy 4 tickets, you win 4 times. It's impossible, right? So you have to convince others. You have too many to convince them. So this is scientific part showing before on the left part. The protein has 7, that's called 7 fingers, 7 transmembrane helices before the design. Then after design, it's gone. So become water soluble. We showed the protein stable with all the bumps is here, shows precisely then have the folded structures similar to the native one. It can from some dimers, monomer, just what you observe in nature. It's also stable. You can see the alpha helical structure still maintained, not destroyed. So that's exactly what we want. That's what design, power of design. Yeah? The outcome should be similar. Finally, the function protein remain very, very similar. So we did not destroy the protein. Just a design, you want to design a nice Swiss watch, right? You design the outcome before you design production, the watch should be what you wanted. That's exactly what we wanted. Shows by the scientific terms, we call it the ligand binding. The affinity is very similar. So we 1, 2, 3, 4. Okay, we did it 4 times. So we did it 5 times. So it's not a luck. It's a design. Okay. Furthermore, we got also engineering as a design. We want to put 2 different molecules into 1. We can eventually make a sensor use this protein, makes totally different use. Put the antibodies leg onto the G protein coupled receptors on the receptors. Nature never made them. I made them, right? Just nature never made a computer either. Human made them. Nature never made a telephone for you. You made it. Okay? So here shows the very similar. The purple is one is come from the nature made. The teal color I made, I designed. Look, they're very much similar. And the green one, nature made, and the blue one, I made. So pretty good. So they also explain so why they're stable by so many internal hydrogen bonds in them. So as he explained, the 1 penny, you could not buy anything. It's almost useless these days. But you have 1 million penny. Yeah, 1 million penny, you can buy a lot of things, right? So like DNA code, A and T, G and C is reversible. You know one side, you know other side. So here I made a QTY code, also reversible. You have QTY, LVI, F on the bottom. Q can replace with L, T with V and I, and Y with F. It's reversible. That's power of the code. Finally, it's Alexander Rich that inspired me to pursue this work. And he frequently asks the question, why not? Right? These 2 words are very useful because it means you have an open mind. Why not? Right? When you go have pizza or go have pasta. You say, if you're so fixed and you cannot find an Italian restaurant and have a pasta and a pizza same place. So you have an open mind, you can say, why not? That's what Alex Rich frequently says that. Okay, so this person is have an open mind and produce so many things we take for granted. Thank you very much.
Good morning, everyone. Thank you, Shugang. I think this is a perfect example of an inspired person giving an inspirational talk. And I have a comment to Shugang's presentation here. So there's a saying that luck favors the preferred mind.
Yes.
And he knows enough to see when something great is happening, even though it wasn't by design. It was a serendipity discovery
right.
He was talking about today, and we heard about serendipities yesterday too. So keep learning and keep looking for good things when they come, because they do come along if you search for them, wait for them. Do you have any questions for Shugang? Welcome. In the back?
Always ask questions, big and small.
It's a so great achievement to use QTY code to design so many proteins. So I wonder, you have worked on GPCR. What's the next family of protein you are interested to design use QTY code?
Yes, and in fact, several people here from our group use QTY code to design other proteins. Turn out, it's not just useful for membrane proteins. It's also useful for other proteins. After the paper published, somebody got inspired in Shanghai, and then working on the protein that was water insoluble, hard to express. And after use applied QTY code is the enzyme. The protein becomes water soluble, maintains enzyme function. So I believe this code can be useful for many other proteins, perhaps including many neurodegenerative disease protein involved, Parkinson's, Alzheimer's, and so on. And so far, it's extremely difficult to generate monoclonal antibodies with those proteins, but you can change them into water-soluble version, if they keep the structure, they have the same function. That could be very useful. Yeah. By the way, that person who asked the question published a paper in August, and he also did this convert the water-insoluble protein into soluble form in the alpha helical structure. He was from UCSF, now at MGH.
Any
student?
There, the student. Yeah. Please say your name also and where you come from, what school.
My name and what school? So I'm Jennifer Santana, and I come from Notre Dame. And I was just wondering if the research that you've made is open to the public for us to see, or if it's something that your lab sort of has, like, not stored away, but kept until they'd have, like, solid information, or is all of the research just kind of open to the public?
Once a paper gets published, it's on the internet. It's also on a website. It's mostly on the internet. So everybody can have access to those information. Those academia then are obligated to put the information for everybody to see, because most research is funded by your taxpayers' money. So it's important to get the information out. Other people can use it and make their own inventions as well. My wife, Dory, she made my website, moleculararchitecture.org.
We would have time for one more question there, please.
Hi, I'm Wilson Chu. I'm coming from Hong Kong. How can protein be linked up to electronic circuits, as you mentioned, in potential sensor applications?
Yes, very good question. The best sensor in our body is nose, right? You can distinguish millions of different flavors and in a fraction of seconds, and don't make mistakes. However, protein are very fragile once taken out from the membrane, as we mentioned, especially olfactory receptors. So far, despite all the years' efforts and $100 million spent, not a single person has obtained olfactory receptor structure. However, it should not deter us to work on something. Once we can use QTY code to make protein become soluble, you can then couple the protein with graphene. Graphene is carbon-based material. It's conducting material. It is known when since compound graphene, something interfere with that, the conducting material can have interference. So it could be very useful. So that's why we're working on the next crazy ideas. Very good question. Imagine sometime you can have called smart bathroom. It's detect early detection of disease. You walk in because you go there every day, and then you can detect some disease early. But you need the sensors. The sensors detector part of the sensor is most difficult to work out. Electronic sensors has very limited capacity or selectivity, and have very little diversity, but sometimes very sensitive, but they cannot detect a wide range sense, like what we now know. Very good question. Yeah.
So I think we have
Good.
to move on now. Thank you so much, Shuguang.
Yeah.
And now we're fortunate to have Li Wei Chao here. Dr. Chai is a director of the Picor Institute for Learning and Memory and a professor of neuroscience and a senior member of the Broad Institute here at MIT and Harvard. And she will talk on unique visual stimulation, maybe the new treatment for Alzheimer's. Very exciting topic, and very welcome, and please.
Thank you. Good morning, everyone. I'm very pleased to be here and share with you our new approach to treat Alzheimer's disease. It's going to be a very different talk than the previous one. So, yeah. So, just a little bit about myself. I'm a neuroscientist, and I'm a professor and director of the Picor Institute for Learning and Memory, which is part of MIT's neuroscience complex. My God, what happened?
Enter your code, please.
Can I just?
You should be all set to go.
Are you going to switch? Well, it's okay. Actually, I can't really. I just put it here. Okay, so I think I'm ready to go. So as you can tell, my approach is highly unconventional. So I'm going to tell you how to use light and sound to treat Alzheimer's disease. And this is just just disclosure that we license our technology to Cognito Therapeutics, so they can bring the technology perhaps to more people who might benefit from it. So, just a few words about why we care about dementia. So, as you know, that the world is aging, with many more diseases that can be managed, such as cardiovascular disease or cancer. So people enjoy a longer lifespan. However, that also means that age-related diseases such as neurodegenerative diseases are rising sharply. So today, 46.8 million of people live with dementia. And this number is expected to triple in the next 3 decades. And this is happening in every single continent, okay? And Alzheimer's disease account for about 80% of all dementia cases. So what is Alzheimer's disease? Alzheimer's disease is not normal aging. And the disease takes many, many years to develop, at least 20 years now, people believe. And obviously, you all know people suffering from Alzheimer's disease have memory problems. And eventually, they develop dementia and they need full-time care. So this is really a huge socio-economical problem facing the society. And the feature of the disease is really defined by the two pathological hallmarks known as amyloid plaques, which are extracellular deposition of small amyloid peptides. And they are very small. And the major species in the brain are so-called A-beta 40 and A-beta 42 peptides. So there are 40 or 42 amino acid residues in size. And then also the brain show very abundant so-called neurofibrillary tangles. And the tangles are intracellular aggregation of the hyperphosphorylated, so this is chemically modified tau protein. And tau protein is believed to normally be important to maintain the integrity of your brain cells. So in addition to the amyloid and the tangles, the brain also show tremendous loss of volume, and this is believed to be mainly caused by loss of neurons, so your brain cells die, and the size is tremendously atrophied, and also the synapses. So the synapses are known to be the little connections that are between the brain cells, and they are super important for the communication and for information processing of your brain cells. And so the synapses also are lost. And finally, there is a very conspicuous inflammatory response that's going on in the brain as well. Okay? So for many, many years, decades even, the field, really the hypothesis that explain Alzheimer's disease is dominated by the so-called amyloid hypothesis. So it is believed that it is the initial accumulation of the small amyloid peptides that triggered cascades of events that eventually leading to development of this other pathology as well as dementia. And so far, in terms of therapeutics, it has been very, very depressing. So there are altogether 4 drugs that were approved for treating Alzheimer's disease. And these drugs provide very temporary relief of the symptoms, but only for like a few months. And none of these drugs really change the course of the disease. The disease continues to progress. So there has been a lot of effort in the pharmaceutical industry to identify better drugs that can alter the course of the disease, okay? So in the last 15 years, we've seen many, many new drugs that brought to clinical trials, human trials, but every single one of them failed. And majority of these drugs, they were designed according to the amyloid hypothesis. So they were designed to reduce amyloid. And, you know, I mean, the hope is not completely lost yet. I think, Biogen, IDAC, for instance, in Cambridge, they recently show some very promising news about their new anti-amyloid drugs, show potential. And we are still waiting for their late phase human trials result to see whether that drug will be eventually approved. But suffice to say that I think now everybody gets a sense that we really need to think more broadly when it comes to coming up with effective approaches to either treat the symptoms of Alzheimer's disease or to alter the course of the disease. So one thing that my lab and my collaborators, such as Ed Boyden, that you heard his presentation yesterday, are thinking about is really that, so far all of these drug trials are really all based on genes and molecules. So in the last 3 decades, many genes have been identified that seem to play a role in the etiology of the disease, and also a lot of molecules that people want to reduce and target to develop so-called small molecule chemical drugs. But little attention was paid to think about the brain as a network and as a circuit, because we have billions of brain cells, okay? Many of the neurons, but there are also other supporting cells. And as I mentioned before, these neurons form trillions of synapses with each other to really ensure a very efficient and rapid processing of information from region to another, such as, when I touch something, my brain immediately knows what I'm touching, because this is the information travels all the way from the tips of my finger to my prefrontal cortex, and I can interpret what this is. Okay, so this is super important. This is so-called the function of the brain. I mean, in a way, it's not that different from, say, a computer motherboard, right? But people don't really think that way. At least not people in the drug discovery industry. So we started to think about, what has gone wrong in Alzheimer's disease, especially in the very, very early stage, probably before you can even detect any impairment in your behavior, in your cognitive capacity. What's wrong with, information processing in the brain? So I'm going to tell you today mainly about a very particular type of neural activity known as rhythms or oscillations. So what does that mean is that when your brain cells, many connected brain cells, they fire together synchronously, then brain oscillations or rhythms are generated. And today in the mammalian cortex, there are at least 10 different classes of oscillations identified, and they cover at least 4 orders of magnitude in frequency, from the infra-slow to the ultra-fast. So from sub hertz to several hundred hertz. So among all of these different classes of oscillations, and I think you can appreciate, these different classes of oscillations are all very highly evolutionarily conserved, from very small mammals such as mouse and rats, or bat even, all the way to primates, including human brain. I mean, the frequency basically of these different classes of these oscillations, they're completely conserved, indicating that they play very important roles in fundamental brain functions. So, but today I'm going to particularly discuss one class of oscillation known as gamma oscillations. So these gamma oscillations, they are ranging between 30 to 80 hertz. So meaning 30 to 80 cycles per second. So it's kind of fast oscillations. And they are known to be associated with higher order brain functions such as working memory, sensory processing, spatial navigation. When you know you are trying to concentrate to solve a puzzle or you're trying to beat a deadline, the gamma oscillations become very active in your brain, okay? And in a publication from my lab in 2016, we raised this first possibility, which is to address, can boosting gamma rhythms can reverse pathology in mouse models of Alzheimer's disease, okay? And let me give you a little bit of background. How do we link gamma oscillations to Alzheimer's disease? So previously in the literature, it has already been shown that gamma oscillations are impaired in mouse models of Alzheimer's as well as in human subjects. Okay, so here are just some examples, a couple of publications in different Alzheimer's disease mouse models. In this publication from Leonard Milke and Jorge Polap's group at the UCSF, they use this particular mouse model of amyloid, and they show that gamma oscillation around 40 hertz is reduced. And this is associated with increased sort of like epilepsy kind of neural activity in the brain, which is very unstable, very dangerous. And also, these mice show cognitive impairment. And in another mouse model, lipoprotein 4, which is today the most significant genetic risk for people to have Alzheimer's disease. And this model also show reduced gamma oscillations. And in human patients, it's very interesting. So, as I told you, that this rapid and efficient information processing really requires synchrony among the different brain regions. So these different brain regions have to be in sync. So they found, so this group of investigators measure the synchrony among many, many different brain regions. Human brain is very huge, okay? So they measure the synchrony among all these different brain regions at this gamma frequency range. And they found that in general, Alzheimer's disease subjects show significant reduction compared to healthy subjects, age-matched. So we were very intrigued by this kind of observation. But I also told you that Alzheimer's disease in humans takes at least 20 years to develop, okay? So it is believed that this beta amyloid, I told you, accumulation happens very, very early on, okay? And this is dementia. So this is like a 20 years time scale. So we first see the accumulation of amyloid. And you can see, even when the amyloid already plateaued in the brain, there is still very little clinical symptoms that can be detected in these human subjects. Okay? So this amyloid accumulation is followed by synaptic loss, and then this tau tangle pathology starts to accumulate, and brain structure begins to reduce. And after all of this happened, you start to see mild cognitive impairment. So all the structural changes happened way earlier, okay? So, so we wanted to know when during this course of the disease, we see this gamma oscillations impairment. Because if it happens very early, then impaired gamma oscillation may contribute, may feed into the pathophysiology of the disease, but if we cannot detect it until the symptomatic stage, then this is merely the consequence of the disease, okay? So, so we decided to team up with Ed Boyden, my very dear colleague in our neuroscience building. So Ed, basically he has a lab here in the media lab, and he has a lab in the neuroscience community. And Annabelle Singer, who was a postdoctoral fellow in Ed's group before. So again, for this kind of temporal event, we initially started with the mouse model. So we decided to use this so-called 5-FAD amyloid model. So here you can see this brown sort of puncta. They represent this amyloid plaque deposition in the brain. So you can see in a 2-month-old animal, there is a little bit of deposition in the brain, and by 4 months there are more, and by 6 months there are even more. But here is the very key structure. This is so-called the hippocampus. And the hippocampus is known to be the center for memory formation, okay? And also plays a key role in memory recall, meaning to remember something. So in the hippocampus, there's really no amyloid pathology until about 6 months, where you start to see a little bit of amyloid. And this is the time point when this animal show memory deficits. So we decided to evaluate gamma in the much earlier stage, 3 months old. So when the brain relatively is cleared of plaque, especially not in the hippocampus, and there is no symptoms that can be detected. So this is an experiment that Annabelle did. So what she did was to train the 3-month-old 5-FAD mouse to run on a trackball, okay, through a virtual reality maze. She then can stick an electrode into the hippocampus of this mouse to record gamma oscillations when the mouse was running, but then the mouse would stop when there is a wall, say, okay, and the mouse would sort of think about what to do, and then he would turn the ball and then to find another path so that he could keep running. Okay, so we were able to record gamma oscillations when the mouse was running or when the mouse rests. So to cut a long story very short, we found that when the mouse was resting and trying to figure out strategy, then so this is so-called ultra-fast oscillation, and underneath there is this gamma oscillation centered around 40 hertz. And we found that in the very young amyloid mouse model compared to the littermate control, this is a heat map. The warmer the color, the higher the power of gamma. You can see that this gamma power of gamma is already reduced in this very young pre-symptomatic mouse. So we started to feel that this reduced gamma probably play a role. But what is it, right? So at the time we decided, rather than really just keep figuring out what does that mean, we said we should see what happens if we can somehow increase gamma in the brain of these animals. So the approach we used was to use optogenetics. So I'm sure that Ed Boyden told you about optogenetics, using light, blue light, to manipulate activity of neurons, okay? So, and it's called genetics because you can selectively introduce an opsin, a light channel, into the particular subpopulation of neurons you want to manipulate, right? So we previously collaborated with Jess Cardin and Chris Moore, published a paper in Nature in 2009, showing that if we use this optogenetics to particularly drive the so-called albumin positive fast spiking interneurons, then we can create gamma oscillations in the mouse brain. So we decided to use the same approach. We specifically targeted these albumin positive interneurons in the hippocampus, and then we introduced 40 hertz blue light to drive these neurons. So we will use 40 hertz laser pulses, and what we found that when we do electrophysiology, we can see the very sharp increase in the 40 hertz gamma power in these animals. But if we use other frequencies, such as we use a random frequency, so just random, it can be 1 hertz, or it can be 30 hertz, it can be 25 hertz, but average 40 hertz. then we cannot produce this kind of very sharp increase in gamma. So then the question is, then what? Now you increase gamma. What happened in the brain? So we saw the most dramatic result. So this work was done by my former graduate student, Hannah Alcarino. So we found when we use 40 hertz light pulses, then we see a drastic reduction of A beta peptides, both the 40 and 42 species that I told you they are the major species in the brain, that they constitute the amyloid plaques. And we use other frequencies such as lower frequency, 8 hertz, or the random frequency. They don't alter, or they don't reduce, at least, the level of this little beta amyloid peptides. So this was really very striking results that you can just manipulate this synchronous neural activity to control the level of beta amyloid. And this happened very fast, too. All she did was to stimulate these animals for 1 hour, and we can already see such dramatic reduction of amyloid. So we wanted to know, how does this happen? And so that involved a lot of experiments. I just want to use a couple of animation to sort of first briefly explain to you what we found. So normally, now this brown, this little stuff, we use to represent amyloid. So normally, these amyloid peptides are mainly produced by neurons. Okay, it's a physiological phenomenon. They just produce amyloid. That's what they do. But we found that after we stimulate them with this optogenetic stimulation at 40 hertz, this production of this amyloid is reduced by these neurons. And this is pretty exciting. But this is not the end of the story. What we found something else very dramatic is another brain cell type that responded to gamma oscillation very dramatically. So these are known as microglia, depicted by these green cells. So microglia are immune cells, actually. They are brain's immune cells. And their normal job is they constantly survey the environment looking for foreign substances such as pathogen, like a virus or bacteria. They will immediately engulf this pathogen, okay, to protect the neurons. And they also normally eat up any toxic metabolite from neurons, such as amyloid. That's their normal job. But in Alzheimer's, it is well documented that these microglia, they are no longer active. They don't do their job anymore. Okay? But what we found is that this gamma oscillation changed the property of this microglia in a very profound way, that not only their size increases, but they become more active, engulfing this amyloid to clear it up from the brain milieu. So I'll just give you a piece of real data so you can appreciate how dramatic this is. So we use a specific antibody to label microglia in the brain sections of mice following controlled stimulation or 40 hertz stimulation or random stimulation. And I think you can appreciate, right? These microglia, they look very different from microglia here or microglia here. And so we wanted to know exactly in what way these microglia are different. So it turned out there are just more microglia, first of all. And then their cell body is very significantly increased. So they are just larger now. Moreover, these microglia, also many more of them have this A beta peptides in the belly, okay? Meaning they eat up more A beta. So this is a very dramatic increase from about 35% to about 90% after stimulation. So almost all of these microglia become functional again. So far, what I presented to you painted a picture that potentially one can just manipulate the synchronous activity of the brain cells to alter the pathogenic molecules that may underlie Alzheimer's disease. However, as pointed out by my colleague, Professor Emery Brown, he said optogenetics is very invasive. You know, I told you, you have to use a virus to introduce this light channel into the brain, and then you have to insert a fiber optics to introduce light into the deep part of your brain. How can you do that to humans, right? So Emery said, you got to figure out a way to non-invasively introduce gamma to the brain. So how do we do that? So I really appreciate Emery because, you know, he and me and my team members, you know, we pore over literature looking for any precedent that people have tried this kind of thing. And eventually we actually found some old literature. A German neuroscientist group, they shined a light to cats. And then they did recordings in the cat's visual cortex, part of the brain processing vision. And they found that the cat can actually respond to the light that you present to them. And their visual cortex will fire according to the frequency of the light flickers you present to them. So we say, well, you know, I don't know whether I believe it, but, you know, it doesn't hurt to give it a shot. So we came up with this approach. So Hannah purchased some LED light strip from Amazon, and she surrounded the mouse cage with this LED light strip. She then used this very simple Arduino system to drive this LED light strip in different frequencies. Okay, so then she presented the mouse with this LED lights, and then, you know, we found out we can totally reproduce the previous literature. So when we shine the light at 40 hertz to this mouse, we then recorded the neural activity in the visual cortex, as I said, the part of the brain processing vision. Then we see this very similar sharp increase in the 40 hertz gamma power. Okay, again, this is very specific to the 40 hertz flicker because if we present it at random frequency, we don't see this kind of increase in gamma power. And this is additional recording, so-called multi-unit recording, to show that now you can see this firing pattern of this neuron become very periodical at 40 hertz. So basically the brain can be trained to fire at particular frequency based on your sensory visual input. So we call this approach gamma entrainment using sensory stimuli, or genus. So now the question is whether genus can also reduce amyloid in the brain. So Hannah did the following experiment. Again, she exposed the mice to the different frequencies of light flicker for 1 hour. And then afterwards, she dissected out this visual cortices and then look at the amyloid levels in the brain. And I think this is a very beautiful experimental results. As you can appreciate, with 40 hertz light flicker, you see again about 50% reduction of A beta 40 and A beta 42. And this is not reproduced with constant dark, constant light, or 20 hertz or 80 hertz, or random frequency stimulation. So far, I show you that with sensory entrainment, here with visual light flicker, one can somehow recruit the local circuitry in the part of the brain, in the visual cortex, to produce oscillations. And this neural oscillatory activity, again, somehow can recruit neurons to respond, microglia to respond, and this led to reduced amyloid levels and perhaps can restore neuronal health. So after this, looking at this very remarkable effect of just very non-invasive sensory stimulation. What we would like to know now is, can the effect of genus propagate to regions beyond the primary sensory cortex? Because in Alzheimer's disease, extensive regions of the brain are affected. It's definitely the sensory cortices, like the part of the brain responsible for processing vision or sound or smell. They are actually, affected very, very late in the whole schema of the disease. Okay, so you do want to know whether the part of the brain more responsible for learning and memory and executive function, working memory, they can benefit from this kind of sensory stimulation. And also, what are the circuit and molecular mechanisms mediating the beneficial effect of genus in neurons and microglia? And can genus be mediated by other sensory modalities, such as, beyond vision? What about sound? What about, other way to introduce gamma? So I'm going to show you a few pieces of our data that in the pipeline but have not been published yet. So this is an experiment to show that turned out that using visual stimulation, gamma oscillation actually is not only detected in the primary visual cortex. You can see, again, this is a spectrogram. Again, you look at the heat map, at the warmer of the color, the higher power of gamma oscillations. And we can clearly see this very sharp increase of 40 hertz when we present the 40 hertz light flicker to the mice. But we also see increase in gamma power in other sensory cortex beyond the visual cortex, and even in the hippocampus and in the prefrontal cortex. That means when this visual flicker comes in, and my visual cortex in the back of the brain, in humans, it's here, become recruited to produce gamma oscillations. But then this oscillation can travel from the visual cortex to hippocampus, to somatosensory cortex, and all the way to the prefrontal cortex. And furthermore, we also look at synchrony among these different brain regions, okay? So-called co-activation. And we found, this is, again, the index of synchrony. We found increased synchrony between the visual cortex with somatosensory cortex, the visual cortex with hippocampus, and even visual cortex with the prefrontal cortex. So this means these brain regions are fired in synchrony. There is better, more efficient connectivity among these brain regions. There is more efficient processing of information. So what's the big deal about propagation of these gamma oscillations to the rest of the brain region? It has huge implications, actually, in terms of the pathology. So here, we go back to this 5-SFAD amyloid model, but instead of 3 months old, now we are looking at the effect in 9 months old. So by 9 months old, this animal develop this plaque here is this red puncta. These are amyloid plaques. You can see they're very abundant throughout all the different regions of the mouse cortex and also in the hippocampus. Okay? So we expose the mice to light flicker 1 hour per day for a total of 22 days. Okay, so if we just expose for 1 day, 1 hour, we don't really see a reduction. But when we do this repeatedly for 22 days, this is what we see. We see a general reduction of the load of this amyloid in multiple brain regions, also in the hippocampus. Okay, this is a quantification of the experiment. Okay, you can see reduction of plaques in all the different regions. Moreover, we found that this treatment actually tremendously restored the ability of the mice to learn and to remember. So, so there is a battery of different mouse behavioral tasks that one can use to test different kinds of memory in the mice. And I'm going to give you an example. So this is a well-established so-called Morris water maze to test the spatial memory capacity of the animals. So we place the mouse in a large swimming pool, and we kind of muddy the water with some sort of white paint. So when the animal's in the water, you know, he can't really see what's in the water, okay? So we place the mouse in the water, and then somewhere we put a platform which is submerged underneath the surface of the water. So the mouse can't see where the platform is. And the mouse naturally doesn't like to be in a cold water, okay? They don't like to swim, although they can. They don't like it. So they will swim around, and once they found the platform, it will climb to the platform and no longer swim in the water, okay? So we basically train the mice to remember where that platform is. And in the room, in the testing room, we also place some sort of, some sort of patterns to help them remember what's their location. So one can put a mouse in the swimming pool and, train them every day for a total of several days. And usually each day is composed of 3 or 4 training sessions. And you can see in the first day, the mouse would take a long time to locate the platform, but usually with each day of training, they start to kind of get a hang of it and start to remember where the platform is, okay? So you can see a normal mouse, it would just, no problem identify the platform with each day of training. But with this Alzheimer's mice, they have trouble. Remember, usually it would take them always longer to find the platform. And then at the conclusion of the training, we can wait for a day, and then we would put them back to the water, and usually we will remove the platform. We kind of play trick with them. And then see how long the mouse would spend time in the area where the platform used to be, okay? Sort of a very quantitative way to measure how well they remember. So this is called a probe trial. 5 minutes, okay. So you can see these Alzheimer's mice show really very severe impairment, but following 22 days of treatment, the spatial memory capacity is basically completely restored. Okay, and I'm going to show you 2 more sort of pathology slides, and I'll try to stop here. So, as I told you in the very beginning, Alzheimer is not just about amyloid, right? There are neurofibrillary tangles as well, and there is real loss of your brain cells. So we use another mouse model that's known as the tangle mouse model. This is so-called tau P1S mouse. They develop neurofibrillary tangles, and here you can see this green staining. The cells show this very dense green kind of thing. These are tangles, okay? And again, the same thing. 8-month-old mice, we treated them 1 hour per day for a total of 22 days. And really, I really didn't expect to see anything like this. But what we see is an almost complete reversal of this tau tangle pathology in all of these different brain regions. So this blue bar represents the tangle in the wild-type mice. Basically, this mice never developed tangle. There's no pathology. And after 22 days of treatment, in many of the brain regions, there's no tangle that can be detected. This reduction of tangle seems to be even more complete than the reduction of the amyloid. And I told you there is loss of neurons. So in this particular mouse model, they actually show loss of neurons. You can see these magenta cells. These are neurons. The density is reduced in the group without stimulation. But with the stimulation, you can see the density is kind of restored. So this treatment is actually neuroprotective. And this is even more obvious with a third mouse model that we used. This is known as the CKP25 mice. They develop very profound degeneration. You can see grossly the brain size is reduced. And the lateral ventricles very much expand, because when your brain mass is reduced, then it's filled with this water in the brain. But in this mice, we treated for a total of 6 weeks, and you can see the brain mass restored, the ventricles totally reduced, and the thickness of the brain is restored. So basically, this is saying that just with this light flicker stimulation, one can prevent neurodegeneration from happening. So I think I'm gonna stop my talk here, just to give you a very promising outcome or a perspective. So here at MIT, we started to bring this non-invasive method to humans, okay? Because there's no downside. A lot of people told us this doesn't cause discomfort. And we try on a lot of human beings, and nobody complained that they even get headache. Okay, so just want you to know, we use a large LED light panel and present it to a human subject, and then we use EEG. So this is an EEG cap, so we can detect brain rhythms in all the different parts of the brain. Okay, so again, this is a heat map of your brain. Okay, this is the visual cortex in the back. This is the prefrontal cortex. And we look at gamma oscillations in the baseline. And then once we turn on this visual flicker, and you can see gamma oscillation tremendously increase in the visual cortex, but also in the prefrontal cortex. So the propagation of gamma happens in mice, but it also works extremely well in human subjects. Okay, I'm gonna stop here. And if you have any questions, I'm happy to entertain.
Thank you very much. Thanks for a very spectacular research presentation. It was very, very interesting stuff. We have many questions, I see. I think we take a few. Okay, let's have the one in the back there, the student.
There.
Okay.
Thanks for the talk. Very fascinating findings. And I was wondering, what if the patient or the mice are blind, so they cannot sense the light? And
yeah, that would be very unfortunate, right? So okay. So I didn't have time to talk about this, but I will go back since this came up in question. So precisely, right? Especially in older population, right? A lot of people's sensory function also declined. But you know, as an animal, as a human, can you turn on the slide again? I will appreciate it. So we can touch, we can see, we can taste, we can smell, and we can hear. So we are really asking whether these other sensory modalities can mediate gamma oscillations. So I show you visual stimulation works, but then we tested whether auditory gamma can induce gamma oscillations as well. So again, we collaborated with Ed Boyden and his graduate student, and Annabelle Singer, who is now a PI, a principal investigator, assistant professor at Georgia Tech. So we came up with an auditory gamma approach, and just sort of, you can hear it maybe. Yeah, it's not loud enough. But anyway, we use this auditory gamma tone. Yeah, I can hear it. So this is designed specific for mouse, but we also designed some gamma tone for humans as well, and it seems to work in humans. And this is just to show you that this auditory gamma tone works extremely well to train the neurons in the auditory cortex in the hippocampus to fire in 40 Hertz synchrony.
Okay, we'll take a question from the students there. That's close. You've asked too many questions. Sorry. Submit your question online. We can take them in the afternoon.
My question is if Janice could work on any other type of brain diseases and not
That's a very good question. So a lot of people ask me the same question. And I would think that it might, because there are other neurological disorders with gamma impairment implicated, such as schizophrenia and epilepsy. So I think that people are looking into this possibility, but it's a very, very good question.
Okay, okay, let's take the one in the back, the lady in the back. Sorry, we have to cut it after that, I think, and then save the rest for this afternoon's panel.
Thanks for the great presentation. And I have one question. Why is gamma oscillation decrease the level of A beta or tau? I mean, is there possible explanations such as maybe it activates the microglia?
Yes, I think activation of microglia play a very important role, but I think that actually it's not just microglia. We recently have also evidence that the blood vessels in the brain, they also become activated. So we can see that the lumen of the blood vessel dilated, and more A beta can be sucked into the blood vessel and circulated out of the brain into the peripheral, like livers, to be degraded. So there are multiple mechanisms at work, I think.
Thank you.
Will you be here for the panel in the afternoon?
What time?
Between 5 and 6.
I have another presentation. Thank you.
Let's take one more question, and the gentleman there. Okay.
Okay,
Yeah.
thank you. I have a quick question. How do you think so many people, so many companies failed in developing drug for the A beta amino? Yeah.
Why do so many people develop A beta?
Why? How do you think so many companies failed?
Because really, right now, it's still, the major hypothesis is still the amyloid hypothesis, and no one else come up really with the alternative hypothesis. So that is a hypothesis that, people would like to explain how, Alzheimer's disease, develops. So, but I think that I can see that people started to think about other targets, such as develop treatment against brain inflammation and to increase the function of microglia and to remove tau proteins. So I think there are now quite a few different new approaches, but it's going to take quite a while for all of these new approaches to go into clinical trials.
So let's take the last question. The gentleman there. Yes, please.
Very exciting and elegant talk.
Thank you.
In the medical world, when we encounter patients with dementia, we really want to distinguish whether it has Alzheimer's disease versus, let's say, multi-infarct dementia. So in multi-infarct dementia, we know that there's no amyloid or those tangles. I was wondering, in this dementia situation, do you see a gamma, oscillation
Reduction?
Yes.
Yeah, I don't know whether people have looked. I think it will be interesting. But I think, for instance, right now, usually when people, enroll patients for treatment, they first do an amyloid PET scan to screen for people who truly have Alzheimer's disease versus other type of dementia. So, so I think now, it is a good development that people start to try to stratify patients' population, and hopefully this will lead to more efficient drug treatment.
another very curious point is that when you show the inactivation or reducing number of microglia in Alzheimer's situation, we know that in cancer, like glioma, there's an activation and increase of microglia. I was wondering to see if the reverse with increase in the gamma oscillation in cancer cases.
We actually have done, kind of transcriptomic analysis of microglia following gamma treatment. And so we actually see a reduced inflammatory profile. So I think this is a different kind of activation of microglia because, microglia or macrophages can, display this alternative activation state. We think that this is more beneficial activation state. Okay.
Okay, thank you so much. And let's take a break and resume in 15 minutes.
Okay. Hi, Chris. Oh, okay. It's much more.
Do you prefer to use a lapel microphone or use the podium microphone?
Either one's fine. Whichever you think is better.
It's up to you. If you do a lot of turning.
anyone try that?
Well, it's maybe not so pleasant, but it's not dangerous.
Really?
Oh, it depends on the altitude. Yes, it's like looking. I can calculate on what altitude you need. I mean, if it's but there's also another
I don't know. So let's take our seats. So we'll follow up on the theme of neuroscience and welcome Dr. Feng Zhang, who is a professor of neuroscience here at MIT in the departments of brain and cognition sciences, and he's also a member of the Broad Institute. His talk has the title Advances in Genome Editing. So welcome, Feng.
Okay. All right. Good morning. Thank you so much for the opportunity to present here. Wanted to give you an update on some of the things we've been working on in the area of genome editing. So by way of getting started, one of the really exciting advances in biology has been the completion of the human genome. And now the cost for sequencing DNA has dropped quite a bit. So even though the first human genome costed hundreds of millions of dollars to complete, now we can sequence individual genomes for $1,200 or maybe even less. And so that opens up the opportunity to sequence many, many genomes. And so now there are large projects internationally that are sequencing 100,000 or 500,000, maybe even more individual genomes. And so through a lot of efforts like this, we're now able to compare genomes across individuals, either individuals who are affected by certain diseases versus healthy individuals, and try to identify genetic differences that may underlie disease. And so from that, we already have learned some things that are harmful. So for example, certain mutations can affect sickle cell disease or cystic fibrosis or degeneration of the eye. And so to date, there are over 6,000 genetic differences that have been identified that underlie disease, and some of these are called monogenic diseases. But not all genetic differences are necessarily harmful. And so in fact, some can even confer health benefits, reduce disease risk. So some examples of this include mutations in the gene called CCR5, which can confer resistance to HIV, or different variants of the ApoE, or a cellular carrier protein that can alter disease risk for Alzheimer's disease or diabetes. So you might be sitting there now and thinking that if we know what mutations are beneficial and which ones are harmful, why don't we go into the genome and just get rid of the bad ones and put in the ones that are beneficial? So if we wanted to do that, then we'll have to be able to change our DNA. And so this very idea, or this tantalizing idea of being able to change DNA, has been worked on by many scientists over the past couple of decades. And one of the main ways for making changes in the genome relies on a discovery that was made by Jim Haber and also Maria Jason more than 2 decades ago. And that centers around the idea that breaks in the DNA can stimulate repair processes and to be able to effect precise and also efficient modifications to the genome. And so this picture here highlights the basic way through which gene editing is practiced now. So by using nucleases that can be localized to specific locations on the genome, we can introduce double-strand breaks in the DNA and to be able to trigger either non-homologous injoining or homology-directed repair processes to be able to make specific changes to the DNA. And so this idea is very nice, but one of the main challenges has to do with how do you easily reprogram this nuclease to target specific sequences. So it really revolves around the idea of how do you build a DNA binding domain that can work efficiently and also be easy to reprogram. So this very question has been worked on through multiple different approaches. Some of the earliest approach include an enzyme system called meganuclease, and this is an enzyme that can recognize somewhere between 18 to 20 bases of DNA, and you can engineer this enzyme to some extent to get it to recognize and also cleave novel DNA sequences. But it's very, very hard to be able to reprogram it. So then some of the subsequent technologies involve the use of zinc finger proteins, which are proteins that come in specific modules. One module recognizes 3 DNA bases, and if you have a chain of 3 modules, then you can recognize 9 bases, or 6 modules can recognize 18 bases. And so that's what is shown over here. Zinc fingers really demonstrate the idea that if we can reprogram proteins to target sequences, then we can build nucleases that can cleave the genome and stimulate repair. But zinc fingers have also been remarkably difficult to work with. So in fact, even for someone who has a lot of know-how, it can take them several months. And even then, you're not guaranteed to make something that works effectively. And so then another technology came along, and this is something that we played a role in, which is called the tail technology. And this is a different DNA binding protein found in microbes. And these proteins use modules as well, but these modules recognize individual DNA bases. And so you can also link them together to form what we call a tail array to be able to recognize a long stretch of DNA sequence. So despite being easier than zinc fingers, tail systems are still quite difficult to work with. They are highly repetitive, which makes the engineering and assembly of customized arrays quite cumbersome. And so as we were working on these, I thought, could there be easier solutions through which we can develop a nuclease that can much more easily and also more efficiently reprogram to target a novel DNA sequence? And so one of the answers to this question lied in a different area, which is a bacterial immune system called CRISPR. CRISPR is an immune system in bacteria. It's adaptive, so that means it can remember previous infections and to be able to mount defense against viruses that it has encountered before. The way this works is that CRISPR is, so imagine this gray box being a bacterial cell. In nature, this bacterial cell can get invaded by all sorts of viruses that are called phages. And the first thing the phage does upon infecting that bacterial cell is to inject its DNA or RNA genome into the cell. So once it's inside, the CRISPR system acts as a surveillance system of the bacterial cell, and it can go and try to see whether or not it recognizes that virus. So the CRISPR system consists of proteins, which are called Cas proteins, are encoded by Cas genes in the CRISPR locus of that bacteria's genome. And then there are these repetitive but non-coding sequences that are nearby. And in fact, these form the hallmarks of the CRISPR systems. They are repetitive, and so each one of these rectangles here is one of these repeats. They're identical to each other. And they are interspaced by short stretches, usually about 30 base pairs long, of non-repetitive sequences. And these sequences, called spacers, are derived from previous viruses. And so they are the memories of previous infection. And so these CRISPR arrays can get transcribed into RNA. And initially it's transcribed as one long RNA, but then it's processed by different enzymes into short individual RNAs, each one carrying one spacer, and we call these guide sequences. So the RNA and the protein can work together to form a protein-RNA complex that can then survey the bacterial viral sequence and see whether or not the RNA matches the invading virus's DNA. And if the RNA matches, then the enzyme gets activated, and if serves as a nuclease, it can cleave the virus DNA and then circumvent infection. And so this is how the immunity is achieved by the CRISPR system. So it's this RNA-guided recognition mechanism that has been harnessed to develop genome editing technologies. And so one of the most widely used ways for gene editing is to take these CRISPR proteins, engineer them so that they can work in human cells or eukaryotic cells, and then reprogramming them with the RNA that has been designed to target a specific sequence in the mammalian cell's genome or the eukaryotic cell's genome. And so by introducing this into the cell, then this enzyme can go and search along the genomic DNA until where the RNA matches the DNA. And then the enzyme will get activated, cleave the genomic DNA, and then stimulate either non-homologous injoining or homology-directed repair. So here's a short video that kind of summarizes what I just blabbered about. And so the blue glob here is the Cas9 protein. This is one of many different types of CRISPR proteins. And you can program it with the RNA guide shown here in red. So if you engineer this and introduce that into the human cell, you can go into the nucleus, search along the human genomic DNA, which is here in blue. And when the RNA recognizes the DNA by formation of a Watson-Crick base pairing, the enzyme will get activated and then cleave the DNA into 2 broken ends. This DNA double-strand break is the equivalent of a cursor in Microsoft Word. So wherever you place it in the genome, that's where gene editing happens. And so the first way this gets repaired is called non-homologous injoining, and simply the 2 broken ends are rejoined together. But a small mutation is introduced in the process of that repair. And that mutation can inactivate a gene. So if you want to get rid of a deleterious protein, you can use that to be able to inactivate knockout gene function. The second repair pathway is more powerful. So you can take advantage of these 2 broken ends and at the same time provide a piece of synthetic DNA where the 2 ends of that synthetic DNA is homologous to the 2 broken DNA ends. So that whatever the sequence is flanked by those 2 homology arms can now get incorporated into a specific location in the human genome. And so this is called homologous recombination. And by introducing a double-strand break, we are able to increase the efficiency of this process by multiple orders of magnitude. And so this is the basic way through which the RNA-guided nucleases have been used for gene editing. But these CRISPR enzymes are much more useful beyond just doing gene editing, cleaving DNA. In fact, the DNA cleavage is mediated by these 2 nuclease domains called RUBC and also HNH. You can introduce point mutations into these 2 catalytic domains to inactivate the cleavage activity, but preserve the RNA-guided DNA binding activity. So then you can turn it into a RNA-guided homing device. So wherever you want to home to in the genome, you can use this to home to. And so that opens up the opportunity to then tether different effector modules to this homing device. So these effector proteins can do a number of different things. If you want to turn genes on, you can bring a transcription activator over to a specific gene and activate the expression. If you want to repress it, you can bring a HDAC complex into that location to condense the chromatin and silence the gene. And you can visualize the dynamics of the genome by fusing GFP to it, so you can monitor how the genome reorients itself inside of a cell. Or you can recruit other catalytic functions like silent deaminases to be able to modify cytosines into uracils or adenosines into inosines. And so many different ways to use this RNA-guided DNA recognition function to be able to perturb and also to study biology. So what I thought, so there's a lot of work that has been done in this Cas9 enzyme, but what I thought I would tell you about today are some of the work we've been doing to explore the CRISPR diversity, because there's much more out there in the CRISPR systems beyond this enzyme called Cas9. And so in order to go forward, I want to show you this picture from 2011. This was a review published by the CRISPR community that summarizes the diversity of CRISPR systems known at the time. And so each one of these enzyme systems represents one type of CRISPR system. And so you can see there are many, many different types of CRISPRs. Cas9 is right over here, and it's one of many, many types of CRISPR systems. What is unique about Cas9 is that it's simple. It uses a very small number of proteins to be able to do all of the defense function that CRISPR provides. And then there are, of course, these other much more complicated CRISPR systems. So just based on that generalization, we can reclassify CRISPR system by grouping them into 2 classes. The first class is called class 1, and these are CRISPR systems that use a whole cluster of proteins that together form the effector module. And so this is the multi-protein that come together to form this RNA-guided recognition, also destruction complex. Cas9, on the other hand, we can classify this as the class 2 system, which use just a single protein to be able to both recognize the target and also destroy the target. So if you go back and look at this roadmap or this landscape of CRISPR diversity, you can see that really there's only one class 2 system, which is Cas9, and then there are many, many more class 1 systems, these multi-protein encoding CRISPR systems. So we asked the question, if there are many different class 1 systems, could there also be other forms of class 2 systems? Is Cas9 alone, or could there be other cousins of Cas9? And so we set out on a computational expedition. We surveyed the diversity of bacterial genome sequences that have been accumulating, and we asked the question, are there other single protein CRISPR effector complexes? So the way the search works is we take advantage of the fact that CRISPR systems are bacterial. And so that means if you find one gene that's CRISPR related, the nearby genes are also most likely CRISPR related because bacterial systems are encoded in what's called operons. Genes are grouped together. And so by taking one of the most well-conserved CRISPR proteins called Cas1, we can then do a computational search in the bacterial sequences, agnostic to which type of bacteria it is, and ask the question, what are all the different sequences that carry this CRISPR-Cas1 gene? And so by doing that, we can then find a large number of contigs. So contigs is just a jargon for a piece of bacterial genomic sequence. And we can find many, many fragments of bacterial genomes that carry the Cas1 gene. So by annotating these contigs, we can then figure out what are the genes that are encoded next to the Cas1 gene. And so that allowed us to then annotate what's called open reading frames next to Cas1, and then based on homology to other proteins, we can annotate the function of each one of these open reading frames. So for many of them, we can then figure out that it corresponds to genes of known function, something that we have studied before. But then for a small number of genes, we don't know what they do because they don't share similarity to anything else that we have studied before. And so these are what we call the unknown genes. So because we're trying to find new CRISPR systems that people haven't discovered before, we only focus on these genes of unknown function because they are more likely to tell us that there's something new out there. So now we have a list of genes of unknown function, and we'll have to figure out which ones are the most probable candidates to experimentally investigate. And so then we apply two selection criteria. The first one is, for each one of these genes that we discovered, does it represent a whole family of proteins? That's an important criteria because if it only occurs once, we don't know what it really does. But if it occurs many, many times and used by many different bacterial cells, then we at least know that this is important. Evolution has conserved this gene, and it's being used by many, many different bacterial cells. The second criteria that we have is for each member of the family that we found, do most members of the family co-locate next to a CRISPR repeat sequence? And if that's true, then we know that by proximity, it's probably a CRISPR-related protein because it's not only conserved, but it's always found next to a CRISPR repeat array. And so that probably means that it's important for CRISPR activity. So it's based on this that we were able to discover a number of new class 2 CRISPR systems. And so this is a summary of what's known to date. So mainly, over here on top are the Cas9 containing CRISPR systems. So based on this computational approach, we then found a second family of CRISPR proteins called Cas12. These are also RNA-guided DNA targeting enzymes. In fact, we have also developed these for a variety of gene editing applications. The third family that we found is called Cas13. These are RNA-guided RNA targeting systems. And so they also represent a whole diverse array of different enzymes. So to date, at least based on the sequences that the scientists around the world have collected into the bacterial genome database, there are three main families of class 2 CRISPR effector proteins. And those are two DNA targeting systems, Cas9 and Cas12, and then one RNA targeting system, Cas13. And so we've done a lot with Cas9 and Cas12, but I thought I'll tell you more about Cas13. It's quite interesting, and there are some very interesting applications of the system. So how do we study a new CRISPR system? Most of these CRISPR systems are found in bacterial species that are very hard to culture. In fact, most of them are anaerobic, and we can't really grow or manipulate them at a genetic level. So the first thing we do is we take that CRISPR system, the whole section of the bacteria's genome, we synthesize it, and then we transplant that into E. coli. E. coli are much more amenable to genetic manipulation, and we know how to study them. So for one of these Cas13 systems from Leptotrichia shahi, we take the entire locus of that CRISPR system, and we transfer that into E. coli, and then we do RNA-seq. We try to see are the RNA and the proteins expressed. And so if you look at the RNA-seq trace here, you can see that each one of these humps over here correspond to one of the CRISPR RNAs. And this is a very good sign because it suggests that the system, after being transported into a different type of cell, can still be expressed. And if the CRISPR RNA is processed from this long RNA into individual guide RNAs, this means the enzymes may also be active. So by analyzing the primary sequence of Cas13, we can find specific domains that are highly conserved. And so if you look at LSH Cas13, what you'll find is that there are two very conserved motifs called hairpin domains. Hairpin domains are nuclease domains found in RNases or bacterial toxins. And that's what gave us the idea that this may be RNA targeting. So it's based on this observation, we can then hypothesize and come up with an experiment to test that hypothesis. So the experiment we did is to set up a screen. So there's a bacterial virus, it's called MS2, and it's an RNA virus. So the genome is encoded in RNA. It never undergoes a DNA intermediate during replication, so it's only in RNA form. And so we can test whether or not this enzyme can target this RNA virus. So it happened to be that MS2 has a pretty small genome. It's only 3,800 bases long. And so that means we can design guide RNAs to tile along the entire length of this genome. So we can go from the beginning of the genome all the way to the end, design specific guides that cover the whole length, shifted by one base at a time. And so for 3,800 bases, we roughly have about 3,780 guide RNAs, because each guide is about 20 bases long, that cover the whole genome. So using solid phase DNA printing, we can synthesize the DNA oligos corresponding to each one of these guides. And this can be done very inexpensively. And so once we have this library of guides, we can then clone them into individual plasmids. Each one of these plasmids carries the Cas13 cluster of enzymes, but also it receives one of these guide spacers. So it's this library of plasmids we can then transform into E. coli, and then we can generate a library of cells that we can test to see, do any of these spacers coupled with Cas13 provide defense against this MS2 RNA virus. So we take this library of cells where each cell carries Cas13 and a different guide that recognizes a different spot on this virus, and then we inoculate with the MS2 phage, and we see what are the cells that survive. And so if any cells survive, then that suggests that the system is functional. It's able to protect the cell against the RNA virus. But then also by sequencing the guides in the surviving cells, we can figure out what are the targeting parameters that governs how this enzyme is able to recognize and also destroy the RNA virus. So we found that based on sequencing, the targeting requirement is quite simple. So this schematic shows if this is the RNA we're trying to target, and the guide RNA recognizes this section of that target RNA, it only needs to be flanked by a single base. So on the 3' end, this is more zoomed in of this schematic, it just needs to be flanked by a single base that we call PFS. It can either be a A, a C, or a U. Just cannot be a G. So that's a very simple targeting requirement. So then we thought to understand the cleavage mechanism of this enzyme a little bit more. So we purified the protein and tried to biochemically understand how this system works. And so what we did is we purified Cas13 and we designed many guides to target different locations on the target RNA, flanked by different bases. And so here is the target RNA going from 5 to 3' end. And then we designed many, many different guides that target different locations. So guide 15 targets here, guide 17 targets over there. And different colors indicate the base that they are flanked by. So when it's red, it's flanked by a U. When it's blue, it's flanked by a cytosine, and so forth. And so if you test all of these different guides against the same target RNA, what you find is that the motif that we observed by sequencing is valid. And so if you have the input RNA, as long as it's flanked by a C, a A, or a U, that RNA can get cleaved into different fragments. So that shows how RNA is getting cleaved by Cas13. So this is cleavage of the target RNA. But then there's something more to that. And that's a second mode of cleavage we call collateral activity. This is quite unique to Cas13. So the way this works is shown here. So consider this schematic. We have a guide RNA, and we have a piece of RNA that's not targeted by this guide RNA. So there's no homology between the guide and also the target. So if you add Cas13, there's no recognition, so there's no cleavage. And so this non-target RNA remains intact. What happens if we have the RNA that's recognized by the guide? So in this experiment, we have the target RNA as well as the guide RNA, and the guide recognizes the target. So we know from the previous slide that the guide will recognize the target and then allow Cas13 to cleave the target. But what happens to the collateral RNA? So it turned out that the Cas13 enzyme will not only cleave the recognized RNA, but it will also cleave other RNA that's not even recognized by the guide sequence. And so that's what we call collateral RNase activity. So upon recognition, so once it recognizes the RNA, the enzyme remains switched on. It can go and cleave other RNA even after cleaving the recognized target. So what does this mean in the context of the biological system? Well, biologically, this collateral activity allows the CRISPR-Cas13 system to provide two modes of defense. So the first mode of defense is the normal form of immunity, where a virus comes into the bacteria and it gets detected by the sensor of infection. And then it recognizes the virus and then cleaves the virus to circumvent spread of the virus. But what if the virus escaped the sensor? So it bypassed the sensor and it began to replicate. At that point, it's a more dangerous situation because if the virus began to replicate and leaves the cell, there are going to be many viruses and it will start to infect many more cells. So what Cas13 can do is that it can recognize the infected virus and also replicated virus. Each one of the replicated virus can trigger one molecule Cas13 to be non-specific, and enough molecules of Cas13 can go and cleave enough molecules of the cellular RNA and be able to destroy the cell. So that sequesters the infection and it prevents the spread of the virus. So that's what we call the second mode of immunity, which is programmed cell death. So either the virus gets recognized early and gets destroyed, or the virus began to replicate, and at that point, altruism kicks in. The bacteria, the infected bacteria gets destroyed, and the population is saved. So that's all great, but what is this really useful for? So turned out that we can use Cas13 for a couple of really interesting applications. The first application is developing this collateral activity to be able to recognize infections. And so for viruses like Ebola viruses or Zika viruses, we can turn Cas13 into a sensitive and also rapid diagnostics assay to be able to recognize whether or not the virus is present in a particular patient sample. So the patient sample can be either urine, saliva, or blood, and so we can apply this test quite easily. This is how this works. So we take advantage of the collateral activity, and we design a simple chemical reaction where we have the Cas13 enzyme programmed with a guide RNA that can recognize Zika virus. And we put this into the patient sample that we're trying to see whether or not Zika is present. If Zika is present, the guide RNA will base pair with the Zika RNA, and then Cas13 will cleave the Zika RNA. But that's not the only thing we put into the reaction. We also put in a reporter RNA. And this is a short RNA that has both a biotin and also a FAM molecule attached to the two different ends. So when Cas13 is activated after recognizing Zika, it can cleave Zika, but also cleave these reporter RNA. So if the reporter is cleaved, biotin is separated from the FAM molecule. And so this is what ends up happening. This allows us to set up a lateral flow assay. And so this is a paper strip test that's very similar to a pregnancy test strip. And so there are two lines. So the first line has stripped habidin, which can bind to biotin. And then there's a second line, which has an antibody that can bind to this FAM molecule. And so if virus is not present, you flow this reaction on this flow strip, and everything will get captured by the strep habidin line because FAM remains tethered by that reporter RNA to the biotin molecule. But if the virus is present, then biotin and FAM are separated. Biotin will get captured by the streptavidin line, and then FAM will keep on flowing, and it will get captured by the second antibody capture line. So if virus is present, you see 2 lines, and if virus is not present, you see 1 line on this paper strip. So very simple test. And so this is how this works. So we took samples that carry Zika, and we had the sample at various concentrations of Zika virus down to sort of nothing. And we found that 200 attomolar is kind of the sensitivity range that we're able to go down to using this paper strip test. And so 200 attomolar correspond to about 100 molecules of virus per microliter of the patient sample. And so that's actually a pretty good sensitivity for being able to report that there is either Zika infection or Ebola or even HIV infection in a given patient. And so by looking to see whether or not the second band shows up, then we know whether or not the infection has happened. So this is for detecting a single virus or a single pathogen. One thing that we wanted to do is to set up a more multiplexable system. What if we want to detect multiple things within the same reaction? And so one of the things that, one of the ideas that we had was inspired by this initial experiment that I showed you earlier. There's something a little bit strange about this gel, which is that when Cas13 targets RNA, it doesn't matter where the guide recognizes on that RNA, you end up with exactly the same cleavage pattern. So it doesn't matter where this guide recognizes, you get exactly the same cleavage pattern. And so that suggests that Cas13 is not cleaving at a recognition site, but instead is cleaving maybe a motif or a secondary structure that's in this transcript, because it's cleaving in exactly the same locations regardless of where the enzyme is sitting on this target. So that then gave us the idea that maybe different members of the Cas13 family have different preferences for either motifs or secondary structures. And so then they cleave different locations on the target RNA. So then we set up a second assay where we evaluated a number of Cas13 family members. So these are Cas13s from different bacterial strains. And then we incubated them with different reporters. And the difference has to do with the sequence that tethers the 2 different reporting moieties on this RNA. So either GG, CU, UU, GA, UC, and different sequences. So what we found is that different Cas13s from different bacteria preferentially cleave one sequence but not other sequences. So LWA Cas13 cleaves AU but not UC, AC, or GA. And then CCA cleaves UC but not AU, AC, or GA. And so by evaluating different Cas13s, you can find a set of Cas13s that have orthogonal reactivity, so they don't crosstalk with each other's intended reporter. This is nice because it means we can go and design an assay where we can have multiple Cas13 family members together and also have different guides for each one to be able to detect different things, all in the same reaction. And so this is how this system works. So we try to see, can we set up a 3-plex assay, try to detect Zika, dengue, or a piece of synthetic RNA sequence. And so we have 3 different Cas13s paired with their corresponding guide RNA and also their respective reporter molecules. So this is what the data looks like. If nothing is present, then no signal in any of the 3 channels. If one of the 3 is present, you get 1 channel out of 3. If you have 2 out of 3 present, then you get 2 channels reporting. And if you have all 3, then you get all 3 channels reporting. And so this is a very nice way to be able to multiplex the assay. And so there are hundreds of different Cas13 family members. And so by evaluating more of these family members, then we'll be able to have a pretty large repertoire of Cas13s to be able to set up multiplex assay. And recently we solved the crystal structure of Cas13, and based on that, we can also do a more structure-guided engineering of this enzyme to be able to detect many different things to cleave different reporters. So this is one application of Cas13 for diagnostics. But a second application is that we can use this to be able to provide therapeutic intervention in cells as well. And this has to do with using Cas13 to home into specific RNA sequences and then bring a catalytic domain called ADAR to convert adenosines into inosines. So adenosine is one of the 4 different bases on RNA. And when you deaminate inosine, you can convert this into a base called inosine. Inosine now has the base pairing property of guanosine. And so if you can convert A to I, then you can convert A to effectively become a G on the RNA molecule. And so this means you can change the protein that's encoded by a given RNA by editing at the RNA level. So the way we do this is we go into Cas13, we inactivate the 2 heparin domains that are responsible for cleaving RNA because we simply want Cas13 to just bind. And by taking this mutant Cas13, we call D-Cas13 or deactivated Cas13, we can then use it to bring ADAR domains to a specific RNA. And we can program this protein to recognize a specific RNA by giving it a different guide sequence. Now, when Cas13 binds to a target RNA, that target RNA will have many, many adenosines on that RNA. And so how do we dictate which adenosine is edited by this Cas13 ADAR system? And it turned out that ADAR has a very unique way of recognizing which adenosine to edit. And that's through the recognition of an A to C mispair, a bubble in the RNA duplex. And so when a bubble is established, then that mispaired adenosine is the one that's deaminated and converted into inosine. And so that's how we achieve a single base editing at a messenger RNA level. This is a more detailed schematic. So for example, here, this is the mRNA sequence for a luciferase gene. And there are many A's that are recognized by this guide sequence, but we can dictate this A to be edited by mispairing that A with the cytosine on the other side. So by forming that bubble, we can specify which base to change. So we set up the system, and we thought to see how well this system works. The first version of the system is called Repair V1. And so we found that you can very effectively edit that adenosine into inosine. This is over 90% editing. But if you sequence the whole transcriptome of that cell, we find that there are a number of off-target sites. And so this is all of the different chromosomes lined up. You can see many different transcripts across the different chromosomes can have off-target edits affected by Cas13 ADAR. So we thought to understand what may be contributing to this off-target editing activity. And so what we found is that even if you use a non-targeting guide, a guide that doesn't recognize this luciferase RNA, we also get a bunch of off-target RNA. And these off-target sites are identical to the one where we have a targeting guide. And so this suggests that the off-target effect is not necessarily mediated by this guide that's misrecognizing other RNA, but probably just by promiscuous recognition by the protein and either Cas13 or ADAR. So that gave us the idea to then look at ADAR and see, can we improve the substrate recognition of this enzyme? And so fortunately, Peter Beal's group at UC Davis has solved the structure of ADAR2 in complex with the RNA substrate. And so from this, we can identify a number of different residues that make non-sequence specific contact with the RNA. So previously, we had engineered ADAR, we had engineered Cas9 to be more specific by reducing the non-specific interactions. And so we thought maybe we can reduce non-specific interactions by weakening some of these bases, these amino acids, so that they can sort of more depend more on the Cas13 RNA guide mediated recognition of the target. So we tested a number of different residues, and what we found is that some of these can significantly increase the specificity. So we can really increase specificity. It does reduce on-target activity a little bit, but it's disproportionately able to increase the on-target editing specificity. So then we tested this on the transcriptome. So here is Repair V1, where we sequence the transcriptome at pretty high depth, 125x. And we now find there are about 20,000 off-targets. So because we're sequencing deeper, we pick up more off-target. But with Repair V2, and just by introducing one single point mutation, T375 to a glycine, we can really clean it up. So we now get 20 off target, so 1,000-fold reduction, and then we can still get pretty good on-target editing. So on-target reduces by about 50%, but then the off-target reduces by about 1,000-fold. And so that's one really promising way to go in and engineer the system. So I have begun to test how well this may work in a more therapeutic relevant context. And so we engineered an AAV virus that encodes the Cas13 ADAR system and the guide RNA. And we can put this onto primary neurons to see, can we edit endogenous RNA in these primary cells to be able to alter the way that these RNA are translated into protein. And so what we found is that the system can work pretty well. And so in this example, when we targeted a particular mRNA, the GAPDH mRNA, we can get about 40% editing at the target base. And so that's a pretty good place to start. So that's one thing we're working toward. We're trying to now apply this in animal models of disease to see, can we use this to treat single point mutations either in the brain or the liver or other tissue areas. A second thing we have been doing is to try to improve the repertoire of changes we can make in a cell. And so here, what we thought to do is to convert this into a silent deaminase. There are natural silent deaminases, but they don't have single base precision. And so ADAR has this nice mechanism of recognizing a mismatch bubble. And so we began by testing ADAR to see whether or not it can deaminase the cytosine into your cell, and we saw the activity is very, very poor. But by engineering the pocket that recognizes the substrate, even just with a single additional mutation of V351G, we can now edit the cytosine with pretty good efficiency. This is about 20%. This is version 1 of the rescue system, C2U editing, but now we're up to about version 10, and we can get pretty robust editing, sometimes up to 80% in specific cytosines now. So we're continuing to try to get that technology matured. And then we're continuing to understand more of the basic structural function of this enzyme. And so we began to explore the diversity of Cas13 enzymes. We're finding that Cas13Bs, the blue ones, are the most active in translation into human cells. And then by solving the crystal structure of Cas13B, we're now able to do even more structure-guided engineering to be able to make the system more powerful and more robust. So this is what we've been doing with CRISPR Cas13. Another area that we have embarked on recently that we're really excited about is to ask the question, are there adaptive immune systems beyond CRISPR in the natural diversity? So the reason this is interesting is because if you look at these 4 molecules, RNA-guided nucleases like Cas9 or antibodies, VLRs, and tail proteins, these are different forms of adaptive immune systems found in nature. And each one of these have been very powerful in building new novel molecular technologies. And so by asking the question, are there other novel programmable systems we can see, are there interesting to find? So I'll just quickly show you a couple. This is a putative reprogrammable adaptive protease from Streptococcus. There's a protease domain with repeats that we might be able to engineer and adapt. Here is a secreted immunity protein from Photorhabdus that's very conserved but with a fixed hypervariable region. And so these are things that we'll continue to explore. So there's a lot of data that we can still explore more. So there are probably a lot of interesting things to discover. So finally, just want to acknowledge my lab. This is really the important slide because these are the students and postdocs who work with me to develop them. We've been lucky to have a number of collaborators and funding agencies to support our work. Sorry for running over time. Happy to answer any questions, and thank you so much for interest in our work.
Thank you. Thanks. There was a lot of new developments. Very interesting. Yeah. So we can welcome some questions, please.
Yeah.
Hi. Thank you. Thanks for the very fascinating speech. And I'd like to ask if we'd like to do genome editing or transcription editing. do you recommend to see the off-target in genome-wide or like transcriptome-wise on the transcriptome level?
Yeah, good question. So there are two ways to go about altering nucleic acids in cells. There's editing the genome or editing RNA, so the transcriptome. And so each one of these can have off-target. And the consequences of these off-targets are different. If you're editing the genome, changes at the DNA level will persist along with that cell. So as long as the cell is alive, those off-targets will persist. And depending on what they are, they could have long-term consequences. RNA off-targets are transient. As soon as the mRNA is destroyed or degraded, then that off-target disappears. And so because of that, there are different considerations depending on the application. But the good news is that there's good advance in both DNA and RNA editing to make systems more specific. And so hopefully what you can do is really to figure out, do you want to edit RNA, which is transient, or do you want to edit DNA, which is more permanent, and not worry so much about off-target, because we're trying to really get systems to be very precise.
Yeah. Yes? Mm-hmm.
Uh-huh.
Because of the bubble, so it finds the
Right.
So why isn't the bubble repaired before that system finds another?
Good question. So it turns out that when there's a bubble in DNA, you can have what's called mismatch repair to repair the DNA. But when you edit RNA, there isn't a mismatch repair at the RNA level. And so in fact, inosine naturally occurs in RNA. There's natural RNA editing process that happens in glutamate receptors and so forth. And so when we put an inosine in there, it stays with the RNA, and that's how we're able to effect a change at the protein level. Yeah. Oh, wow. Yeah. Yeah. Yeah.
In the back, yes, please. I should say to the students that this young lady who just asked the question, she's been working on this, so she doesn't just understand the details.
Very cool.
Like if it were all new to her, then maybe she wouldn't ask that kind of
very good question.
Yeah.
So my question is, if you just target the RNA, then since some parts of RNA may be similar to other parts of another RNA, so can you just target some region of this RNA so to minimize off-target damage?
Sure. So thank you for the question. On a piece of RNA, there can be the protein coding sequences and also other signal sequences. And so what we have been trying, so you can use RNA editing to affect both the signal sequences as well as the protein encoding sequences. The off-target can happen really anywhere along a piece of RNA. And so what we have been doing is try to really increase the stringency of binding, that really requiring recognition between the guide and the target in order to be able to edit. So if there are other secondary structures or other sequence motif-based binding, we'll try to get rid of them so that they don't introduce off-target. Yeah.
Yeah. Oh, okay. Another question there, please.
When I hear you talk about the structural components of some of the splicing that goes on and the site identification. When you talk about secondary structures, how are you exploring the shape and geometry and chemistry of these umbrella ideas of secondary structures? Right. This is a very curious area.
Yeah, that's a really good question. So we try to do it a few different ways, but we always need more ways to be able to do it better. So the first way is we can predict where secondary structures may be, just based on looking at sort of complementarity or hairpin type of sequences within the RNA. So you can computationally predict where some of these secondary structures may be. based on that prediction, we can then generate libraries of RNAs. It's easy to synthesize a lot of different RNA sequences. So we can then experimentally test to see what are different secondary structures that are prone to cleavage by Cas13 or not by Cas13. And so through the computational prediction and then collecting a lot of data, we can then start to try to form a more sort of emergent model to be able to predict what activity of these systems may be. And so that's what we try to do. And then the really low throughput, but quite informative way is to solve the structure. And so by doing structural biology, we can start to look at what are the different hydrogen bonds that are being formed with a particular structure. What is a pocket that on the protein that binds to this looks like, and then use that to then tweak the system even further. So it's kind of, we'll try to see whatever is available tool-wise to be able to apply this problem. And yeah, thank you.
Shuguang has a question? Yeah.
One question about science, a question about ethics. The science first.
Sure.
People have tried to use CRISPR system for treating disease, and is CRISPR the proteins cause immune problem in human?
Yeah, that's a really good question. So first of all, these CRISPR proteins are from bacteria, and so if we're trying to use them in a therapeutic context by injecting them into the human body, then it's possible that our body will recognize them as foreign and mount an immune response. So that's a really good question. There have been some studies that published suggesting that in some individuals there may be natural immunity against these proteins. And so that's one thing to consider. When we're treating patients therapeutically, we don't actually need the protein to hang around for a long time. And so what we can do is to do what gene therapists already do: immune suppress the patient before you deliver these potentially immunogenic proteins. And the protein will last for on the order of maybe a week. And so if you immune suppress the patient for just that period of time, you can allow the editing to happen, but then it will disappear. And the immune system won't even recognize that something was there before. Other ways to work on this is to humanize these proteins. We can computationally, also experimentally, identify sections of this protein that are immunogenic, and we can engineer those out to make it more tolerant to the human immune system. This has been done with antibodies already. Yeah.
Second, ethics. So you can treat genetic disease, but it's also a slippery slope.
Right.
Eventually can enhance this and that and so on, make a superhuman being and so on, eugenics or whatever.
It's a very good question. So it's definitely an important question to keep discussing and considering. I mean, it's hard. So the stuff that, things that we talk about, eugenics and so forth, it's more science fiction than reality. We can't even treat cancer that well yet, much less to be able to engineer much more complicated human traits. So we'll have some time to before we have to worry about that, but these are also questions that have existed for a long time. Since the early days of recombinant DNA, these have been questions that have been grappled with by philosophers, ethicists, and scientists. So we'll have to continue to think about it. I think the question is not whether or not it will happen. It will probably happen because anything that confers things that are beneficial to human, I think will get adapted. But what is important is to figure out how will the society adapt to the effect of these technologies, and also how do we manage and potentially prevent the creation of more inequality as a result of that. And so those are the things that I think as a society we'll have to think about.
One of the things with the current political nonsense, saying people probably would like to engineer white skin because the current terrible president is racist.
Well,
I
thank
think
you.
There's one question in the back there. Let's. Can you hear? Maybe we can have the microphones and. Just hold on for a second, please.
Yeah.
Thank you. Wonderful talk. My question is, you know, the microbiome, they say we are more bacteria than human. Will CRISPR have an influence on that microbiome?
Yeah, great question. CRISPR is related to microbiome in many ways. In fact, many of the CRISPR proteins that we have been working with and turning into tools are originally from the human microbiome. So Cas9 is from Streptococcus or Staph aureus, and Cas13, some of them are from bacteria that live in our gut. And so they are naturally there. And they're used by these bacterial cells in the microbiome to defend against viruses or to sort of wage warfare in the microbial world. The second thing that's relevant is you can potentially use CRISPR systems to develop new antibiotics. So a lot of bacteria already carry CRISPR. In fact, over 60% of bacteria naturally have CRISPR system. So then if you can figure out a way to deliver just the guide sequences into those bacteria, you can exploit their own CRISPR system to destroy themselves. And that might be a way to counter infectious disease using their own system. So those are a couple of ways that we can use the CRISPR system. But then there will certainly be other mechanisms as well. You can use it to better engineer bacteria, maybe to shape the composition of microbiome to be able to treat disease. Yeah, thank you.
Okay, we take a last question now, then we have to break.
Thanks, that was an awesome presentation. I was wondering if you could speculate as to why CRISPR systems are only found in microbes. If they're so useful and powerful as kind of a molecular memory,
right.
Why don't we have a CRISPR or plants or whatever?
Yeah, that's a good question. So it is true. So far, CRISPR systems, things that use these repeat sequences, have only been found in bacteria and archaea, and it's very prevalent in bacteria. 60% bacteria, 90% archaea. It has not been found in eukaryotic cells. Partly because maybe these CRISPR systems are multi-gene encoded systems, and so they somehow got lost when sort of the eukaryotic development happened, or maybe they evolved independently of eukaryotic cells after bacteria and eukaryotic lineage have split. But I think what is interesting to recognize is the principle of using nucleic acid to recognize nucleic acid is exploited by all organisms. So we have the RNA interference system that uses RNA to recognize other RNA. We have the PIWI system, which also uses nucleic acid to recognize nucleic acid. And so there are many other enzymes that use this same principle of protein mediating sort of nucleic acid recognition. And I think that's the universal thing that nature uses. The specific Cas9 enzyme or Cas13, that's probably specific to the bacteria.
Thank you.
Thank you. Okay, thank you, everyone.
Okay, thanks to... Thank you, Dr. Zhang. And let's break for lunch and resume at 1:30.
and the dangerous chemicals involved in their creation, use, and disposal.
Mycotexture is a subgroup of bioterials that uses fungal mycelium to bind organic waste into strong composites. Material made from nothing but waste and fast-growing organisms can be structural, insulative, fire-resistant, and sound attenuating.
Bioterials can be grown and locally fabricated. Eliminating the need for imports empowers local communities. This is the new building revolution. Whole economies, including homes, fab labs, and businesses, can be built using agricultural waste, microorganism, and know-how. A truck delivers equipment, supplies, and staff to make a mushroom farm, homes, and businesses. Temporary inflatable building forms are laid flat and tied to weighted foundations. Using an air compressor, the forms are inflated within minutes. Rammed earth floors are created from dirt on site to ballast the form. Outer rubber shells are added to provide weather protection. Walls and window frames are made from rammed earth sourced on site. The structure is livable on the very first day. Time for rest. Tomorrow begins work on the farm. Jobs are created at a mushroom farm. Workers learn by doing the new skill set. Mycelium is seeded into square columns. Gourmet mushrooms grow through holes in the columns and are readily harvested. Nutritious, delicious, high-protein health food are made to feed the community. Excess mushrooms can be processed and packaged, creating high-value goods to international markets. The waste is taken from the square columns and used as unit masonry for construction and manufacturing. The composite is brought back to the shelter and placed within an integrated growth chamber. The mycelium, still alive, continues to grow within 2 layers of the inflated structure, and the masonry units fuse together. Heating mechanisms embedded within the membranes, rendering it inert and resistant to any further microbial growth. The inflatable form is removed and can be used again and again. The outer membranes can be customized, opening up sponsorship opportunities. The building can be a biohub, a home, a biofab, fabrication lab, with business incubator, a tool lending library, and education center. This is not a house. It's an ecosystem, an economy, a community, the future.
welcome to the Team Dimension Data Zwift Academy Finals here in Cape Town, South Africa. 35,000 athletes applied, 3 remain. 1
toughest one of all. Tired legs are taken into the longest.
This is not a house. It's an ecosystem. Leveraging research by MIT and NASA, researchers and architects have developed new way of building using bioterials. Bioterials are building materials that use the growth of microorganisms in their manufacture. They are poised to replace plastics, leathers, and foams, and the dangerous chemicals involved in their creation, use, and disposal.
Mycotecture is a subgroup of bioterials that uses fungal mycelium to bind organic waste into strong composites. Material made from nothing but waste and fast-growing organisms can be structural, insulative, fire-resistant, and sound attenuating.
Bioterials can be grown and locally fabricated. Eliminating the need for imports empowers local communities. This is the new building revolution. Whole economies, including homes, fab labs, and businesses, can be built using agricultural waste, microorganism, and know-how. A truck delivers equipment, supplies, and staff to make a mushroom farm, homes, and businesses. Temporary inflatable building forms are laid flat and tied to weighted foundations. Using an air compressor, the forms are inflated within minutes. Rammed earth floors are created from dirt on site to ballast the form. Outer rubber shells are added to provide weather protection. Walls and window frames are made from rammed earth sourced on site. structure is livable on the very first day. Time for rest. Tomorrow begins work on the farm. Jobs are created at a mushroom farm. Workers learn by doing the new skill set. Mycelium is seeded into square columns. Gourmet mushrooms grow through holes in the columns and are readily harvested. Nutritious, delicious, high-protein health food are made to feed the community. Excess mushrooms can be processed and packaged, creating high-value goods to international markets. The waste is taken from the square columns and used as unit masonry for construction and manufacturing. The composite is brought back to the shelter and placed within an integrated growth chamber. The mycelium, still alive, continues to grow within 2 layers of the inflated structure, and the masonry units fuse together. Heating mechanisms embedded within the membranes, rendering it inert and resistant to any further microbial growth. The inflatable form is removed and can be used again and again. The outer membranes can be customized, opening up sponsorship opportunities. The building can be a biohub, a home, a biofab, fabrication lab, with business incubator, a tool-lending library, and education center. This is not a house. It's an ecosystem, an economy, a community, the future. This is not a house. It's an ecosystem. Leveraging research by MIT and NASA, researchers and architects have developed new way of building using bioterials. Bioterials are building materials that use the growth of microorganisms in their manufacture. They are poised to replace plastics, leathers, and foams, and the dangerous chemicals involved in their creation, use, and disposal.
Mycotexture is a subgroup of bioterials that uses fungal mycelium to bind organic waste into strong composites. Material made from nothing but waste and fast-growing organisms can be structural, insulative, fire-resistant, and sound attenuating.
Bioterials can be grown and locally fabricated. Eliminating the need for imports empowers local communities. This is the new building revolution. Whole economies, including homes, fab labs, and businesses, can be built using agricultural waste, microorganism, and know-how. A truck delivers equipment, supplies, and staff to make a mushroom farm, homes, and businesses. Temporary inflatable building forms are laid flat and tied to weighted foundations. Using an air compressor, the forms are inflated within minutes. Ramped earth floors are created from dirt on site to ballast the form. Outer rubber shells are added to provide weather protection. Walls and window frames are made from ramped earth sourced on site. The structure is livable on the very first day. Time for rest. Tomorrow begins work on the farm. Jobs are created at a mushroom farm. Workers learn by doing the new skill set. Mycelium is seeded into square columns. Gourmet mushrooms grow through holes in the columns and are readily harvested. Nutritious, delicious, high-protein health food are made to feed the community. Excess mushrooms can be processed and packaged, creating high-value goods to international markets. The waste is taken from the square columns and used as unit masonry for construction and manufacturing. The composite is brought back to the shelter and placed within an integrated growth chamber. The mycelium, still alive, continues to grow within 2 layers of the inflated structure, and the masonry units fuse together. heating mechanisms embedded within the membranes, rendering it inert and resistant to any further microbial growth. The inflatable form is removed and can be used again and again. The outer membranes can be customized, opening up sponsorship opportunities. The building can be a biohub, a home, a biofab, fabrication lab with business incubator, a tool lending library, and education center. This is not a house. It's an ecosystem, an economy, a community, the future.
Years ago, Stone Age Britons raised this mysterious monument, Stonehenge. Nobody knows how they did it, how they moved and stood these massive stones. It's a scientific mystery. This man swears he's cracked. He's building his own Stonehenge in his Flint, Michigan backyard to prove it. Wally Wallington's not a scientist, but he knows a thing or two about moving rocks.
That's a 300-pound block. This is a 1,600-pound block. Not too difficult.
He's a retired construction worker whose passion is moving heavy items. His feats always draw a crowd, mostly family.
I thought he's crazy. Who cares about moving blocks around? But then when you look at the magnitude of the weights that he's moving around, it is really impressive, and he's not using any equipment or anything.
In playing with blocks, Wally thinks he's discovered how Stonehenge was moved.
This is my first Stonehenge arch I permanently put in place. It's 3 blocks, they weigh over a ton each.
It's all based on a very simple technique.
I found a simple explanation for this, to move a block about the weight of a minivan, would be to place a stone underneath it. And once I balance on it, I can spin it. Of course, with that spin, you've seen I didn't go anywhere. But I got 2 handles on my lever, and I could place another stone on this side. Now, every time I spin a half rotation on each stone, I move the block horizontally, the distance between the stones. With my own output, I could move a 1-ton block 300 feet per hour.
Using this technique, he's moved everything from 1-ton blocks to buildings.
He moved up to a pole barn at one point, a 30 by 40 pole barn that he moved 300 feet for me, and it was more or less, you know, what else can we move? And he looked at me and says, hey, can we move your barn? And I said, sure, let's, you know. Next thing you know, it was 300 feet in the other direction onto another piece of property.
It can move barns, and Wally's betting it moves Stonehenge's 25-ton rocks. But raising these giants is a whole different puzzle.
This is 19,200 pounds. Yeah, it's 128 square feet of cubic feet of concrete.
Today, Wally hopes to solve it. He'll put one of his theories to the test and try to stand this 19,200-pound block by himself.
I've tried to do this without any mechanical machinery at all. I've used mostly sticks and stones for my equipment. No pulleys, no hoists, no metal levers. Just try to use gravity, too, I believe is my favorite tool. Whoop.
The first goal is getting this block 3 feet off the ground.
In order to move it up to this point, I just rock the block back and forth, adding weight to that end, and that opens a gap on this side, and just slide a board in. Then I add the weight to that end. There she goes. And slide the board in on this end.
This shoring box acts like a jack, slowly raising the block. It's 3 feet off the ground, but tomorrow is the big experiment, standing it up. The grandkids have taken the day off school, and the cameras are rolling.
Hey, you guys watch Papa. He's going to do something wild.
They're here to watch Grandad lift a block the weight of 2 bulldozers.
It's ready.
Yes, it is.
Here's how it's supposed to work.
The first thing I'm going to do here is release this temporary shoring I have set, and come over here, release some of my counterweights, and that's going to put the entire weight of the block on this rope. So then I'm going to release the rope, come back in, and the rope's going to be my brake. I'm going to guide it into the pit. The easiest way I can explain this is this is just a big teeter-totter, and I got the big kid on that end, and he's going to go down, this end's going up.
He believes in his technique, but he's had some setbacks in the past.
One day I got thrown over top of a block, and a couple times I had the blocks roll off the top and almost land on my feet. One time I knocked myself out cold on the concrete, and so it's been challenging. Okay. Are you ready? Yes! All right. Now just start spraying the sand. The sand will wash out and the block will start coming down.
Once the sand is washed from the pit, the block's own weight slowly stands it up.
Okay, finally, she's between the lines, guys. Looks good.
He did it.
Thank you, guys.
Raised a massive block using a technique so simple that Wally believes it must have been used to raise Stonehenge.
If they were to use this technique to raise the stones at Stonehenge, I believe they could have got it done with a lot smaller crew than anybody has imagined before.
It won't be long before this backyard is a little more crowded.
This is my first block in the circle. I plan to have 8 of them standing on end like that, all up 11 feet in height. The next one may be a little heavier. We're going to keep going a little further with it.
Until then, the only mystery that remains is this stone circle's name.
Wally's Wonder Circle.
How about the Wallington Circle?
What's that? Stonehenge Reloaded.
There you go.
Stonehenge Reloaded. There you go. That's not bad.
This is not a house. It's an ecosystem. Leveraging research by MIT and NASA, researchers and architects have developed new way of building using bioterials. Bioterials are building materials that use the growth of microorganisms in their manufacture. They are poised to replace plastics, leathers, and foams, and the dangerous chemicals involved in their creation, use, and disposal.
Microtexture is a subgroup of bioterials that uses fungal mycelium to bind organic waste into strong composites. Material made from nothing but waste and fast-growing organisms can be structural, insulative, fire-resistant, and sound attenuating.
Bioterials can be grown and locally fabricated. Eliminating the need for imports empowers local communities. This is the new building revolution. Whole economies, including homes, fab labs, and businesses, can be built using agricultural waste, microorganism, and know-how. A truck delivers equipment, supplies, and staff to make a mushroom farm, homes, and businesses. Temporary inflatable building forms are laid flat and tied to weighted foundations. Using an air compressor, the forms are inflated within minutes. Ramped earth floors are created from dirt on site to ballast the form. Outer rubber shells are added to provide weather protection. Walls and window frames are made from ramped earth sourced on site. The structure is livable on the very first day. Time for rest. Tomorrow begins work on the farm. Jobs are created at a mushroom farm. Workers learn by doing the new skill set. Mycelium is seeded into square columns. Gourmet mushrooms grow through holes in the columns and are readily harvested. Nutritious, delicious, high-protein health food are made to feed the community. Excess mushrooms can be processed and packaged, creating high-value goods to international markets. The waste is taken from the square columns and used as unit masonry for construction and manufacturing. The composite is brought back to the shelter and placed within an integrated growth chamber. The mycelium, still alive, continues to grow within 2 layers of the inflated structure, and the masonry units fuse together. Heating mechanisms embedded within the membranes, rendering it inert and resistant to any further microbial growth. The inflatable form is removed and can be used again and again. The outer membranes can be customized, opening up sponsorship opportunities. The building can be a biohub, a home, a biofab, fabrication lab, with business incubator, a tool lending library, and education center. This is not a house. It's an ecosystem, an economy, a community, the future. This is not a house. It's an ecosystem. Leveraging research by MIT and NASA, researchers and architects have developed new way of building using bioterials. Bioterials are building materials that use the growth of microorganisms in their manufacture. They are poised to replace plastics, leathers, and foams, and the dangerous chemicals involved in their creation, use, and disposal.
Microtexture is a subgroup of bioterials that uses fungal mycelium to bind organic waste into strong composites. Material made from nothing but waste and fast-growing organisms can be structural, insulative, fire-resistant, and sound attenuating.
Bioterials can be grown and locally fabricated. Eliminating the need for imports empowers local communities. This is the new building revolution. Whole economies, including homes, fab labs, and businesses, can be built using agricultural waste, microorganism, and know-how. A truck delivers equipment, supplies, and staff to make a mushroom farm, homes, and businesses. Temporary inflatable building forms are laid flat and tied to weighted foundations. Using an air compressor, the forms are inflated within minutes. Ramped earth floors are created from dirt on site to ballast the form. Outer rubber shells are added to provide weather protection. Walls and window frames are made from ramped earth sourced on site. The structure is livable on the very first day. Time for rest. Tomorrow begins work on the farm. Jobs are created at a mushroom farm. Workers learn by doing the new skill set. Mycelium is seeded into square columns. Gourmet mushrooms grow through holes in the columns and are readily harvested. Nutritious, delicious, high-protein health food are made to feed the community. Excess mushrooms can be processed and packaged, creating high-value goods to international markets. The waste is taken from the square columns and used as unit masonry for construction and manufacturing. The composite is brought back to the shelter and placed within an integrated growth chamber. The mycelium, still alive, continues to grow within 2 layers of the inflated structure, and the masonry units fuse together. Heating mechanisms embedded within the membranes, rendering it inert and resistant to any further microbial growth. The inflatable form is removed and can be used again and again. The outer membranes can be customized, opening up sponsorship opportunities. The building can be a biohub, a home, a biofab, fabrication lab, with business incubator, a tool lending library, and education center. This is not a house. It's an ecosystem, an economy, a community, the future. This is not a house. It's an ecosystem. Leveraging research by MIT and NASA, researchers and architects have developed new way of building using bioterials. Bioterials are building materials that use the growth of microorganisms in their manufacture. They are poised to replace plastics, leathers, and foams, and the dangerous chemicals involved in their creation, use, and disposal.
Mycotexture is a subgroup of bioterials that uses fungal mycelium to bind organic waste into strong composites. Material made from nothing but waste and fast-growing organisms can be structural, insulative, fire-resistant, and sound attenuating.
Bioterials can be grown and locally fabricated. Eliminating the need for imports empowers local communities. This is the new building revolution. Whole economies, including homes, fab labs, and businesses, can be built using agricultural waste, microorganism, and know-how. A truck delivers equipment, supplies, and staff to make a mushroom farm, homes, and businesses. Temporary inflatable building forms are laid flat and tied to weighted foundations. Using an air compressor, the forms are inflated within minutes. Ramped earth floors are created from dirt on site to ballast the form. Outer rubber shells are added to provide weather protection. Walls and window frames are made from ramped earth sourced on site. The structure is livable on the very first day. Time for rest. Tomorrow begins work on the farm. Jobs are created at a mushroom farm. Workers Learn by doing the new skill set. Mycelium is seeded into square columns. Gourmet mushrooms grow through holes in the columns and are readily harvested. Nutritious, delicious, high-protein health food are made to feed the community. Excess mushrooms can be processed and packaged, creating high-value goods to international markets. The waste is taken from the square columns and used as unit masonry for construction and manufacturing. The composite is brought back to the shelter and placed within an integrated growth chamber. The mycelium, still alive, continues to grow within 2 layers of the inflated structure, and the masonry units fuse together. Heating mechanisms embedded within the membranes, rendering it inert and resistant to any further microbial growth. The inflatable form is removed and can be used again and again. The outer membranes can be customized, opening up sponsorship opportunities. The building can be a biohub, a home, a biofab, fabrication lab, with business incubator, a tool-lending library, and education center. This is not a house. It's an ecosystem, an economy, a community, the future. This
Welcome to Emirates, Mr. Jones. Just sit back, relax, and let us entertain you. With over 3,500 channels of entertainment, including the latest movies and box sets from around the world. We even have live sports and news channels. And your free Wi-Fi will start shortly. Enjoy your flight, Mr. Jones.
World's best in-flight entertainment. Fly Emirates. Fly better.
Back in the 1980s, a laboratory of misfits foresaw our future. Touchscreen.
Good afternoon. Welcome back. So before I hand over the chairing of the afternoon session to Shugang, I decided to introduce myself, since there may be some people who were not here in the afternoon yesterday. So I'm Magdalena Eriksson. I'm the CEO of Molecular Frontiers. And I would like to ask the students, I talked to some of you over lunch. How many here are from high schools, please? Raise your hands. Uh-huh. And how many of you are graduate students? And postdocs? And professors? Or PIs? Okay, good. Thank you. It's just good to have a sense of who's here. So Shugang, please, I hand over to you. Shugang is a board member of Molecular Frontiers, and he's a MIT, what should I say, researcher and and more than that. I think you introduce yourself maybe. Yeah, he's the treasurer of the Molecular Frontiers Foundation. Yeah, we gratefully receive donations. We can say that. Thank you. Bye bye.
All right. And the next speaker is David Kang. David has been an MIT product, and he also is a direct descendant of Confucius. So knowledge of China. So he spent years at MIT, and now he went to Lincoln Lab and came back to MIT Media Lab. He did his PhD here in the Media Lab, and undergrad MIT and PhD here, and he's an MIT product, that's why. Now he's managing something remarkable, managing hundreds called community biotechnology. So you can hear what he will tell us.
Thank you. Thank you so much, Shugang, for the introduction. Can everybody hear me okay? Yeah? If you hear me, can you say yeah? Yeah. Great, awesome. So glad to see so many high school students here. I grew up in the area as well, as Shugang said. My dad actually was a professor here at MIT, so I literally, like I was a little baby at MIT, and then I've been here my whole life, so I can't seem to leave. So it's a real pleasure and an honor to be here to speak with you all. It's one of my favorite things to do is to speak with young people, so I'm so glad that you're all here. And as Shugang mentioned, I direct an initiative here at the Media Lab called the Community Biotechnology Initiative. And so what we do is we explore the interface between very, very diverse communities from all around the world and the life sciences and biotechnology. And so, Shugang mentioned, I basically did my PhD in Shugang's lab, actually. I would not have been able to have any of the academic kind of success in my life without Shugang. So Shugang, you're always one of my dearest mentors and colleagues, so thank you so much. Yeah, thank you to Shugang. I think one thing that's really important, for all of you, I'm sure, you're students and you've got really great teachers and mentors in your life, and these are really important figures. But you also are a teacher, right? You also have people that look up to you, and so I think that's a really big, important part of life is always having great mentors, and then also always kind of paying it back and finding young people that you have experiences that you can share with them. So for me, as Shugang mentioned, I've been here at MIT for a very long time, doing a lot of work in a field called synthetic biology. How many of you heard of synthetic biology before? Hopefully all of you now, because you've heard some of the most famous synthetic biologists in the entire world over the past 2 days come and speak. But I'm also a community organizer, and I'm also a photographer and a beatboxer and a DJ. So I love the arts, and I think there's actually a really, really strong relationship between the sciences and the arts. And actually here at the Media Lab, in a big way, we kind of explore all these 4 domains of creativity. So at the Media Lab, we like to think a lot about science, art, engineering, and design. So all 4 of these are kind of different ways to be creative, and we explore all of these at the Media Lab. So I wanted to start today by asking kind of a strange question, okay? So my question is, can hip-hop save biotechnology? How many people here listen to hip-hop? Okay, a lot of you, cool. So I promise we're gonna come back to this at the end, okay? But I'll leave this here for now. And part of why I asked this question is because, where we are right now with the life sciences and biotechnology, there's a lot of different ways that it could go. And so I think there's a really important role that the arts play and that culture plays when connecting with the life sciences. So leave this question in your mind, and we'll come back to this at the end, okay? But can hip hop save biotechnology? And what do I mean by biotechnology, right? And it's interesting, you've learned and heard a lot, I think, about biotech, but very quickly, I think a lot of people, when they think about engineering the living world, I think a good reference point to always keep in mind is that human beings have been manipulating and working with the living world for like 10,000 years, right? From early agriculture to being able to select and breed animals for certain traits. And really, again, it's really kind of in the 60s into the 80s, and again, a lot of Xu Guang's work and a lot of Xu Guang's mentors were involved in some of the very early development of recombinant DNA technology. So being able to, for the first time, being able to cut, copy, edit, or modify DNA molecules. But what we've been learning about over the past 2 days, we're in this new era of synthetic biology, where you literally can design a new molecule of DNA that's never before existed, hit print on a machine called a DNA synthesizer, and then out comes a brand new molecule of DNA that's never existed before. And you can use this molecule to basically reprogram an organism and give it a new property or a new function, which is really profound if you think about it, right? Before with recombinant DNA, we're always kind of cutting and pasting from the samples that we found in nature. And now all of a sudden, you've got this magic machine that you can hit print on and create a molecule that you literally can design in a computer. So it's a very profound technology, and one of the cooler papers that came out in 2000, I highly recommend you read this. This was the first demonstration of something called a ring oscillator. So the idea here was to try and get a cell to behave like an electronic circuit would, actually, right? So to get a cell to behave like something like we see in electrical engineering. I won't get into the details of how this thing works, but basically you're making one protein that can inhibit the expression of another protein, and there are 3 of these guys on this plasmid, this circular piece of DNA. And if you tie the expression of one of these proteins with a fluorescent molecule, so as a lot of this protein gets made, then you get a fluorescent signal. What you see is that this thing actually oscillates, and it behaves like something in electronics called a ring oscillator. So this was kind of one of the very first demonstrations to show that, hey, we can actually engineer a biological system much like we can in electronics. So this is a really exciting work. And now, people build these very sophisticated called genetic circuits. So have lots and lots of different features and components. I'll explain a little bit more what this one is later, but just for now, just to say, you can do very sophisticated things in cells that behave a lot like a computer program, actually. So up here, this is a bunch of kind of a language. It's called digital Boolean logic. So it's basically saying if these 2 molecules are present and if these 3 molecules are not present above a certain threshold, then in this case, make a fluorescent red protein, okay? But you can basically program cells to do a lot of very interesting things now using this type of DNA synthesis technology. And so we're in this really kind of amazing world now where, and I love this little cartoon. This is a cell, but it's got a potentiostat and all these circuits. And we're in this kind of very interesting place where we're kind of able to merge a lot of different types of technical fields like electrical engineering and computer science with something like the life sciences and biotechnology. And this is the founder of the Media Lab, Nicholas Negroponte, who over the past number of years has been saying, biotech is the new digital. And I interpret his statement basically as reflecting the idea that what we're doing now with the life sciences is going to have as much impact, if not more impact, than what we're seeing with digital technologies, which is a big statement if you think about how every day we all carry around a very powerful supercomputer a very powerful computer in our pockets. And we're going to see something similar, I think, with biotechnology as it continues to emerge. And so what are people doing? People are making really cool types of living organisms that can act like factories, for example. So being able to turn a cell into a factory that can make fuel, molecules, new materials. And one of the really kind of exciting ways that people are doing this is through something called metabolic engineering. So again, we right now, our bodies are making all kinds of interesting molecules, and all of the bacteria in our bodies and trees, plants, all the kind of living world, there are all of these what are called metabolic pathways that can basically start from one compound, and then if you kind of follow these and have these enzymes mediate these chemical reactions, you can get to another compound. And so nature has produced this incredible array of these metabolites, but you can actually engineer that, right? So when I was in grad school here at the Media Lab at MIT, some of my very close friends were these guys and gals that created a company called Ginkgo Bioworks, which is now a very important synthetic biology company based here in Boston. And this is basically what they do. They take organisms like bacteria and yeast, and they figure out how to turn them into factories to make all kinds of really interesting molecules. So this is a really exciting big area that a lot of people are researching and working on. You can even do things like turn these genetic circuits that I was describing into a way to classify a cell type as, for example, cancer or not cancer, and then kill that cell. So this is a project that I worked on in collaboration with Professor Ron Weiss, who directs the Synthetic Biology Center here at MIT. And so we built libraries of these genetic circuits to try and basically test different types of cells to see what type of cancer they were and see if you could classify them and actually destroy them. You can even install cells inside the body, right, that can actually directly sense and respond to disease. So a lot of people are very excited about these cell-based therapeutics. Or this whole field called cellular agriculture. So how many of you have tried something called an impossible food before? I think, a bunch of you have, right? So some folks are now actually figuring out how to grow animal products in a lab without having to kill animals, right? So literally using a fermenter or a large type of a bioreactor, you can. This is actually not from a cow. This is what was actually grown in a laboratory. So there's a lot of really exciting products now that you're starting to see that connect with the life sciences, from food products to even clothing, right? So this is work from Modern Meadow from Suzanne Lee, where she's able to take these bacteria and actually grow these very, very large kind of sheets of material that are excreted from bacteria and turn them into clothing. Or even being able to make spider silk ties, right? So instead of actually having to get spider silk from the spider, you can actually engineer a process and have the silk proteins be grown in a giant vat, right, in a laboratory. So this is a lot of what's happening, and probably one of the crazier applications is something called a gene drive. Have any of you heard of a gene drive before? A few of you? So this is from my colleague Kevin Esvelt here at the Media Lab, and he's doing something really remarkable. He's figuring out how to actually engineer the ecosystem of the living world by taking a genetically engineered trait, using a technology called CRISPR-Cas9, which I think you've heard a lot about the past 2 days, and actually to drive a genetically engineered trait through a wild population of organisms. So as these mosquitoes sexually reproduce, the trait that you want gets pushed through the entire population. And that means you could do things like wipe out malaria in a certain population of mosquitoes. So literally sculpting the environment around us, which is really, really profound, right? And so one of the questions is, what are we going to do with all of this powerful technology, right? How could this all go? This is one of my favorite shows on TV. It's something called Westworld. It's probably a little bit too mature for, or maybe not, I don't know, for some of the high school kids. But it's a great show, but it's a very scary show, right? We're talking about a future where the very, very ultra elite control all of this very powerful biotechnology and artificial intelligence. And that to me is a very scary thing. But there's another version of the future that maybe looks more like this. So this is a photograph that I took from my community center in Central Square called EMW, which is actually an homage to my dad's field of research called electromagnetic wave theory. So EMW for electromagnetic wave. It also stands for East meets West. And there, we've got this amazing, diverse group of individuals that come from very diverse socioeconomic backgrounds, cultural backgrounds, creative and technical backgrounds. We have DJs, musicians, et cetera. But everybody here could also be a participant in the life sciences, right? And this, for me, I think is something really, really exciting. And so, we're talking about a technology that is literally affecting the entire living world. So how do we ensure that we have this very broad, diverse participation? Or framed another way, who gets to create? Who gets to be a scientist, right? And to me, I feel like everybody should get a chance to be the scientist, right? I think that's really, really important for science to be for everyone. And right now, biotechnology, it's in this interesting historical moment where biotech is still a pretty privileged art form, right? If you want to learn about the life sciences, like where do you go, right? Genzyme, they're probably not going to let you inside if you just showed up and asked them to use their lab. But right now, biotech happens in these pretty privileged places like biotech companies or places like MIT, which still require a lot of privilege to get into. But something started happening around 8 or 9 years ago, and people started setting up their own laboratories in some really interesting places. So this is my friend and colleague, Rob Carlson, and this is from a paper in Nature in 2010. And the headline is, the era of garage biology is upon us. Do you want to participate? So basically, the cost, and this is, Joey Ito, my boss at the Media Lab, has a saying around innovation at the edges. What happens once the cost of technology starts to get down so that lots and lots of people can start to participate? And this is a really kind of funny diagram of Rob inside his garage with all these machines, and then he's got his dryer over here next to his PCR machine. But this was a thing that started happening around 8 or 9 years ago. And a lot of people were excited about this idea of innovation happening in unconventional spaces. Does anybody know who these two guys are? Yeah? So these are two very famous Steves, Steve Jobs and Steve Wozniak. And these guys built, in large part, the first personal computers outside in their garage, right? You had a very mature industry around computation, and it took a couple of guys in a garage to kind of build what now has led to us carrying our smartphones in our pockets. So And this is my boss at the Media Lab, again, Joey. And Joey, a number of years ago, when we were getting Joey first excited about synthetic biology and the life sciences, we wanted to figure out a place where he could do experiments. And we actually couldn't find a place at MIT to do it, believe it or not. Because for Joey to do an experiment, he had no previous experience before. So we needed a professor running a very advanced research laboratory to let him come in. And even for Joey, as somebody that was new and had no training, it was a really good option, right? So at that time, and it's gotten a lot better now, there weren't a real large set of these community labs. And so we had to do this work in Joey's kitchen, actually. Right? But fortunately now, there's a number of these really amazing spaces that are being set up all around the world that are called community biology labs. So these are spaces that the public can come and actually learn about the life sciences, engage, potentially work on a startup idea or do educational programs. And so this is one of the very first ones set up in Brooklyn called GenSpace. This is BioCurious in Sunnyvale. For those of you that like hip hop, this is sort of like the Biggie and Tupac of the community labs, basically. It's like the West Coast, East Coast rivalry. But these labs are now doing really wonderful and amazing projects. So how have you heard of something called citizen science before? A few of you. So the basic idea is that everybody can be involved in science. So through these community labs, they collaborated in this case, for example, with Cornell University, and they sampled the microbial diversity in a local Superfund site. So they're looking at their local environment and sampling the microbial diversity there. And folks have been doing all kinds of really cool projects. Again, we talked a little bit about the personal computer, but people are now interested. What's the personal computer of biology, right? What is a little lab in a box that I could build? And these are all projects that, again, are not coming out of a traditional academic or corporate lab, but from these community labs. So we're starting to see these really, really neat hardware projects and kind of speculative art projects. Glowing Plant, which was a really big Kickstarter about 8 or 9 years ago, is now a very big MIT project, but it started actually, the idea came from a community lab. And so there's a really interesting thing that's happening with these distributed spaces. And now these are labs being set up all around the world in public libraries, inside hospitals. This is one of my favorites, the BioBus. This is literally a bus that travels around. You can go inside and learn about science. And this is one of the more exciting ones. So one of my former students, Benno from Peru, is building a biofab lab. He's literally making a lab, and the vision for this thing is for it to float down the Amazon River and engage with different communities along the river as it goes. So we're starting to see these really remarkable spaces. And they're also really big educational institutions. So how many have heard of the International Genetically Engineered Machines Competition, IGEM? A few of you. So this is kind of like a giant science fair for young people like yourselves, actually. There are now high school teams that can participate in this competition, so I highly recommend you check it out. Around 3,000 or more students come to Boston, to the Heinz Convention Center every year, and they share their projects. So I've been involved in iGEM since it first started in the early 2000s, but probably my most important role with iGEM is that I'm the official iGEM DJ. So I get to DJ the big party that happens every year, and it's several thousand of these crazy students. This is the only DJ gig that I have every year where I literally fear for my life, because the students are just so excited. So it's really, really fun. But that's kind of what I do, is I DJ for science nerds. So if you are a science nerd, come hire me for your function. And so this is kind of another one of the big international things. And as I mentioned a little bit more locally, and as Shugang was mentioning, my father and my mom, they actually had a Chinese language bookstore here in the Boston area called East Meets West, which stands for EMW and also electromagnetic wave. And at our space, we've also, again, started doing work in the poetry and in the arts, and then over the years, basically got into the life sciences. So we also have our own community laboratory and have been doing wonderful work teaching classes and doing workshops and having galleries. And again, it's a place where the public can actually come and learn about the life sciences. So please come. If you're interested, you can please send me an email, and I'll invite you to come to our space anytime. You'd all be very welcome. And of course, we do a lot of work with young people, getting them excited about synthetic biology and the life sciences. And so, I just want to make sure I have enough time. One of the big things that I did when I first came to the Media Lab last year was to organize this very, very big global conference, bringing together all of the folks around the world that are setting up these community labs. And so we had our very first event last year. We were expecting maybe a smaller event. We had more than 200 people show up from all around the world, from throughout Africa, Southeast Asia, Australia, that are setting up these spaces. And we had a really, really amazing event here last year. And this is with Joey and some of our other great colleagues that were participating in the event. We had all of these wonderful hands-on workshops where you could learn about CRISPR and plasmid mapping and plant tissue culture and so on. Hands-on experiences, I think, again, are a very, very critical part of learning. And of course, we had a great party, right, which I helped the DJ. And And again, George Church, who I think spoke yesterday, commented on the historic nature of last year's event as the first time this many labs had been able to be brought together and convened. And we just had our second annual event, literally it was just 2 or 3 weeks ago, right here in this exact room, and we almost doubled in size. So we had more than 400 people from around the world apply to participate. More than 350 people came, and again, more than 32 countries represented. And one thing we did was we came together and we drafted something called a statement of purpose, so something that we cared about all together as a community. And again, these are these independent community labs that are being set up. And we decided that our shared purpose is, and this is our version 3.0, we want to fundamentally transform the life sciences by democratizing biotechnology to inspire creativity and improve lives by organizing life science changemakers and bioenthusiasts to build an inclusive global network, cultivate an accessible commons of knowledge and resources, launch community labs and projects, and enable local educators. Okay, so this was, how do you guys feel about this statement? Is it pretty good? Yeah? So this is something that, again, we had a whole kind of mediated process to get to, and it was something that we were pretty proud of at the end of. So after that event, there are all of these regional events that have been organized all around the world. And last year, I had the opportunity and the privilege to go to Africa for the first time for an open science summit that was held there. So this is me with a bunch of my new friends and colleagues. And one of the things that I work on in our lab here at MIT and in my research group, we think a lot about participatory biotechnology, right? We're pretty smart. We think we're pretty smart people at MIT, but we're actually not that smart, actually. There are a lot of people way smarter than us that we need to be listening to and be very humble about. And so one thing that we've thought a lot about in our group is how do we really bring the community in a very fundamental way when we develop a new technology and make it more participatory? So working with colleagues at Harvard Business School, at Lincoln Lab, and then also with our colleagues in Ghana and Kumasi, with the photograph I just showed you, we've been developing this new participatory framework to think about biotechnology development. And it looks a little bit like this. So part of it is working with the local community to figure out what their needs are. So in the case of Kumasi and Ghana, one of the areas that came up, which I'll talk a little bit more about, is mining, okay? So once we kind of have a priority that the community cares about, we can then go and talk to a series of experts that can give us some feedback on what types of technical parameters might be useful in a device or in a system. And then one of the cool ideas here is actually to challenge communities around the world to try and make a device or a system that fits this technical description that will ultimately help this community, okay? So this is sort of a new type of community-driven process we've been developing. And again, our collaborators in Ghana really, really care about gold mining. Something like 40% of the economy in Ghana is built on gold mining, but there are really big issues with water quality, right? People are worried about contaminants from the gold mining industry affecting their water. So we've been working on developing these new technologies. Again, one thing that I work a lot and was working on with Shuang many years ago is something called microfluidics, or lab-on-a-chip technology. How many of you have heard of microfluidics before? Okay, a bunch of you. Okay, cool. But the idea is basically to take a giant biology lab and shrink it down into a chip. And again, the size scale of what we're talking about here is really small. So the width of a human hair, right, it's about 100 microns. The volume of a printed period on a piece of paper is about 1 nanoliter, 10 to the minus 12 liters. And a microliter is about a typical kind of droplet, 10 to the minus 6 liters that you might have in a pipette that you might use. And then a whole cell, a human cell here, might be about 1 femtoliter, 1 picoliter, excuse me, and then this is a kind of bacteria that have infected this cell, and these are about a femtoliter. So you can see about 1,000 times smaller each time. And so in microfluidics, we're working with devices that basically can manipulate fluid around a nanoliter, so the volume of that printed period, to volumes around the size of a human cell. And so you can do really cool things, like with a computer control how the fluids move around and mix. You can kind of mix all kinds of different fluids together and really precisely control these different biological experiments. And so part of what we did with these microfluidics, and again, with both Shuang and with Andreas, we've done a lot of this work over the years. You make this device, but a lot of times the only person that ends up using the device is the person that made it, which isn't very satisfying, right? So we built something called metafluidics. It's basically this big open source repository that lets people share all of these different types of devices that they made so other people around the world can use them. And I thought it was a pretty simple idea, but it was something that we really needed in this microfluidics community. And so now, if you go to this website, metafluidics.org, you can see all of these amazing devices that people have built from all around the world. And critically, they share all of the design files. So if you want to make your own device, you can go and look at the instructions and figure out how to make your own system. And so this is the big global community that we're challenging to try to make devices that could be useful for our friends and colleagues in Africa. And finally, okay, so this is some of our amazing global community. Oh, I'll mention this really quickly. You guys know this guy now, George, right? George was here yesterday. So George and I teach a really wonderful distributed course called How to Grow Almost Anything, which is, again, biotechnology across scales, and we teach it globally to this network of fabrication labs. So it's just another global network that we're trying to bring the life sciences to. One thing I'll mention about George that I think is pretty funny, because I love George a lot. George is going to be in a movie next year, okay? So there was a book that was written about George called Woolly. Any of you seen this book before? Well, did George talk yesterday about the Woolly Mammoth Project? Yeah? So George is trying to de-extinct the woolly mammoth. Their lab is trying to bring the woolly mammoth back from extinction. And they're making this big, big movie about George next year. It's going to be like a $100 million movie. You're going to see it all over the place. And they've got a couple actors that they're trying to figure out to play George Church. Anybody have any guesses on who's going to play George Church? Huh? Jeff Bridges, that's a good guess. Who else? You guys are so good. Yeah, so this is Jeff Bridges, who is one of the options, and the other one is literally Tom Hanks, okay? So these are the two prospective George Churches, okay? They haven't finalized this yet, but these are the two guys, okay? So watch out for this movie. It's gonna be really, really good. All right, so in closing, okay, I'll close now with the original question I asked: can hip hop save biotechnology, right? And part of why I shared those images with George too is because right now a lot of times when you hear about the life sciences or biotech, it's something like outbreak or contagion or something really kind of scary sounding. And and there is very legitimate things we have to be concerned about with safety and security. But there's so many other amazing stories about how the life sciences can change the world, and a lot of that connects with culture, right? And so through George Church's work and the book like Woolly and a movie like Woolly, we can start to see more of these types of stories. How many of you saw the movie The Martian? Okay, oh wow, that's probably the biggest hand raise so far. The Martian, what a great movie, right? Matt Damon, like on Mars, like, really using science as a positive way to change the world. And so culture is a big part of that. And again, when we go back to hip hop, right? I grew up listening to hip hop music, and I feel like hip hop is such a cultural force in our society, both in the US and around the world. How many know who this is? Okay, yeah, Childish Gambino. This is one of my favorite rappers right now. And again, Childish Gambino, I think, is doing some of the major work in our culture, raising a lot of social consciousness and awareness. And it's interesting, right? If you look at the culture, this is a great quote from Franklin Foer. He says, innovations don't magically appear or simply proceed on the basis of some scientific knowledge. The culture prods them into existence, right? So science can't happen by itself on its own. It has to happen in connection with culture and society and the arts. And so that's why I keep going back to hip hop, right? So in our community lab, we thought to ourselves, well, is there an interesting way we could combine the life sciences with hip hop? And so again, I mentioned this before. How many of you heard of the human microbiome? A number of you? So very briefly, you've got about 100 trillion of these microorganisms that are inhabiting the human body. They influence your cognition, your sleep, your development, and so on. But we asked this question, what does your microbiota sound like? What if we could actually take data about the microbiome and somehow make hip-hop music out of it, right? And so we made a project called Biota Beats, which is a microbial record player that translates data about microbes into sound and music. And so again, DJs, they can scratch vinyl records, right? So this is what I do when I DJ. But what if you could scratch a biota record, right? So this is, notice the good sterile technique here, right? What if you could scratch a biota record? And so what we did was we built this really cool system. This is actually a turntable, but it's also an incubator. So you can take these, what we call biota records, you can sample different organisms from the body, put them in this incubator, the microbes grow over time, and then we extract data about them. We image them, and we get all this data about these microorganisms, and we have an algorithm that converts that data into sound. And so this is some of our biota records. Again, these look just like EP and LP vinyl records. And then this is one of our students, Annie, who's like sampling her toes. And then we can basically inoculate these organisms, image them over time, and get a bunch of data about them, which we then convert into a MIDI file. And we have a producer in Los Angeles that converts that MIDI file into music. So do you guys want to hear some microbial music? Yeah? All right, so check it out. Here we go. So let's listen first to some feed bacteria. Can we get this a little louder? Awesome. All right. Nothing needs to be said here. Very menacing sounding music. So the belly button microbe is very ethereal. This is the sound of your armpit. Your oral microbiome. And then finally, if you put it all together, it sounds like this. do you guys think? Is that pretty cool? Yeah? Awesome. So, and I'll close here. We had an amazing group of folks that worked on this project over the last couple of years. And we thought to ourselves, right, people have vinyl record collections. What if you could have a biota record collection, right? What if you could actually sample the microbes from like a really awesome artist and make music from their bacteria? And so this, I mentioned I'm a photographer. This is a photograph that I took of Q-Tip, who's one of my all-time favorite MCs and DJs. And this is another photo I took of Questlove. And so I had the opportunity last year to speak at an event with this guy. I don't know if you know who this is. This is DJ Jazzy Jeff. This is probably beyond the era for some of the young people. You might recognize him from here. This is the Fresh Prince of Bel-Air. This is Will Smith. This is DJ Jazzy Jeff. So I was at an event with Jeff, and then I basically set up a little DIY lab in the green room. And I said, Jazzy Jeff, you're a hip hop legend. We have this project, Biota Beats. We're making music out of people's microorganisms. Like, can we sample some of your bacteria? And DJ Jazzy Jeff, he looked at me and was like, this is the weirdest thing I've ever heard, but it sounds awesome. Let's do it. So this is Jazzy Jeff, one of the hip hop legends of all time, sampling his oral microbiome. And this is probably my favorite picture I've ever taken. Look at how happy DJ Jazzy Jeff is to be inoculating some of his oral microbiota onto this biota record. And notice the good brand placement too, the reflection of the Media Lab logo in his glasses and everything. Anyway, this is one of my favorite pictures. And Jeff, just loved it. And we were working together on trying to create a little secret track for his album that could have been a biota beat and so on. But anyways, I think I'm just about out of time, so I'll end there. Thank you all so much again, and keep up the great work, learning science. Thank you.
Any questions?
Oh, questions. Oh, yeah, sure. Sorry, I apologize.
I was just wondering if there's work being done to make the sort of bio music a sort of more school accessible project to make it something that students can get into.
That's such an awesome idea. You want to work on that with me? Let's do it. Let's do it. No, seriously, it's such a great idea. We have a lot of people that want to basically do that. So because when you see a project like that, of course the question is, what does my microbiome sound like? I want to know what mine sounds like, right? So we've actually done a lot of work with students and young people on that, but one of the dreams would be to make a kit, right? Like a little thing that you could buy and just kind of play, sample the organisms and then do that at home. So let's talk. Let's work on that. Let's work on that. Other folks with questions? Oh, over here.
Hi. I was wondering what you envisioned for the community labs. What do you see them accomplishing in the future?
Oh, what a great question. I think in its most aspirational, totally change the way we do science, right? If you think about what happens right now in an academic lab or in a corporate lab, right? The unit of the currency in an academic lab is a publication. You got to publish a paper. Now, which is great, but one thing that doesn't happen when you publish a paper, you don't share all of the mistakes that you made along the way. The only thing you share is the shiny result at the end. And there's something, the National Institute of Health here in the US, they estimate there's something like a $10 billion reproducibility crisis, meaning you publish a result in a paper and people can't get that same result again, okay? And a lot of that is because of how our whole system is set up. If you want to be a professor, you have to get tenure, you have to publish all these great papers, so you're not going to share all the things the mistakes that you made and all the problems that you had to overcome and so on. The community labs, they are all sharing all of that stuff. So, because they don't care about publishing papers because they're not trying to be professors. So there's a totally different type of unit you can think of basically how do you share something and get credit for it and have other people be excited about your insight, right? So to me, there's something really exciting about the incentive structure that we have in these community labs. People just want to do great science. They don't necessarily care about making money or trying to publish a paper, right? So there's something kind of fundamentally new there that lets people do weird, crazier projects. And literally, I don't think it's an accident why this whole personal computer for biotech is coming out of these laboratories, or glowing plant, or these other kind of ideas that you wouldn't necessarily get at biological engineering at MIT. And again, I love biological engineering at MIT. I'm from MIT, but there's a certain type of constraint and a type of question you can ask in an academic environment that can be different in a community environment. So that's at its biggest, I think, is just totally changing the way we do science. Yes.
So the computer in 1950s, it was like a room size big, and now it's just a portable with you on the stage. And the biology lab is now a huge room to fit in. And do you think it's going to be possible? Like a portable size in everybody's home and can diagnose the disease all the time?
Such an awesome question. Yeah, I mean, I think that's part of the dream, right? I mean, that's what I've been working with on Andreas and Shuguang is this whole idea of being able to miniaturize a lot of these very complicated biological processes into tiny chips. And that's literally, for my PhD thesis, that's what we worked on and what I've been working on for years as well. So I think that's a big part of where the future is headed. And mobile biolabs too, something that you could carry around with you into the field. Some of my colleagues, like Manu Prakash and Stanford, their whole idea is, why are we trying to bring the backyard to the lab? Because that's literally what you do right now, is you collect a sample and you go to the lab. Why don't we bring the lab to the backyard, right? So why don't we bring like a really low-cost microscope and actually, interrogate the living world outside, right? And the smaller and more kind of compact and inexpensive the lab is, the more you can do that. So some of it's happening now. Some of it's happening right now. It depends what you're trying to do. If you want to do a full-on, metabolic engineering on a chip, that's really, really hard. Microscopy on a chip, that's a lot easier, right? So it depends on the complexity of the process you're trying to execute. Yeah, and maybe one last one just to be mindful of time.
How did you find out that the bacteria made sound?
So I should be a little clear because we had to go through it really quickly. What we did was we collected data about the bacteria. So we basically took information like the growth rate, the colony size, the density, what body part the bacteria came from, and we took all of that data and we fed that into an algorithm to create the MIDI. So one of the technical and creative challenges for us in this project is when you listen to the music, you really want to listen to the music and learn something about the microbes of your body, right? So you want to listen to it and say like, oh, okay, because it's base here, it means that like my foot bacteria are really growing strong, right? So that's sort of where, again, I would say this project is more art than science right now. There's definitely science to it, but it's very, very art. But part of making it more science would be, for example, sequencing the bacteria, right? So you know exactly what family or what species they are, right? And then you could say something more about the real details around the science behind the microbiome of your body. So that's where we're trying to move the project right now. A great question. Okay, yeah, I think we should probably let the next speaker come. I want to make sure we stay on time. But thank you all so much. This is a real honor to be here. Thank you all.
Thank you, David. That was incredibly cool. I'm loving it. I'm loving it. Thank you so much. So it is my great big pleasure to introduce Simi Foster, who is an MD PhD. You're good. Look at him. He's also a skateboarder and an acrobat. Did you just see that? So Simi's trajectory is untypical, and this is to go out to the people such as Shuguang and Michael Skuhersky, who will show his data again later on today, of people who have a approach to science that is not what you would think is normal. She went to, without having been to high school, went to Berkeley for early admittance at 16, then to Yale for her MD PhD, got her first Nature paper, first author, at 27. And I have to admit, she puts me to shame completely because she's also my wife. So originally, she is a neuroimmunologist and a practicing clinician, so she sees patients as well as does experiments in the neuroimmune system. And at first, we wanted her to talk about that. However, Simi was kind enough to prejudge all the molecular frontiers inquiry questions, and she got a brainstorm, and she got a big excitement, and she started noticing patterns about how these questions are being submitted, about what topics, about who from, and all over the planet. So I asked her to redo this whole presentation, and she's doing something that she's never done before, and it is her take on what just happened over the last year about the questions from the children and the young people who were submitting them. The point here is to understand that there's such huge, big variability from mind to mind, and there's not one great ideal way to do science. Are you ready? Yes. Okay. Simi, please take it away.
Let me just say thank you so much, Andreas, for the wonderful introduction, and to Shuguang and to all the organizers of this incredible symposium. And I really had it was such a privilege to be asked to look at all the questions this year. They were amazing. And while I was sort of going through all the questions, I went through them twice, and it occurred to me that these questions are sort of like an ocean. And the kids who are asking the questions really had sort of currents and streams and seemed to be right in the midst of what's going on in the world today. And these ocean currents had themes, and the themes that kind of came around and I could sort of identify were themes of color and time, curiosity, pain, emotion, memory, genes, gender, viruses and cancer. And as I was reading through all the questions, I started to get really excited because, one, my whole scientific career really started with a question I asked when I was a kid. And two, a lot of these themes seem to have direct relation to what I'm doing for research now. So that's how I've organized this talk. So when I was a little girl, I was really curious, and I asked my Daddy, what am I made of? And my dad said, you're a bunch of little simi's called cells. And that just immediately made a picture in my mind. I'm like, oh, I'm a bunch of little simi's. And all these little simi's had to breathe and eat and talk and poop and all that other stuff. And since then, really, when I'm doing science, I sort of think of everything a little anthropomorphically in form of stories. And so some of the way I view what I do for research owes a lot to Lewis Thomas, who is a physician and a scientist, and he wrote this wonderful book called Lives of a Cell, and also to my dad. And so as a psychiatrist, sometimes I feel like different cell types have personalities, and some even have personality disorders. For example, cancer is clearly sociopathic, right? Completely antisocial. Stem cells are very innocent, but also can be narcissistic. They're like, me, and everybody has to take care of them and help them grow, but they're also full of potential. Eukaryotes in general are really dependent, and Andreas didn't really like this, but I feel like eukaryotes and mitochondria have a maybe codependent relationship, and that's total pseudoscience, but sometimes the relationship works out really well, and sometimes it doesn't, and if the mitochondria get sick, everything gets sick, right? So they might have, in some cases, get a little dependent personality. Eukaryotes are just cells that have a nucleus and organelles like mitochondria. And then, of course, fibroblasts are obsessive totally. If you grow fibroblasts on a plate, they make a straight line. They have to, right? Because they're the things that organize the forms of our organs and our tissues. So we could similarly start thinking about a sociology of cells, a cultural anthropology of cells. But moving on, so going back to that's where my sort of scientific perspective comes from. Moving on to the questions the kids asked. So the first stream was really had to do with color and emotion. So let's take a look at these three pictures. We have, oops, let me go back. We have a bear, a flame, and a red swollen finger. And we could ask, what is the same about these three things? We have an angry bear, a fire, and something that looks really red, swollen, and we might even call it angry. And for me, the thing that's the same is the idea of defense or protection. The bear is angry because it's trying to defend itself against something. It's pushing something away. Our ancestors used fire to protect themselves from all the wild animals that were threatening them. And this red swollen finger is a sign of inflammation, which is the body's defense response. So talking about inflammation, it's the body's response to infection or to injury, and it aims to defend the body and restore homeostasis, which just means make things right again. And it's characterized by redness, swelling, pain, heat, and loss of function. And all these 5 signs were, we think, were first identified around 50 AD by Celsus. And still persist today. And you could also, if we start thinking about sociology, we could think that when we have defense responses like wars, ideally we also have a restoration phase where everything gets healed up again. But that doesn't always happen. Okay. So thinking about inflammation, really I got my start in science in my PhD studying inflammation and how it resolves. And my project was really sparked by 2 questions. So first of all, this is my was my graduate school mentor, Ruslan Medjitov, and this was his mentor, Charlie Janeway. And Charlie, when he did not want to be disturbed, would put on this red fishing hat, and that meant don't bother me. And then he also loved to fish, and so there's lots of pictures of him out on his fishing boat. And right when I started my PhD, Charlie got really sick with brain lymphoma. And one of his doctors, his physicians, while he was in the intensive care unit, asked Ruslan, is there a way to boost the immune system? So one of the ways they treat cancer and lymphoma is to give chemotherapy, which kills the cancer, but also can wipe out the part of the immune system that defends us against viruses and bacteria. It's called the adaptive immune system. And so all that remains is what we call the innate immune system. It's the immune system that's not thought to have any memory. And so the physician was asking, can we boost the innate immune system so that it can help us with the viruses and opportunistic infections that come? One of the problems is when we try to boost the innate immune system, we get this overwhelming inflammation that then kills people. So too much immunity is bad, too little is bad. How do we find a balance? And so that's really where my project came from. And we asked this question just in a cellular model, and really found that some of the small proteins that lead to this really big defense response of the body in normal situations are quickly shut off. Makes sense, right? So you don't want to be on fire and defending yourself all the time. That takes a ton of energy and it's not sustainable. So that gets shut off. But what remains inducible, what's still able to be induced, are things that are antimicrobial and that are tissue repair factors. So you try to maintain a healing response at all times, but you shut off the quick defense response. And so that was kind of shaped the way the rest of my studies, actually. So this was while I was doing my PhD. When I went back to the clinic, I started seeing a lot of people who had pain. And I started wondering about the similarities between what I'd studied for the PhD, these questions of memory and inflammation, and what they had to do with pain. And I wondered, why do we have pain? Why does it sometimes persist, even if there's not actual danger? And what's the same between immune memory, cellular memory, and persistent pain? It seems like sometimes our pain neurons just forget and they keep going when they don't need to. So why does that happen? So to start to answer these questions, after I finished my residency, it took a long time, I joined the lab of a pain neuroscientist, Clifford Wolf, and started studying pain neurons. And so I'd been viewing everything previously through this immune window. So there's immune cells here, and there's some skin and bacteria, and the immune cells are going to attack and eat the bacteria and then make an immune response. And so then I had to start learning a whole new way of looking at things through a neuron window. So I knew nothing about the brain. I had to learn about the brain and peripheral neurons. And so I kept going sort of back and forth and looking at immune cells, looking at neurons, looking at immune cells, and then I kind of realized that they're part of the same system and they work together. So the nervous system, sort of depicted on this side, really depends on immune cells for its function. So immune cells help neurons regulate blood pressure and breathing and wound repair, and neurons, especially sensory neurons and some motor neurons, help immune cells defend against bugs, bacteria, and viruses and things that want to hurt you. So they're working together in every tissue in your body. And there's this, so we call it an integrated, so they're working together, neuroimmune system. And so if we look a little bit more closely at a little piece of skin, for example, so here's some cells that are making up your skin. Here is a pain neuron or a nociceptor that's reaching out little sensors up towards the skin. Here is some bad stuff that want to get in and maybe infect you. So we have a bacterium and a worm of some sort and some little toxins. And we have an immune cell sticking out of a little protrusion. So both the neuron and the immune cell are both trying to sense these things that want to get in. And so what we find is that neurons can secrete they don't just, so these are sensory neurons, but they actually can secrete these little molecules called neuropeptides from their terminals. And these peptides act on the immune cells. They can cause, and on the vasculature. So on the vasculature, they cause some of this dilation of blood vessels, so you have redness and swelling. They cause extravasation, or immune cells to get out of the blood vessels, so we have even more inflammation. And then you also have signals that go towards the brain and cause pain. So the neurons are regulating the immune system. And what about in the opposite direction? So then we have immune cells that are secreting these little proteins called cytokines, and the cytokines talk to the nerve cells and cause more pain. So we have this reciprocal communication. The immune cells talk to the neurons talk to the immune cells, and we get both immunity and pain. So if we ask what's the same again and what's different, so what's the same between the nervous system and the immune system? They're organized kind of similarly. So they both have a sensing part that can detect harm or danger. They integrate the information. They generate a memory. Then they have an effector response, a response that does something, and that response can either do something locally in that tissue that's affected or systemically throughout the whole body. And then we ideally have resolution, where everything goes back to normal. But there's also differences. So immune cells, like why do we have both? Why don't we just have this one system? So immune cells are mobile. They can crawl throughout every place in your body. Neurons can't necessarily do that. But they're slow, so they take a long time. And they're sort of autonomous. So like a macrophage, for example, it can, whatever it wants, it'll eat a bacterium, right? A neuron has more networked, it has a little bit more responsibility. So the neurons are sort of in a hierarchy. They're more like the control system, but they have the problem that they're fixed in place. However, they're super fast. So if you have a bacteria enter at one point in your body, your neurons can send a signal everywhere and say, immune cells, be on the lookout. We had a bacterium enter, call out to try to find the right immune cell to come and take care of that bacterium. So that's why we need both aspects. However, if you go another level, things start to look the same again. So here is a pain neuron, and here is a macrophage. And if you look at how they sense the environment, they sense a lot of the same things. So they can sense dying cells, they can sense bacteria, they can sense pore-forming toxins, they can sense some of the molecules that immune cells make. But again, there are some differences. And so neurons actually have this special class of sensors that can sense temperature changes in the environment. And a lot of these receptors, there's a family of them called TRP channels, transient receptor potential channel. And if you look here, this is just a range of temperatures, and here is where different channels sort of have their maximal activation. And you can see that different of these channels, like TRPA1, sort of maximally activated really cold, like 17 degrees. TRPV1, on the other hand, is activated between 42 and 43, really, really hot. So TRPV1 is a channel that gets activated when you touch a hot stove. All your TRPV1 channels go, ah, and that's why you pull your hand back immediately. So these are important. But what you might notice is that they don't just sense temperature. They sense a bunch of other crap. So, sorry, not crap, substances. So TRPV1, for example, is the receptor that senses capsaicin, chili pepper, and that's why capsaicin tastes hot. TRPM8, on the other hand, also is activated by mint or menthol, and that's why menthol is cold. Interesting, right? So these temperature sensing channels that sense a lot of other things, including many noxious substances, turn out to be the ones that are most important in regulating the immune system. So I'm going to move on to stream 3, which has to do with temperature, cancer, and infection. And subtitle is Tales My Mother Told Me. I figured I talked about my dad. I have to talk about my mom or she's going to get jealous. So how many people have moms who say, if you don't wear your coat, you're going to catch your death of cold? Yeah, right? And I always would say back, Mom, cold doesn't cause colds, viruses cause colds. And it turns out there's always an element of truth in things that your mom tells you. temperature and immunity are actually really tightly linked. And one of the first instances that was really major in the modern era that sort of tells us this was instituted by this man, Dr. William Coley. He's known as the godfather or grandfather of cancer immunotherapy. And what he sort of observed is that patients who had a high fever and also had cancer, in some patients, their tumors would regress. And it wasn't clear whether it was the infection that caused the regression or if it was the fever. But the bacterium that caused the highest fevers tend to do the best with regressing the tumor. And so he started injecting many, many patients with mixtures of bacteria, and of course, a lot of those patients died of the infection, but some of them got better of their cancer. Now, more recently, there's this wonderful scientist, Dr. Betsy Rapaski at the Roswell Park Institute in Buffalo, and she has been doing studies just looking at comfort of mice and different temperatures and what happens to mice given tumors at different temperatures, what happens to the immune response against the tumor. And strikingly, what she found is most mice are kept at temperatures that are pretty cold for them. When she raises the temperature to a temperature that is warmer and more comfortable, she sees that if they're injected with tumor, so for example, this is lungs and lung tumors. You can see the animals that are at the lower temperature have a ton of tumor in their lungs. The animals that are at the warm temperature don't. And this correlates with more immune cells, more anti-tumor immune cells infiltrating the tumor and presumably killing it. So again, there's something about warmer temperatures that seems to fight cancer, but does it do anything else? We don't know. People get fevers all the time, but why do we have the fever? It's really not quite clear. Okay, so on the other hand, sometimes warm temperatures might be anti-inflammatory. So we're collaborating with Charles Raison, who is a psychiatrist, and he's been using hyperthermic treatment, not for cancer, but to treat depression. And we think depression is an inflammatory disease, and so in this case, he treated patients with depression with whole body hyperthermia, so we stick them basically in a sauna-type device, and he found that 60% of the patients that were treated, compared to, I think, 5% of the patients who were treated with a sham treatment, had a response to this treatment where their depression went away or was much reduced. And this lasted out to 6 weeks after a single 1-hour treatment. And so in this case, it seems like how might this be working? One way it could be working is that hyperthermia might be anti-inflammatory. So we have somewhere where hyperthermia and fever are pro-immune system and somewhere they're anti-immune system, and why they're one versus the other isn't quite clear. Okay, so moving on to stream 4, there were some questions that dealt with gender and sex. And keeping with this theme of temperature, there was this interesting article in the BBC that they actually reprinted because it was so popular, talking about office wars over temperature, right? So people working in an office, guess who wanted to turn the thermostat up? Women or men? Women, yes. Women like it hotter. Men like it cold. And so guess who wins most of the time? Men. So the office temperature average is actually a little chilly. And so there was this sort of argument, is office temperature sexist? Maybe. But temperature definitely has something to do with sex as well as with immunity. And so here, there's another interesting thing that I didn't know about before I started researching for this talk, is that turtles and a lot of other reptiles have a temperature-dependent sex determination. So female turtles tend to become female at higher temperatures and male at lower temperatures. What? Like, why? How interesting is that? So there's something about elevated temperatures and female sex, which is seems very poorly understood at the moment. I think it might have to do something with inflammation, actually. So there are also huge sex differences in inflammation, where most autoimmune disease, 80% of all autoimmune diseases happen in women. So some of the ones that you might have heard of are lupus, where there's 8 to 9 women affected for every 1 man, rheumatoid arthritis, where it's 4 to 5 to 1 man, even neuropsychiatric disorders like depression and anxiety, where there's still a 3 to 2 women to men, and then also pain, for example, migraine, and of course fibromyalgia probably has the greatest sex difference. So in my research currently, things that I'm asking that are sort of tying all this together is, what are the links between neuroimmunity, temperature, and sex? Why does elevated temperature seem to boost some immune responses and inhibit others? What's the difference between neurons and immune cells and how they sense and respond to temperature changes? So, lastly, to make this short and sweet, so Andreas has often said that there's a statement that being at MIT is like drinking from a fire hydrant. And the image that I got after reading all these questions especially is that being a human with a capacity for curiosity is sort of like navigating in the ocean. And sometimes you get lost or you go in circles, you don't know where you are. Sometimes you join like a really cool stream and you end up finding an island that you never knew existed. And you find things that are so cool on this island. So I would encourage you to please share with me your adventures as you're navigating, and I would love to hear your questions. And lastly, let me just give acknowledgments to all of my mentors and collaborators at Boston Children's Hospital. Dr. Wolf has been my mentor for the past 4 years, and then all of the Wolf Lab members, including wonderful undergraduates and students and postdocs. Mass General Hospital, the Depression Clinical Research Program, I mentored Dr. Maurizio Fava, Dr. Mara Nair and David Michelin, all the staff. And then I'm part of the Building Interdisciplinary Research Careers in Women's Health that's really studying sex differences with Jill Goldstein and Ursula Kaiser and all the Birch Fellows, and other mentors, Stuart Calderwood, colleagues Sebastian Talbot and Andreas Merschen, and my funding. So thank you so much. I really appreciate your time. And I'll take your questions.
Oh, quick. Is it possible to like psychologically convince yourself to like selectively take away pain, basically?
Amazing question. Such a good question. Short answer is yes. Yes, through many different ways. There's a recent paper that showed that there actually can be we call cortical inhibitory control, which means that your thinking brain can actually suppress some of the pain signals that are coming from the periphery. And even sometimes you might not have pain signals coming from your like arms and legs and body, but you still feel pain. What's going on there? Your mind can still control that. The question is, how do we access that? And there's probably several different ways. Hypnosis might be one. Biofeedback might be another. And there's also medications, which obviously have problems with them.
Do you have any thoughts on, I don't know if you're aware of Wim Hof, this guy from Amsterdam who can supposedly control his autonomic nervous system through breathing. I was just wondering what you think about that.
Doesn't he also go in ice cold water baths and swim in the like icy ocean? Yes, so the conscious control of your autonomic nervous system is fascinating, and we know it can happen. It hasn't been studied that much. A lot of the studies were done in the 70s and 80s. So interestingly, so there's this sort of conscious control, and I think there's also probably modulation by temperature, like cold, of some of your autonomic processes that are going through the vagus nerve, for example. And so you can consciously change your breathing pattern and then perhaps stimulate your vagus nerve and thereby start regulating some of like heart rate, lung function, etc.
So do you know, so the difference between males and females and how the, like fibromyalgia, how those diseases affect women more than men, do you currently know the reasoning behind that, or do you think it has something to do with like body fat or something?
Well, it's such a good question. It hasn't been very well studied. So my hypothesis, something that I was interested in looking at, was that I told you how the immune system and the nervous system are talking to each other. I'm wondering if in women they talk to each other more, so that generates better immunity in some cases, but then anytime you have a lot of communication, there's chances for it to break. So I think maybe it breaks more in women. Now, if you look at more details, there's ideas that, well, some of the immune genes are on the X chromosome, so you have double the dose, for example, of TLR7, right? So then you have double the chance of recognizing DNA in a bad way. There's also hormonal influences, so estrogen and progesterone, depending on the levels, can have either immunosuppressive or immunostimulatory properties, so they either boost your immune system or inhibit it, but all of that really is still, we're still trying to figure out how it works. It's an excellent question.
Okay, final question.
I was wondering, excellent talk. I really enjoyed it. I was wondering if is there a census or some sort of information of, for example, cancer rates in places of the world where the weather is warmer, for example, versus colder?
Yeah, that is awesome question. I actually haven't looked into that directly. My sense was that it was a little bit hard to tease out because a lot of places that are warmer also tend to have more other things that could modify your cancer risk. For example, they tend to be more social and talk to each other more. Their diets are a little bit different, so maybe less bad fats, etc. So there's a lot of confounding factors, but that would be I'm not sure if anyone has done that study, but if not, it would be a great study to do. Thank you so much.
So I will introduce the next speaker. How many of you have heard James Bond? You're almost looking at a live one today. So Andreas Merchant, and he fly airplane, he dive, he climbing, he speak many languages, and he drinks. So he is the one word describe James Bond is the word fearless. So this man shares the same thing, fearless. It's the same thing for people in the media life. That's why I make so many inventions because they are fearless. So he's trained as a theoretical physicist at Imperial College, London. He was born in Moscow, grew up in Athens, and did his undergrad in Imperial College. And by the way, His Majesty was the Queen of England and met him because something he did. And his teacher is sitting right there, Elizabeth Ball. And he was inspired by the Benoit Mandelbrot, the fractal person. You can see. So he will tell you all interesting stories what has been done in the last 15 years. Okay.
Okay. Well, thank you, Shuguang, for this impossible to follow introduction. So everything from now on will be downhill. Questions are important, and while you're watching me online or here in the audience, and if you want to submit a question to me or to the rest of the people who are watching the stream, please go to rebelminds.org, and it can be anonymous. We don't care who, if you write your name, whatever. We just want your questions because they're super valuable. Now, this slide describes some of the things that my lab does. And it's, to some people, this is very threatening because it appears that there's no coherence here, and it's a big mess. And we're talking about bioinformatics and putting noses inside of cell phones to diagnose cancer, and making biophotovoltaics, which means solar power out of waste, agricultural wastes. We're talking about making, isolating genomes and transplanting them whole without breaking them. We're talking about microfluidics that solve a $100 million problem with a 1-cent device. Chris Love, who is here, stand up please, there you go, stand up. He and I, in the early days, created a system that alerts firefighters to incipient fires by harvesting the power of trees themselves. With Simi, who just spoke, and Thras, we accidentally created the world's first pain sensor in trying to do something entirely different. We're using an EEG headband to measure people's brainwaves because we wanted to figure out if I could reproduce my non-ADD state that I enter when I'm flying my plane. And we learned that that was hard, but to prove that the algorithm was working, we wanted to do something simple. So our expertise was not was in pain. Simi's expertise was in pain. My expertise was in electronics and other things such as signal processing. So we figured that we actually, if we could just generate pain in people and be able to determine that, then we'd be on our way to proving our algorithm works. That's not the lesson. The lesson is that it took me 2 years to understand that we've actually invented the world's first pain detector. We didn't realize that that was not a stepping stone. That was actually a cool thing to do. So that's there, and now it's become these glasses. And it's no longer horrible, and it's portable, and you can have a readout not of only just your pain, but many other conditions, such as you've seen, in a wearable format. And of course, where it's going is you're going to have VR, AR, and mixed reality in your glasses. With Kate Adamal and Philippos, who is here. Philippos, stand up. Where is he? He's here somewhere. He gave you the lab tour a second ago. I hope he'll be back. And James Pelletier, who created microfluidics for isolation of genomes, for compaction of genomes, for creating synthesia. We did this using a direct write excimer laser. Unlike every other microfluidic architecture and method that you have, this allows you to literally write something on a napkin, generate a device, and iterate 3 times per day. This cuts down on the cost of exploration. It makes failure cheap. And early and good failure is absolutely key to science and technology and other things. We built the world's most sensitive nose, bioelectronic nose, without actually knowing how noses work. The whole project started with us thinking the nose is an analytical tool. We thought that when you sniff something, whether you're a dog or a human, what you do is, and then you get a list of molecules by name and concentration, as if you're in the Matrix movie and you're watching the code, and if you watch the code real good, then you can see a human bird. No. So that ends up being not the case. However, we did build the nose using a lot of DARPA money, using the same receptors that exist in nature called GPCRs that you've heard a lot about. And we learned so much in building it that we actually came off with this motto, build it to understand it, which I took from Feynman, stole it, and transformed it, because Feynman told us, if you can't create something, you don't understand it. So we did create a photosynthetic solar cell, and we understood a lot about photosynthesis. We understood that one of the biggest mistakes, at least that I had my entire life, I thought that photosynthesis works, the energy for photosynthesis comes from the h-nu, energy of the photon. I thought that blue photons had more energy than red photons because that's what my physics told me. It ends up not being true. Photosynthesis actually works on the number of photons per unit time per square foot. So that was a key understanding shift, a shift in perspective that allowed us to do several patents and millions of dollars worth of research. And guess where that came from? About $5 million worth of research came from Vlad Leperts, who's also here somewhere. Here you go, stand up please. He, 10 years ago, asked one of the inaugural questions of the Molecular Frontiers Inquiry Prize. He asked, why aren't plants black? It's a much better question than asking why are plants green. Because the thinking is, and you can ask him how he thought about it, I'm paraphrasing. He said, well, look, they've had 3-point-something billion years to evolve. How come they don't eat the green photons? How come they only eat the red and the blue? It ends up being that's a very deep question. It allowed us to understand photosynthesis on a deeper level than we had before. And that led us to understand how GPCRs and other membrane proteins operate. How can it be possible that solar power on one end, photosynthesis, and sensing molecules can be in the same lab? Not only in the same lab, they're actually under the same roof, and most of this work is done by a few people that are multi-talented. They're multi-talented because they came from the outside the field. Once you're inside the field, you're like a sheep. You're happily grazing in the field. You're not going to leave. No scientific revolution has ever happened from inside the field. Once there's a field, that's it. The scientific revolutions come from the outside. Think of the people we revere in science. Newton. How many different things? Gravity, light, mathematics, calculus, geometry, alchemy. Think of Einstein, photoelectric effect, general relativity, quantum physics, special relativity, etc. These are people who look at the world from many different perspectives, and they attack problems, like Shuguang said, fearlessly. Now, I'll tell you one story about Shuguang now that I wasn't planning to, but I think it's great. There were early days, we were doing this project with the photovoltaics, and we'd given it to someone, and that someone was a very diligent physicist, and they told us after about 6 weeks of reviewing the literature that what we're about to do is breaks all the laws of physics, and we should stop. And I was convinced. The person did their homework, they presented a very coherent argument, and then Shuguang went under the table and gave the same project to an undergrad who didn't know anything about anything. 2 weeks later, that undergrad had it working. His name is Patrick Kelly. He went to England afterwards, and what he said was, I asked him, how do you do this? And he had missed that meeting where we determined it was impossible, so he told me, well, I didn't know it was impossible, I'm so sorry. I have heard that phrase here at MIT so many times. Not knowing what's impossible is actually quite good. So what does my lab specialize in? It specializes in NOT. That's why it's called the MIT Label-Free Research Group. I'm a physicist, sure, all my degrees are in physics, a PhD, etc. But if I called myself a physicist, then most people wouldn't come talk to me. And if I called my group, I don't know, the machine olfaction group, the one that makes noses, or by the biosolar group, that's all I'd be able to do. Labels block your vision. Labels are not useful. They're shortcuts sometimes, but labels can be damaging. So, label-free. Now, another important thing about science is you must not fool yourself. And I will go through a bunch of errors that I've made and what those taught us, because like David said, most of the time scientists like to present the finished product and how we got there, not the actual way that we got there. Most of the time, what you see is the finished product, not the blooper reel. The blooper reel is an important thing to know how it was done. These are some of the very, very wonderful people that work with me and have tolerated my ADD. So with the J. Craig Venter Institute, we created microfluidics that are aimed at transplanting whole genomes. With Kate Adamala and George Church and Vincent, we actually just sent the my first experiment ever into space, which was done with, again, under the table with almost no budget, and you'll see some results in a second. With Simi, we, amongst other things, invented the world's first pain detector, which was helped also by Thras. Oz here and Michael have been helping with the noses. James and Alex have been helping with the microfluidics. All these people are doing a lot of things that you wouldn't expect them from their label. This is an example of actual data of us lysing, that means exploding cells. What you're seeing is about sped up 10 times. Each one of these is about 1.2 microns in diameter. These are mycoplasma cells that we can explode. And if we had the right filters, I'd be able to show you this is a movie too, but we didn't. So these are individual isolated genomes that haven't been blown up. So they are completely intact, and we can stuff them into another cell, which we did here. This led us to think of the bio-internet. With George Hirsch and Kate Adamala, we are thinking about how we can use 3D printing kind of ideas to 3D print living matter. Imagine you're going to Mars, and you want a spanner and a hammer. But you can't take both because it's heavy. What you do is you have a 3D printer that makes your spanner when you need it. You use it, you stuff it back into the 3D printer, melt it, and make a hammer. Tuck, tuck, tuck. OK, can we do that with life? Sure. So long as we can digitize it and transmit information that allows you to assemble genes in order, let's say, then you can do it. So we have designs, and we've patented this too, and we're about to go and do it. With Philippos, who is still not here. Hope is he here? No? Well, he gave you the tour earlier. Here's our postdoc. He's here. He was a fluid dynamicist for his PhD, and he went into an office building and created a BL2 lab. That means a biological level containment 2. Using this machine, he can print, and that's why we hired him to be a postdoc here. He can print fibers. Whoops, that's not good. He can print fibers. Oh, that's not good at all. Well, thank you, Microsoft. All right, let's see if we'll... So while this is rebooting, I'll tell you that Philippos created these fibers that are so tiny that they actually can reproduce the position of individual cells. That means we can have control over them in sub-micron resolution. Why sub-micron? Well, because cells in your body are about a micron. And what is known now, and in part because of this work, is that if you manage to create the scaffold of an organ, and all organs have a scaffold, you can get, we're one slide over, I almost hit it right. But anyway, let's keep it this way. So you can get what's called localization, and you can get cell differentiation by their geometry. This is something that I actually started thinking when I was 16 years old with Liz Ball as my biology teacher. The question is, every cell in your body has the same DNA. As you're developing, though, some cells become eyeballs and some cells become teeth. And how come you never get confused? How come you never grow teeth in your eyeballs? That would be very terrible. Right? So even though they have the same DNA, they're running different codes, they're running different schedules. So how the hell does this happen? Many, many theories. It's been a good question in science, really powerful question. Lots of work has happened. And one of the things that we never thought is that there's signals that come from the geometry. So the cells know what their neighbors are, and that's how they know how to differentiate. This brings me to this. So this is David Lai, who's a super achiever scientist at one of the world's biggest pharmas, GlaxoSmithKline. And I asked him to share a personal story because I want to show you how you can do great science and be very successful in it, but also be a normal human being. So I asked him to share a story, and he said, well, I guess my story is that I've played Settlers of Catan hundreds of times, and I've never won. And that's statistically impossible. So he created this microfluidic device for making encapsulation of drugs. Like Bob Langer was talking about yesterday, the slow release of certain drugs is important. And in this case, we had the slow release of an HIV drug that is hydrophilic. And if you don't do it in perfectly formed spheres, then you have all sorts of issues. So the solution was to actually go into space and try it in microgravity. And my message to you here is that it took this young person, Nicholas Benz, who's probably not a couple of years older than the people in this audience. And he led this project. And he's at NASA. He was an intern at NASA. And yet he led this project with dozens of professors and others, including myself. And we launched this thing on June 21st. And it broke during lunch. But we learned so much, including a funny thing that we didn't know it was possible to contract with NASA, including the fact that space has become the go-to place, and it's cheap, and it's easy to get to. And there's plenty to be learned from there, including about really simple fundamental physics that we thought we knew. I want to remind you that when NASA went to the moon, the average age of their engineers was 28. This is a mistake. It's not 29. 28 years old. I looked it up. OK, that should tell you something. We haven't gone to the moon since then, by the way. So the other thing I learned is that you can stay up until 5 in the morning to watch this launch, which I did. And then as a physicist, I should have known that 25 Gs means that you stay up all night to see this. That's all that happened. Like, what happened? Where's my rocket? It's gone. That's how fast it went. So it all started when Shugong hired me to fix this problem, which was how to make a more efficient biophotovoltaic, which is, again, a membrane protein that sits in a membrane of a cell. And Shugong had developed these peptides that can be stuffed in there like corks and keep it alive. This became very famous. He went to the Boston Museum of Science. And the problem was that it wasn't very efficient because it was a monolayer. So I was traveling, and I saw this forest in Japan, and I took a picture, and I realized that this tree is cheating with respect to its brothers and sisters. When photons come from the top, and they don't get eaten by the canopy, this guy's got some leaves down the trunk, and the other guys don't. So we decided we're going to make a nanoforest. We're using carbon nanotubes from Cambridge, and functionalized with these membrane proteins. Now, can you tell me what's wrong with that idea? It was a failure. Carbon nanotubes are very, very black. They're actually the blackest thing we have so far. There's even a company that makes Vanta Black, which is so absorbent that if you wear a shirt with this thing, people's eyes kind of get drawn to it, and it's like looking into a black hole. It's really crazy. So the problem there was really black. But the idea was good. So what's the next best idea? Well, if black's no good, let's make them transparent. So we created the periodic table of zinc oxide nanowires, which gave us all sorts of interesting features. And those are not only transparent, they're actually light capturing. So the sources for this biophotovoltaic can be anywhere, including seaweed. And that, believe it or not, is Shubang. So when scuba diving in 8-degree water to get seaweed. And now you'll be a little shocked because in the same breath, I'll take one breath and I'll talk about noses. In the same breath. How can noses and biophotovoltaics be connected? How come I have all these things on this poster? Well, look at here. We learned how to put this protein on top of this nanowire and create an electronic circuit with it. We learned the same thing, the same stabilization protocol, and the same nanowire physics to put 7 transmembrane domain instead of this humongous PS1 and created the world's most sensitive bioelectronic nose ever. It beat the dog's limit of detection by a factor of 200, by the way, during DARPA testing, and that was some hardcore testing. They wouldn't even allow us to touch the machine because they were so afraid that we're going to sniff the various molecules, and having learned, because our noses are very sensitive, that we were going to cheat. So they had everything behind glass. We weren't allowed to touch anything, and still we determined that we beat the dog by a factor of 200 at their limit of detection. That means that essentially we can, or not essentially, actually true, in every case, these molecules detect single molecules. Now, there's something else that we learned, which is this. Structure equal function is a mantra that's overplayed and needs nuance. It is true that structure is extremely useful. It's not true that you need it all the time. And what structure you take, at what level of abstraction, what layer of understanding, is really important. We've divided it into two extremes. The one extreme, we have what I call the mousetrap proteins. There, if you see the structure in an X-ray crystallography experiment or other technique that gives you where every atom is, you might be able to figure out how it works. For instance, the mousetrap, if you see this diagram, with and without the mouse in it, if you know a little bit about what mice are, you might be able to figure out how it works and figure out the trigger and all that. If you transition through these various other types of proteins, you get all the way to the worst of this example, which is the GPCRs. And the worst of the GPCRs are in nilfaction because everything binds to everything, and everything's labeled, and everything changes. And that I call the accordion model. And the accordion model means, first of all, it needs to be moving to work. That's why it's so hard to crystallize them. And secondly, if I play you a piece of music on an accordion, and I'm an alien, I'm trying to figure out what the accordion does to humans, some humans, in some audiences, the same piece of music will make you laugh and sometimes will make you cry. Why? Well, suppose I'm playing the battle song of your enemy that reminds you of the battle you lost. That's going to make you sad if you were on the losing side, make you happy if you're on the winning side. That is context, and that is lacking from our understanding of the lock and key. If you're still unconvinced, this is what happens with olfaction, and it's also 80% of all pharmaceuticals that work this way. These molecules diffuse into the hydrophobic pocket, and this is the important thing. Not only is everything vibrating, not only is everything at 37C, so forget about having any kind of rigid lock and key, it's also the case that upon observing, upon detection, upon interaction with the environment, you change the environment. And that came to me as a surprise, and I should have paid attention more in high school, because the only thing that really, truly, truly, very, very hardcore true, I think, in physics, everything else might be up for examination, is Newton's third law, which is the same as the Heisenberg uncertainty principle, and from another point of view, it's the same as the Gödel incompleteness theorem. What this means is this: you cannot act on the world. You can only interact with the world. Sensing a molecule changes the molecule. Sensing light changes that light. Your reaction to that stimulus is not actually written on the stimulus. Sure enough, there are some pretty hardcore, hardwired reactions. For instance, I actually have my two experimental apparatuses here, Petra and Sol, who I will present when we do the prize in a minute. But one day I was changing my son's diaper, who was about 1 at the time, and Petra walks in, and she says, "What's that smell, Daddy?" My instinct was to start talking about indolence, cattle, and the microbiome and the volatile organic components, and how there's an emergent. Okay. Then I took a breath, smelled it some more. "What's that smell, Daddy?" Every instinct in my body was to point to the smell. "What's that smell, Daddy?" So I had a moment of clarity, and I said, "What does that smell tell you?" And Petra, if she remembers, she told me, "I don't want to eat it." Oh, that's interesting. That's a pre-wired firmware installed response to an aversive smell. I learned something from, she was 3 at the time. So, beat that, whoever has a 2-year-old, please. Now, some myths. Dogs are better at smell than humans. To a certain extent, yes, they can smell things we can't. They have a larger epithelium. However, that is a dog in the night looking for a dead pheasant. That is, I think, a Caltech student looking for chocolate blindfolded. You can see qualitatively it's not that big of a difference. The real difference is this. Dogs are easier to train, and they can diagnose cancer and other things. They can find bombs. And humans are harder to train. And yeah, there's some things that our thresholds are too high for. But the real reason why we don't use humans for these tasks is because, ask yourselves, how much money would you need to be paid to get on all fours and do this all day long at the airport, smelling every piece of luggage? It doesn't work. Dogs are easier to motivate. Another myth that you've heard, and please don't believe it, is all these 3 headlines. Humans can smell 10,000 scents. That's in every biology textbook. It's complete and utter nonsense. It has never been counted, and it can't even be counted. It's like saying, how many objects can you recognize? Well, how many pencils can you learn to recognize? It's not a question that admits a numerical answer. How much time do you need to spend on recognizing all these things? And the other thing that you'll see is this picture, that this is a lock and key combinatorial thing. All of those end up being non-correct. We learned all this after we built a nose, we didn't need to know how I thought we needed to know how the nose worked to build it, but it ended up that building it made us understand how it works. All these molecules are shaped differently. They all smell of musk. Why do they all smell of musk? Well, they bind to different receptors, and somehow nature decided all of these should smell the same. Now, why do they smell of musk? Why do flowers smell of musk? Why do flowers smell of flowers? Why do fruits smell of sort of like flowers? Why does perfume smell like flowers? Because in all cases, something is the same. What's the same? The function is the same. Not the function from the structure, not the function from the receptors. All of these molecules are the product of biosynthetic pathways that can be in the deer's antlers, or the anal glands of the civet cat, or the flower. Totally different pathways. Make totally different molecules. They all, on some level, mean the same thing. In all cases, they have the same function. That is to attract your attention. To attract the attention of a pollinator. To attract the attention such that you can procreate. So we train flies, just like dogs don't know any chemistry. Flies don't know any nuclear physics. And we train them to identify isotopes. And we published in PNAS on the same day. Our work was highlighted in both Nature and Science. So that's some kind of record. And that led us to actually make this nose. You've seen it downstairs, I think, during the tour. It was about this big. And it worked. And one of the big lessons here was that I built it using what I knew from CERN. Because I used to be a theoretical physicist, a high energy physicist. I kept thinking about accelerators. I've climbed all over CERN inside the tunnels before they was built and all that. And I built it as a circle. There's tubes of molecules flying around, and this bay makes a preliminary determination and tells the next bay via this wire what experiment to do based on the result of the first bay. I thought that was a hack. I thought that was something to be embarrassed about because I had a lot of money, had a lot of people, but I did not have enough time to put a supercomputer under this and train it to recognize every single molecule that DARPA wanted us to find. So I built this system to hierarchically prune the data, thinking, let me get the money, I'll fix it later. It ended up being that actually, years later, it was discovered by other people that that's actually most likely the fundamental code of information encoding, olfactory or otherwise, in brains, including they proved it for Drosophila and honeybees, and it's probably the same for all animals. So next came this. Next came more millions of dollars and more years of shrinking this and shrinking it. Now, why am I shrinking it? I'm shrinking it because I want to put it inside your cell phone. I want to put it inside your cell phone because it's already got an eyeball or two. It's already got an ear. And it needs to have a nose, too, because it can smell you day in, day out, learn what your normal smell is. And for some cancers, for instance, detecting it early is the same as curing it. If you detect your melanoma or a normal mole becoming cancerous and becoming malignant in the first 6 months, you go to the doctor's office, they remove it in a 30-minute procedure, you go home, and you don't have to think about it again. If you miss that window, it could be a death sentence. The disparity of outcome from early detection versus late detection is so vast that I changed the entire lab over to do this, to get inspired by the dogs who can do this. And we have seen that the dogs now, after 14 years of exhaustive research, peer-reviewed publications, lots and lots of statistics, that dogs are the earliest, most sensitive, least false positives, least false negatives cancer detector we've ever seen. And not only that, they can alert for seizures. They can alert for epileptic seizures, for diabetic shock, and many other things. But the main thing is their versatility. And to understand it, consider this. You train a dog on a flower, a rose, let's say. If you then take this dog to the city, the context is the city. That's something no analytical tool understands. And you tell the dog, seek. What will it find you is the flower store, even if the flower store has no roses. If you take that same dog with the same training to a flower store, and you say seek, it understands that what you want now is the rose. This salience of zooming in and out is absolutely fundamental. We do not understand how they do it. We don't understand exactly what they're latching on. We know they're not latching on individual molecules, but we know that the signal rides on the molecules. So that's the mission. Now, this mission is so compelling that it actually got us all the way to the Queen. That's me showing the Queen my nose, which I have somewhere here in my pocket. Yeah, there it is. So the nose that I was showing you that kept shrinking is now this. So it's almost there for your cell phone. And on top of that, we now have phones such as this one, which is $1,000 currently. I'm maybe the third person who has it. This nose has a volatile organic compound sensor and a FLIR camera. It doesn't tell you what something smells of, but it does tell you how many ppbs there are. And it's the beginning of the noses in your phones. It's $1,000 today. In a year, it'll be 500, etc. You know how that works. Now, some interesting things about meeting the queen. The dogs that have been trained by a fantastic charity in the UK, which is truly a jewel in the crown of British science. They're a scrappy charity. They survive on donations. They're led by this lady, Dr. Claire Guest, who's a behavioral psychologist of dogs and other animals. And they've been training dogs for 20 years now to basically save lives every day. If you have POTS, it's a syndrome that makes you fall down unpredictably. Your eyeballs roll into your head. You lose consciousness. You can die. You can't drive. Your life is pretty terrible. These dogs trained will save you every time. They will alert you 5 minutes early. And there's no way that we have no other way of doing it. If you have an epileptic child, you have to wake up and poke them every couple of hours. That destroys your family life. That destroys your productivity. The dog will alert even from a different floor. The dog can be on the ground floor. The child can be on the second floor. They will alert. And interestingly, in New Zealand, where this is the money for New Zealand, this is $1 New Zealand dollar. On one side, it has the queen. On the other side, it has the kiwi bird. New Zealand is a fantastic country. It's powerful scientifically. It's the real world Wakanda. Nobody knows about it. And if you go there, they have a space program. They have fantastic scientists. They have technologies that we haven't even heard of here. They're just quietly better than us, I think. And they don't want anybody going there. But this is their money. So I went to New Zealand after I met the queen. And I was trying to brag about this. And I don't care about that at all. And on the other side, they have the kiwi bird. And I tried to meet the kiwi bird. Now, there's only one Queen of England. There's 70,000 kiwi birds. Getting to meet one of those was extremely hard. We didn't actually manage. It's easier to meet the queen than the kiwi bird. Now, this is the team that we just got funded by the Prostate Cancer Foundation to understand exactly what is it that the dogs are picking up on. I'll show you here how the dogs are being trained. And while you're looking at this, I'll talk a little bit and say that, of course, the signal rides on the volatiles. We did the controls. Those are easy. A blind dog or a dog on a different floor can still detect it. So it is by nose. It's not by sound because we did the deaf dogs too. What are they picking up on? This is an ion chromatograph from a GCMS, and it shows you the various peaks. Somewhere here, the dog sees no cancer, and somewhere here, the dog sees cancer. We don't know what it's looking at. But what we do know is if you change this to this, which is a list of volatiles, nothing changes. The information is still here. From here to here, it's a step that doubles the complexity. It doubles the cost of analysis. But the dogs are certainly not doing this. They have no idea what lansoprazole is. Okay? They're not seeing molecules. And the training happens with the easiest device in the world. The training happens by associating a signal with another signal in time doing this. That's it. That's it. That's the training thing. Now, after doing this for a few weeks, these dogs become so sensitive that we can't fool them. Oftentimes, they actually detect cancer in urine that we use here that we totally selected to be clean, to be the negative control. And the people say, oh my God, this dog is alerting on my urine. And we have had this several times now that these people had early stage cancer, much earlier than anything the hospital could pick up on. So to conclude, your questions are very valuable. Intelligence is the ability to adapt to change, said Stephen Hawking. And Einstein told us that if his life depended on it, he'd be looking for the right questions. So the question that we have today is, what are the better questions? So please give them to me because they're very valuable. Submit them here. Submit them to the competition if you're under 19 or 18, as the case may be. And please tell me what you think about all this. Thank you.
So this might be a silly question, but so going back to your daughter who she smelled something and she said, okay, that makes me not want to eat it. So I don't know.
Well, we know what she smelled. It was a diaper.
So dogs don't exactly do that. Like my dog will smell a toad and my dog has picked up the toad, which is bad because the toad's poisonous. Why do they not, like they're so advanced in the way that they can smell cancer, why can they not do that for things that...
That's an amazingly good question. That's part of what we're trying to figure out. I'll one-up you on this. I don't know the answer, but I'll one-up you. There is this completely anecdotal thing, but it happens to be real. I don't know if it's relevant. Dogs and Tasmanian devils are two of only 3 animals, I forget what the third one is, that have a transmissible form of cancer. It's called canine transmissible venereal tumor. And what happens there is super interesting. The tumor itself is a different animal. if a human has a tumor, a cancer tumor, the DNA of the tumor is the same as the DNA of your cells. But this is a completely different thing that grows in the genitalia of dogs, and it compels, when it's emitting odorants, which are the molecules that have scent, it compels the dogs to put their noses and their snouts on top of each other's genitalia. And that's how it gets transmitted. And if you track its genome across the planet, you can see it change and evolve and adapt in different dogs, and we can tell when it came to the New World, etc. Now, it could be that, because it's true that dogs spontaneously will sometimes alert to cancer even without any training. And maybe that's why. Even more peculiar, and this is really what made me change my entire life around and say, I need to put dogs into cell phones. Everything else doesn't matter, because this is going to save lives, and it's so compelling, and we're going to learn so much science. Here's what happens with dogs. You train them, let's say, on urine to detect bladder cancer. And you get these things to be perfect. By the way, we've also trained humans not to detect some diseases such as Parkinson's with 100% success. But back to the dog. You'll see how it connects. You train them to detect bladder cancer in urine, and they do very, very well. And then we'll start spontaneously, let's say, and we have plenty of cases, poking somebody in the breast. And Claire guessed her life was saved like this, because a dog that was trained to detect bladder cancer from urine was poking her in the breast and detected a very early tumor, a very early nodule that would have not been found with a mammogram. Doctors are on record saying she would have been dead by now. She talks about this in her book, which is called Daisy's Gift. That was the name of the dog. That dog was not trained on breast cancer, and if you look at the volatilum, which is an idiotic word that unfortunately exists, but it means the collection of all the volatiles that are coming off of the two cancers. If you suck off all the volatiles that are coming off the breast, off the skin, or even off the tumor itself, and you suck off all the volatiles that are coming off the urine, and you compare them, like in that diagram that I showed, you'll see not a single one alike. The dogs managed to generalize somehow. They see cancer as musky. You see, all those molecules were different, but they all smelled of musk, right? So they're operating on a level where they see it as a sensation. It's like, this is cancery, this isn't cancery. With training, you can actually make them become more and more precise. We can now, the Prostate Cancer Foundation has funded us to solve a very concrete problem, which is determine of the 17 different types of prostate cancer, there's 12 types that treating them aggressively actually may cause you to lose quality of life or even die. And 5, if you don't treat them, you're going to die of them. Of the 12 that you don't need to treat, because you're probably going to die of something else, how do you determine which ones you should treat and which ones you shouldn't? They all give you the same PSA. They all give you the same kind of normal information. So we're training the dogs now to draw this line, and we can do it. That will save, we estimate, about 300,000 unnecessary procedures just in the United States alone. So that's the kind of questions that we're asking. But my answer to you short way, I don't know. Yeah. Another question, I think, and then we have to... You can send your questions online too, remember?
So you said that in order to understand the system, you wanted to build it to gain a better understanding of it. And I'm wondering how you even start approaching a problem in constructing some kind of...
Excellent question. For that one, I do have the answer.
Because you don't have any...
I got you, man. I got you. It's a good question.
The world, right?
I have the answer for you. Because that's an excellent question. I've been thinking about it. So how do you start? How do you start if you don't know? I mean, we lucked out, kind of. I mean, we had a lot of money and a lot of pressure, and we weren't thinking quite straight, and we just went and did it, and then we realized, oh my God, we accidentally invented all these things. Now, how you start is design the experience, don't design the features. Let me say it again. Design the experience. What is the experience of training the dog? The experience of training the dog is, hello, doggy. Here, smell this. Where's my clicker? Ha, there it is. Here's the dog, and here's the clicker. And that's how you start. You design it. You pretend that you have it already. This nose doesn't work. Obviously, it's not plugged in. And the clicker works. If this was a dog, I would go here. I would go here. Nothing. I would go here. I would go here. Nothing. Oops, sorry. Here I clicked it again. So now the dog thinks that this and this have something in common. If you try and make a machine that has the same experience as training the dog, that you would have the same experience, and the machine would just be this on a leash, then you'd get a nose. Or you'd get something else that does the same thing. Design the experience. Look at what you want to replicate and design the experience. Do not design the features. We thought we needed an analytical tool. And we couldn't quite get the features. We couldn't quite design it this way. So we kind of did it, you know, on a very big budget, but on a very short time. So the answer to how you start is think about what it feels like, think about what the experience is, design the experience, not the features. Next, do we have time for one more, or can I should welcome the next speaker? Oh, Natalia?
So A few years ago, Google launched the Google Nose on April Fool's Day. So is there a way of making that a reality in the same way that you can store images and audio? Can you store smell?
It's a great question. I love it. In fact, why don't you join the group already? We have a position open now for exactly solving this problem. How do you visualize the olfactory information so it makes sense to the human? Or how do you audioify it? Or how do you tactile it? I don't know. The point is, we humans, and I've tried, I think we may have skipped over a little bit of a disgusting picture of me trying to train myself on smelling urine. And I'll tell you this, I can definitely tell that the urine is from a sick person because it makes me gag. It is very intense. I can't tell you what type of cancer they have and all that, the dogs can. And I can't tell you whether they're medicated or unmedicated or whether I'm smelling the medication. The dogs can separate all these things. So definitely with most humans, and we're not sure actually that it's for all humans, because even with vision, there's women, only women, there are tetrachromats, like European starlings, which are birds that can also see the magnetic field. About 1 in 1,000 women have that. That's a huge number. There's probably one here in the room. And you wouldn't know about it because of the way they would communicate about color and things. It's very difficult to realize that some people see a lot more colors than you do. For the longest time, I didn't know I had a problem with my color vision. And famously, what color is my phone? Huh? Pink? Yeah, right? No? It's gray to me until I put the right glasses. This looks great to me. So I have a certain type of color blindness. My green receptor links into my red receptor. I put on some glasses where we designed them with a notch filter, kill the green, and now I can see, well, when I'm wearing them, I can see pink. This looks great to me. So even with humans, we have variability. For all we know there's one human, at least one human, who we found through the dog work, and she lives in England, and she told people that she can detect Parkinson's disease because her husband tragically died of Parkinson's, and she associated a change in his body scent with the diagnosis. She was unlucky enough to have this. Basically, something very emotional happened. Her husband was diagnosed with a deadly disease, and she, at the same roughly time, detected a change in his body odor. Now, she does happen to have a very well-trained nose. She was always very interested in cooking, things like this. She has never been fooled when scientists give her T-shirts, in white T-shirts that people have slept in a plastic bag. She has a 100% success rate sniffing them. If you do the volatilum, you never find the same odorants. So she's doing the same thing as the dogs. Most humans don't sniff. I can't sniff the Parkinson's level, whatever it is. I tried to train myself. I can definitely tell with a 100% success rate, from not from urine, but from neck swabs, whether you're medicated for Parkinson's. But nobody takes medicines for Parkinson's unless they have it, so we can't do that control, and I can't tell unmedicated Parkinson's. There might be other humans that can do it. One of the questions is, how do we amplify this sense, or how do we maybe change them to another modality, a color, a scheme, or something else? So thank you for that question, and yeah, please, you saw what Dave did with the microbiome. We would like something like this for the scent thing, to bring it to a sense that we can understand. Okay, are you introducing the next one?
Actually, you are introducing.
I'm introducing the next one, okay. Thank you. So it is, again, the greatest pleasure to introduce Debbie Donnelly, who is a professor, a researcher, a pilot, and an integral part of the Molecular Frontiers Foundation, and she has brought all of you here, and she has been so good at providing us with access to kids. And she'll tell you that her incredible story of being not only one of the first female captains on a plane, but also how she went into research and how we're now finding out things about pilot performance. Thank you, Debbie.
Thank you very much.
Oh, I'll hold it.
Me here this afternoon. It's an honor to be here. And also, my presentation will be a little bit outside of the box, as we've been hearing throughout the symposium, and seeing different types of scientists that are here. I'd like to ask you all a question. How many of you look to the sky when you hear the roar of a jet airplane overhead? I know we have a few pilots in the room, and even if you're not a pilot, I know that entices many people to find out curiosity about aerodynamics and how does that airplane get up in the sky. We all know the basic principles of aerodynamics. This here is the Boeing 767, and I'm privileged to fly that aircraft. And that's the inside of the cockpit. We'll get into details in a little bit later about this. But for many centuries, man has had his curiosity piqued with the interest of flight. Back with Leonardo da Vinci in the 1400s with his flying machine concepts, all the way up to 1903 with the Wright brothers and their Wright Flyer. In today's present day, we actually have a pilot shortage. This data is from Boeing and their 20-year forecast that over the next 20 years, we are going to need almost 800,000 pilots worldwide. That's a huge shortage, and there's many factors why this is occurring, but now is the time to get into aerospace. It's a different kind of science that we've been hearing about from the molecular science today, but it's also a very interesting science. And another statistic, this is from Women in Aviation worldwide, there's only 4% of the pilots in the world are females. And we're trying to grow that number, but it's been pretty steady for over the past 50 years. We're trying to research and find out why there's not more females in aviation. It's a great career. It's interesting. And out of those female pilots, there's only 0.5% are female captains. So again, would like to try to increase that number. I'm just going to give you a little background about how I got started. Sometimes people know their path in life. They're born to do a certain career. But other times it takes a little bit to find your path. You may think you're going one way, but sometimes we get diverted. My path wasn't very clear initially. I wasn't on an airplane for the first time until 1987 when I was 15 years old. And that sounds pretty ironic from a pilot. I'm sure most of you have all been in an airplane at this time. I became a Rotary Youth Exchange student to Brazil when I was 15 years old. So this all started because of my curiosity. For the first time I was ever on an airplane and actually away from home. I grew up in Connecticut in a small town. And I had to learn a new language, Portuguese, which I didn't find out until 4 weeks before where I was going. So it all happened pretty fast. But since then, my Portuguese is still fluent, and we have lifelong friends and family from this experience. But this experience actually changed my life, literally. I would not be here today if I didn't embark on this experience. I knew that I wanted to see new cultures, meet new people, and explore the horizons. So when I graduated from high school, I was 17 years old. And when I turned 18, while I was going to college, I became a flight attendant. So I know it's an unconventional path. I started in the back of the airplane. But guess what got me to the front? Curiosity. I just started asking questions. Within a few weeks, I was on board. I said, how do you become a pilot? I didn't know any pilots, and I came from a relatively small town. So I learned little by little that you can just go to the airport and take a flight. You did not have to be in the military. And with that, my curiosity was piqued. About a month after I became a flight attendant, I started my flight lessons. And there's a picture of my first taste of flight, 18 years old. And that's also in the small planes. That's a Piper Cherokee. I would take my friends and family on small flights out to Cape Cod and areas around here just to build up the flight hours. So from that time when I was 18 years old, it took me 4 years while I was going to college. I stayed working as a flight attendant and studying. And soon I got my first commercial job as a first officer for the same company that I was a flight attendant for. I was literally in the back one day. The next day I went to training, and 6 weeks later I was in the front. It surprised quite a few people when they came on board. But I really enjoyed the job, and that was out of Newark, New Jersey. So I did that job for 4 years, and while I was there, I moved my way up to become a captain, and I also became a check airman and a pilot instructor. So I was actually teaching and training the pilots that I was once in the back as a flight attendant for. So you never know who will be your boss. Be nice to everybody. And now the current plane that I fly for UPS is the Boeing 767. 408,000 pounds is the max takeoff weight. Again, we said the aerodynamics help it get airborne, but it's a miracle that we have this technology today, and it's just getting better and better. I'm privileged to fly this aircraft. I've been with UPS for 20 years flying. And this is the inside of our new cockpit. We're upgrading our instruments with new technology now. So we have continuous education that we have to go through and training at the same time. the dimensions of the aircraft. And I also am very fortunate in my position to get out and mentor young people. I really enjoy volunteering and giving back to the community because I had a lot of people who helped me and encouraged me throughout the way. I'm involved with Women in Aviation, and every year I volunteer at the conference with UPS, and we've had for the past 5 years over 10,000 people in attendance. And also going to schools, talking to students. So it's nice to get the outreach and get out there because sometimes people aren't exposed to these different types of science and different types of careers. I was also very fortunate to win the Pilot of the Year award in 2013 and very honored to be among my colleagues there. So it's been quite a blessing to be at UPS. And I also have the privilege to fly to many interesting places around the world. I fly mostly to South America and Central America from Miami. So we get to see a lot of interesting volcanoes and geography. 8,000 feet is the field elevation in Quito. So there's a lot of different performance issues that we have to figure out for the aircraft. And now I'm moving into the future of where my path has taken me. Sometimes, like I said, it's unexpected. As I continued my education through my bachelor's, master's, and most recently doctorate degree, I've gotten involved with Harvard University, and we've been doing research on the environmental effects that affect the pilots from the cockpit. There's many factors, including air quality, and also ozone, radiation. And they've been unstudied for many years due to many factors. The cabin has been studied, but the cockpit has been more difficult also due to the FAA regulations. So we completed our first study, which was the largest pilot health study in 2015. We had 3,000 pilots from around the world participate in our study. And this led us to our second study, which was more last year, and we studied the effects of carbon dioxide on pilot performance. We were able to get out to the pilot community and raise these issues that they have not been aware of. So it's very interesting making a difference in the community and especially for the future pilots. We were able to publish a paper on depression and suicide from our first study, the pilot health study, which is a really important topic. I know one of our talks yesterday talked about the importance of raising awareness for depression and suicide. And even in the pilot population, we've had a few accidents in the past 5 years that are related to this. So we would like to bring more awareness and get the pilots the help they may need without feeling ashamed. And our second study we recently published was about the effects of carbon dioxide on pilots. Many times we don't know that performance can be degraded from different factors, such as air quality. So our research is continuing to look at these different issues and how they affect pilots, which ultimately affects safety and the safety of every one of you in this room. This is just a diagram of the airplane ventilation and how the air is mixed and recirculated, and that's how some of these issues come about. This was some of the carbon dioxide levels that we found elevated, mostly during boarding is when we found that this takes effect. So we are still looking into how long these will stay in the cabin and also affect the pilots. And I also continue my education. This is the Aviation Health Conference in London, which I attend every year, in addition to my pilot education, which every 6 months we go in the flight simulator and we practice emergency procedures. So it's ongoing education also. This is the flight simulator, and this is the actual flight simulator that we used for our study. That's a picture from the study. And these are some of the issues that we are still studying and that are up and coming. And we have much more to study. There has been a disconnect that we found with pilots, between the pilots, the manufacturers, the engineers, and the mechanics. Sometimes the pilots may be aware of a situation, but the manufacturers may not be aware how it's affecting the pilots or vice versa. So we would really like to get out and educate people, the population, the manufacturers, about these issues that are out there and have everybody come together and bring more communication to this issue. This is my team at Harvard University that we've been working on, Aviation for Health, for the airplane studies. And we are starting a third study on the effects of radiation on the DNA of pilots. That's another picture of the aircraft. And I recently celebrated my 20th anniversary with UPS. It goes by really fast, but I've enjoyed every bit of it. And I know somebody else had said, if you do something you love, you're not going to work, you're going to play. And that's how I feel every day when I do my job. I really enjoy it. And I also just have to add that my daughter Therese is here in the audience. She's 17 years old and recently got her pilot's license also. So I'm really proud to say that she's following in my footsteps. And I just have one last slide. Another honor that I was asked to do for UPS was to fly the historic all-female flight crew in the Thunder over Louisville Air Show. We did that in 2017. And it was really exciting making history and flying the air show, and especially being part of the first female crew ever. So little by little, we are trying to get out there and change the world, make it a better place, educate, mentor, and do what we can while enjoying it at the same time. And I'd like to thank you again for your time, and I hope we're still on schedule. Thank you.
Good. MIT should look you up.
Hello, we were just curious of what was the process of just getting your pilot's license?
That's a great question. The process is there's different certificates and licenses for you to obtain. The first one is your private pilot certificate. And actually, Andres, our previous speaker, is also a private pilot. But the minimum hours that are required are 40 hours per the FAA. And you can just go to any local airport. There's a lot of small airports around. And they have what's called an introductory flight. So you can just go and ask them to do an introductory flight. It's pretty low cost. And they'll take you up and actually let you fly for about 45 minutes. And then you can sign up and take your lessons if you'd like. There's a ground portion and a flight portion. Just like a driver's license. You have an examination with an examiner at the end where you must complete all the required maneuvers, and then you'll have your license. And also a medical certificate.
Me? Okay. Hi, that was a great talk. So
Thank you.
I have a question regarding the ozone. So we know that ozone reaches all the way down to the surface level, hence why you need to replace your tires because they crack, whatever. So what's the ozone exposure to the pilots compared to somebody who's hiking in the White Mountains, say?
Well, the aircrafts typically fly between 35,000 and 40,000 feet. So the ozone is quite elevated. The mountains, you're not there for usually an extended amount of exposure time. So and it's much lower altitude usually, unless you're hiking some of the highest mountains. So you are exposed for longer periods of time and at higher altitudes. So your exposure rates are higher, and also the symptoms would be more. Some aircraft are fitted with the catalytic converters, which help scrub out that ozone. However, they need to be maintained and updated because as their useful life goes, also the quality of the air will diminish. Thanks. Hi.
Hello. This is a very passionate and inspiring talk, and but the question I'm going to ask might be the worst question for a pilot because it's out of curiosity. At the age of AI, we are like a lot of companies are starting autonomous driving. So I was just wondering if it's possible, like autonomous flying in the future, so that pilot could be like sit back and relax and just...
That's a great question. And yes, actually the testing is going on right now as we speak. Boeing, Airbus, NASA are testing it. It is possible. However, we think that this generation of pilots that are just starting out now to start this pilot shortage will have a job for at least the next 30 years. And we see single pilot operation coming first before totally autonomous flying. And also, unfortunately, it would hit hit the cargo industry first, and then it would be tested probably and move on to the passenger industry, which may take 20 years or longer. It could speed up, but we think there's at least one more generation of pilots before they go away, because it's not these aircraft that they will be flying autonomously. They're new aircraft that are just being not even developed yet.
Hi.
Hi.
I have another question. So, how does the ozone exposure and all of the carbon dioxide and all the factors that the pilots are exposed to, how does that correlate to the depression?
Well, there's many factors that we don't know yet if they correlate together or not. The depression, we found that they have the depression levels of the general population, more or less. But there are factors that contribute, such as circadian rhythm disruption, when you're switching your body clock back and forth, because then you're more fatigued. And when you're more fatigued, we all know some people get tired and crabby. And this is an ongoing cycle, and it could lead to alcohol, drug addiction, and depression. So there's many factors. It's not directly correlated, but when you have a combination of them put together. Thank you.
Good. We take a 20 minutes break and come back at 4. Now, before we disperse, there's still a lot of lunch left. Before you go home, please take them. Otherwise, we all throw in the garbage, unfortunately. So take as many as you can, and we'll clean up the table. Okay? So, good.
come to my office, I'll show them to you. Okay, so thanks. You see, that's great too. Hello, hello. Okay, so we're starting a little early for the Molecular Frontiers Inquiry Prize Award, and we're going to have a bunch of presentations, so please, those of you who are talking, come close. I guess some of them are still preparing. We are extremely happy this time because two fantastic things have happened. We have a whole cohort of past winners who are going to announce this year's winners. And we actually happen to luck out this year, and we have one of the winners in the audience. Now, we don't have the prize to give them. We'll show them what it's going to be like, but I'm very proud to point to Felix. Felix Nelson, please stand up. He's one of the winners, and we'll announce his question in a minute. And good job, good job. All right, so I'm going to go very quickly through this. Felix, come show your face here and tell us what your question was.
Which one?
Well, he doesn't know which one is the one. Does everybody remember how many did you do? Tell us about all your questions.
My first question was about quantum mechanics, because if there's like quantum mechanics and there's like physics, shouldn't there be like a bigger scale of like solar system or even like galaxy-wide physics? That was my first question. My second one was how did like early life begin on Earth, and why?
There's some early life for you.
I think that might have been all I did. Yeah, I think that.
How life comes from non-life.
Oh yes, yeah, that was exactly.
That keeps winning. Thank you. We'll point to it again. So let me quickly set up the prize and say thank you again, Nelson, and say that. We determined the questions are important earlier. Einstein has told us that he has no special talent. And let me just tell you that it's a question that gave you the internet. The internet is the result of CERN. CERN happened because Democritus and Lekypos were standing on a beach in Greece, asked the question, and they said, if I take two rocks and I bang them together, I get smaller rocks. If I mash those together, I get sand. If I grind the sand, I get dust. Does this go on forever? Doesn't matter. Go on forever being divisible, or does it stop at some point? This gave us the word atom, which in Greek means that which cannot be cut. Of course, we knew that as soon as we gave it the word, we cut it. So the word is incorrect. But how come the internet came out of there? Well, because Sir Tim Berners-Lee invented HTML, hypertext markup language. And that was because he was too tired of going to the library and photocopying papers, physics papers, because this machine, world's most complex machine that we've ever created, has generated so much data, it was hard to to go to the library and photocopy all these papers. So he invented hypertext markup language, which had this ability to link, and you could click and you go places. If you understand what that means, great. If you don't, think of it as a biological evolutionary event in human history. What Tim Berners-Lee did was not just give you the ability to go on Amazon, he created the ribosome. The first browser, the Mosaic browser, interprets HTML no matter where you are. And it gave us the ability to have one code that will be interpreted correctly or incorrectly sometimes, depending on the browser. You have exactly the same as we have one gene. You can translate from one organism to another. And the ribosome sometimes can fold it correctly, sometimes it needs chaperones, etc. That was a biological level event for humanity. We have a nervous system of sensors and actuators called the internet, and the internet went on fire because of HTML, because of that one invention, because of CERN. The internet infrastructure existed before. I was on the internet back in the early 90s and late 80s, and it was all text-based, etc. The browser is what made it. Now, we say at Molecular Frontiers that the Inquiry Prize is because we want to judge not only a man, but also a woman and anybody else in between by their questions, not their answers. And I'm done for today. I'm going to introduce Liz Ball, and I'm going to show you my kids as I leave, because they are the ones who taught me a very important lesson I just described, which is if you ask the question, and if you ask the question back sometimes, you get a lot of information. They taught me the difference between semiochemistry and perception. Now, I want to tell you 1 second about Liz. Liz, please stand up. Liz is my teacher from high school, and she was brave enough to take on a project with me when everybody else said, I can't possibly do mathematics and physics and biology as my final project. How can you possibly combine those three? Well, Liz said, why not? Same as Shu Guang's mentor. So Liz is a powerful force in my life. I am so grateful to have her. Please bring me my kids here so I can say. And then we pass it on to Liz. Please, Petra, do you have a question for the audience? I promise she can be on stage. Babe, do you have a question? Do you have a question?
My turn.
No?
My turn. Okay. My rocket. Rocket.
Okay. Okay. Now, Liz, please come up. And I want to thank the sponsors of the Molecular Frontiers in CRISPR. Please rise. Liu Yatsen and Chao Hali from Puget Sound West. Thank you for buying the prizes. And thank you for empowering this. And Liz will now tell you about the prize. Thank you.
Thank you very much, Andreas. Well, before we get to the prize itself, I'd like to just explain where I came in with the Molecular Frontiers, because you could say that these questions have arisen spontaneously, beautifully. And it's true that in order to be a good scientist, you need to be able to ask good questions. But for a number of years, we have been concerned with the disparity between the incredible science that we've heard about in this last couple of days and the general public. How can they see, how can they understand the beauty, the magnificence of asking the right questions and hopefully sometimes finding solutions? So especially to children, how can we talk about science with the passion and share with them and get them to share with us? So the way that we've set about this, rising to this challenge, has been by asking questions. And I've been fortunate enough to be able to ask questions at schools, and I'd like to explain to you today a little of what I've done and seen at schools in Greece, in Athens, where I teach, and show several of the successful projects, and perhaps ask you in the audience to share with me and all of us your experiences too. So just going back a little in time, I realized that young children really didn't have the vocabulary. They didn't have the words. They had the concepts, but not necessarily the words to ask the right questions, the right scientific questions. So something that's interested me for some time, and back in Gothenburg Symposium of Molecular Frontiers, we had a project where students would ask questions through art. And this is just one of the posters that we produced where we linked together children's questions with the talks of that symposium, and more questions were generated through the symposium, and so it became one question leading to another. So through this success, the students at school enjoyed. They were able to put down on paper or with sculptures their questions. But clearly, we should give them more opportunities, not just through art. So at my school, the Laureates School in Athens, it's a traditional Greek school which has a strong reputation in mathematics, in science, in classics, literature. But they were mostly in their boxes. How could we ask the students to ask questions that were deeper about these subjects, and how could we get them to ask questions that were meaningful for all of us? Well, some of you have probably asked students questions and then regretted asking. And when we asked the whole school, they asked, is it necessary nowadays to study in a school? That's actually quite a deep question for the moment, and this is what is on students' minds. And of course, we would like to say that the answer is not only. So through the school, at all levels, from the kindergarten all the way through to the senior school, many projects have been developed by teachers, dedicated teachers, and I'd like to just show you a few of them. In the kindergarten, going to the local Mount Parnitha, the questions were limitless. The children were so excited. They were observing. They couldn't understand necessarily why leaves were brown, but they could certainly ask, why do leaves go brown? Can I grow a tree? That tree which is so strong and can support me, how is it so strong? Why doesn't it bend over? Picking leaves from the floor, not quite sure whether these are leaves of a tree. Why don't I have those? Why can't I feed myself? Why don't I have those leaves? Or perhaps they were, through the play, they were antlers. Why do we not have antlers? Many, many questions. As Andreas was asking about smell, all of the different herbs that could be collected on the mountainside. Why does this one smell like this and another smell like that? So these are kindergarten students, and they are asking those questions. If we are listening, we can hear those questions and encourage the development of their ideas and their further questions. So if we go through to primary school, of course, the skills start to develop, and so students will have a topic to consider: water, the most valuable molecule on the planet, without which we wouldn't even have life. So what were the students curious about? And if I just show this picture, sadly you can't see their faces, but you can see the enthusiasm there. They have drawn their questions, and they want you to see them, and they are holding them, and every single child wanted to be at the front so that you could see their questions, which included: how does water go up to the sky? How does a ship float? And these are at primary level, way before physics has started. So around the same time, in literature, in English classes, these students, their native language is Greek, so as they learn English, they are not skilled in the language to form a good question, but they have their questions. So through art and through putting their ideas together, seeing how one question can lead to another, one thought can lead to another, in a line for the moment, but obviously as going through the school, the complexity increases, becoming a web with the interrelationships. And the students are so proud to talk about their observations, what they have seen and their questions. And if you throw it over to them so that they can then ask you, you enter a dialogue, and they are just so much more involved and so much more interested. So obviously other lessons are more, let's say they are more constructive in the technology aspects. How do sensors see, for example, or how can we go faster? But some of the questions at this age can start to become a little self-reflective. Can my robot see me move? And throughout all of this, the students are gaining skills in collaboration. They work together. They're excited. They want to hear, and they want to share, and they want to move forward, and they are just so involved in the lesson. Obviously, as life in the biology lab cannot be seen easily without the use of microscope, the first times that we see down the microscope offer a really exciting opportunity for students to ask new questions. And the teacher here has a box in her room, a little like the urn, Professor Norden, that asks questions. These questions actually came out of the urn. How exactly did the first cells appear? How did the human brain evolve? Why do we exist? So they're already asking big questions. Which on school trips, again turning rather than explaining, if you ask questions, the students became more involved, and rather than just visiting, they would, for example, ask questions: why is there so much pollution? Why is the plastic not degradable? How can we help? And so you start getting greater involvement in community activities, which could be run by the school or outside the school. At our senior school, for many years, the physics group have had the opportunity to go to CERN. It was a small group, very dedicated teachers and students who went, but it remained a small group until we started inviting the artists. Once the artists had been, other people wanted to go as well. We have developed the CERN trip from 12. We have over 40 students wanting to go yearly, annually, and the questions that they have started to ask envelop not only science, music, literature, medicine, and business. So by involving students from different, let's say, areas, their questions were enriched as well. For example, since particles do not die, why do humans? What is empty space? Will we be able to manipulate time in the future? These are from non-scientists. So, what I'd like you to take from this, perhaps, is that in a way we are all able to help young children asking questions by listening to them, by encouraging them to ask more questions. They will become more balanced in their outlook, and even if they are not themselves wanting to become scientists, they will be asking the questions that are important for all of us. And through this, they will learn to take risks. They will learn to have fun, be passionate about what they believe in and their own personal goals and questions. And they will hopefully do that together in a team with mentors, with teachers, as the wonderful scientists who've talked to us today have shown us, can be so inspiring for all of us. So I hope that's given you just a quick look at the kinds of ways that we can encourage, all of us, we can encourage more questions. But I'd like to thank all of the people at school who allowed me to explain from their points of view how they have encouraged questions. But in a way, I would like to thank all of you now, because the other side of asking questions is you have all been listening. Thank you so, so much.
Thank you for that, Liz, so much. And I'd like to introduce Natalia now. Please come up, and I'll tell you a little story about her. She is Liz's daughter, and as an organization, we try to be very honest, and we follow the rules as closely and as strictly and as fairly as we can to make sure that we do the good job of selecting the winners. Now, Natalia was in the first cohort of people who played, but we said up front that she cannot possibly win because she's related to one of the organizers. My kids can never win either. Now, what happened though was that we let her go through the process, and the Nobel laureates selected her questions, and she'll tell you what they were. And then Aaron Klug, who is a famous Nobel Prize winner, didn't see her name in the winners. And he came to me and he started telling me, how come not? That was the best question. And I had to say, sir, I'm so sorry, but you know what happened was we had to give her, we had to eliminate her because she's related. And he was very upset. So we ended up actually having to give her a prize under the table that is not announced anywhere, but she is by all rights also a winner. So she's going to introduce past winners who'll come up here and read the questions of this year's winners, including Felix's winning question. And by the way, Felix, your other questions may have won too, but the rules are that you can play as many times as you want, but you can only win once per lifetime. So please play again. And we actually had another person who would have won this year, and we will send them an email later, that didn't win because they've already won once. And thank you, you know who you are, the people who have played before and they've submitted again. For all you know, you could have won again. Thank you for submitting questions. Natalia, please come up. And you go, where are you? Here. Here? Yeah. Awesome. Hold up, just a sec. Let's get rid of this, and you can start. Oh, really? Just a sec. Okay, can you start talking while I do this?
So thank you, Andreas. I suppose this illustrates the time when actually genetics is and inheritance can be a disadvantage. So, or as Vlad called me, I was the ghost winner. So thank you so much for inviting me. It's a true honor to be sharing my story with you today. Don't worry about the slide, because
I can get it. Oh, if I can only go here. There we go. There it is. Thank you, Microsoft, for, as always, providing an exceptionally terrible experience.
Right. So, like Andrea said, my name's Natalia. I'm from Greece, and I first heard of Molecular Frontiers back in 2008. And I still remember the first question that I asked in the science blog. Why does hair turn grey? So that question led to a conversation about aging and stem cells and telomeres, and each answer seemed to generate more questions. And I found that really exciting. And then the same day, a few days later, I found out that my thread on the blog had the most views on the blog, and I think I found that equally exciting. So the following year, I was a finalist, like Andrea said, in the Molecular Frontiers Inquiry Prize, with my favorite question, why is water wet? And for the first time ever, I was awarded for asking too many questions. A rather annoying trait of mine, as I'm sure some of my old school teachers would say. So 10 years down the line, and I'm now a qualified medical doctor in the UK. I practice in a small town up north called Lancaster, where I studied medicine. And I applied to medicine because, well, I felt it was a really good idea. I loved human biology, and I enjoyed interacting with people. And I was really looking forward to meeting like-minded people like myself. But if I'm perfectly honest, I was slightly disappointed. Students were largely disinterested in anything that wouldn't come up in our exams. The competitive nature and just the sheer amounts of the volume of information that we had to learn in a short period of time meant that I didn't really have the time to do what I loved doing best, which was asking why. So I found that quite challenging at the time. But then, as time went on, I found out that I was learning drugs. I was learning to prescribe drugs to people without really understanding why I was prescribing them. I didn't. Sometimes doctors don't even know how some drugs work, and I was matching names of these diseases with these weird and wonderful names to case presentations. But like Richard Feynman once said, knowing the name of a bird tells you nothing about the bird. And I very soon found out that medicine goes far beyond any textbook. So as a qualified doctor, even within the highly hierarchical and very protocol-driven healthcare system, as I'm sure some people can relate in the US as well, I found that those slightly annoying and forgotten traits were beginning to be recognized once again. I currently work in my psychiatric rotation, so when we finish med school, we rotate through different placements. And I was asked to take blood from this patient who suffered from severe psychotic depression. He was flailing his arms all over the place. He was really agitated and upset, and we needed 2 people to sort of try and calm him down and pin him down as I was hovering this needle above his arm. And then I just thought to myself, why am I doing this? So I put the needle down and I ran outside and I tried to find my consultant, and I asked him the question, and he said, actually, you know what, that's a good question. It's not really a necessary test at this moment in time. So we didn't proceed with the blood test, and the patient was fine. Another lady was brought into the emergency department. She had bathed herself and ingested chlorine. She was labeled as being suicidal, and she was referred straight to the psychiatric team. And nobody had asked one simple question: why did you do this? It turns out this lady wasn't suicidal at all. She couldn't read and write, and she suffered from mild learning disability. It turns out she drank chlorine and bathed herself in chlorine because she'd been raped, and she wanted to cleanse herself. So immediately you've gone from a patient who will go down the suicidal, depressive route to actually a patient who needs the appropriate support for sexual abuse. So even though these are not particularly difficult questions or philosophical questions, they make a big impact on people's lives, and they make a big difference to my practice. So that's mainly the main message that I want to. Is anybody interested in medicine or would like to apply for medicine? No? Yeah, maybe. So it's slightly different in the US. Obviously, you have to do a first you have to do pre-med degree and then another degree, whereas in the UK we go straight to it. But anyway, so that's one of the main questions that I learned, asking questions and asking why. But actually one of the other important things that I learned, and it goes way back to that first question I asked at the Molecular Frontiers blog, why does hair turn grey, or actually why does hair turn white? It was that I shouldn't be afraid to say, I don't know. So patients ask me questions all the time, and it feels very uncomfortable to say that I don't know. Sometimes I don't know, sometimes nobody knows. But it's the same in science, right? And as doctors are always expected to know the answers to all questions, when actually our job is all about asking the right questions. So I wanted to thank Molecular Frontiers for instilling those important lessons right from the start, being curious, not being afraid to say no, and for once making me the cool kid for winning an iPod at a science blog. Thank you. So now the time we've all been waiting for, actually announcing the winners for the 2018. So I'd like to ask Vlad Lepard, who was a co-winner, to announce the first winners.
Not a ghost winner. So, okay, thank you for that. And okay, so I just want to give a quick background and just express my gratitude to Molecular Frontiers really quick. Andres already covered my question earlier. He said that I won with the question of why aren't plants black? And it's quite funny because I asked it on a whim, and I remember submitting 5 different questions, and I felt least connected to that one, especially because biology isn't really an interest of mine whatsoever. My degree is in geology, and I don't really like dealing with living things, but, you know, we each have our own interests, and I have to say, because of this question, I was able to really get involved with the MIT community. I got to work with some great people like Andreas and Shugwang over here, who believed in me all these years and let me move around from one lab into another, from biotech to physics to whatever else. And now I've ended up working, doing research in aeronautics and astronautics here at MIT, which is one of the biggest airspace programs we have in the world, I suppose. And we're studying contrails, working with NASA, working on supersonic flight, so that's pretty exciting, right? Yeah. So some cool stuff. And now I'm thinking about getting a law degree. I don't know. So we'll see where that goes. But anyway, so moving on to, anyway, thank you guys so much. Andreas, Shugwan, the Molecular Frontiers organization, I really thank you. It changed my life, and now MIT is my home, whether I like it or not. So moving on to this year's winners. So we sort of split these up into different themes, and we tried to sort of give them appropriate titles. And for this one, we labeled it as Sensations, Emotions, and AI Driving the Thoughts of Tomorrow. So the first question we have is, how does the brain connect the tone of the voice with the inherent meaning and intended mood of the expression? When did this develop? This was asked by Kylie Long, 14 years old, from the US. So, fantastic question. Moving on. The next one was, could it be possible to insert images and colors into the mind of a blind person? And this was asked by Araceli Franco, 13, US. Makes you wonder, huh? And the last question I have on this slide is, how do dopamine and serotonin make a person feel emotions? Could AI systems feel emotions the same way we do? And this was asked by Ayush Araya, 17, from India. Again, thank you so much, and I'm going to pass on the mic. Who's, oh, Romy, if you please.
There we go. Magic fingers.
Do I talk about my question? Okay. So my name is Romy Rosenbaum, and my question was, is the evolution of humans completed? Or are there going to be further advancements in the human race? I didn't put much thought into it. We just did a big unit on evolution, and we didn't really talk about human evolution. So that was the first question that came to mind about the competition. So that's what I turned in. So yeah, asking questions is good. So the theme for these questions were thoughts on origin, life, and ideas of tomorrow. So the first one is, how did life form from non-life matter? Felix Nelson, 11 years old, from the US. Next one is, where did the first DNA molecule come from, and what was the first molecule containing DNA which reproduced to create life today? By Melanie Guerra, 16, from the US. And the last one on this slide is there a more stable molecule for storing information than DNA that could be used in cells? That is by Samuel Paulini, 18, from Slovakia. And thank you, and
hand it off. Thank you. So my name is Latoya Willis. I am a sophomore at FAU High School in Boca Raton, Florida, and my question was, how can consciousness be measured if not through introspection? So I remember my teacher, Ms. Barkis, or at least the person who would be my teacher next semester, sent out an email to every student at FAU High School about this competition, which I'd never heard anything about, considering that I was really competitive at the time. So I thought the only thing that I need to do is just come up with one question of all the questions I've ever asked. So I knew that I was really busy that week. I had an essay to finish by Sunday and just a lot of things to do, especially my psychology test. And I never actually read the textbook for that. I only read the slides because that's where all the answers were. But one day I just became so fixated on a chapter that I came up with 1,000 questions on everything to do with that one chapter, completely by accident. So, of the lessons I learned that day, always read your textbook, and always do something with the questions you ask. I was told from a very young age that I ask a lot of questions, because I do, and it has been found to be annoying by some teachers and remarkable by others. It all depends on who's your educator. But no matter what, your questions will always get you further in life. That has been the main theme of all the things that have been shown to us today. And I think that's really important to be heard by someone like my age, considering I'm just 15 and I came here from Florida all the way to Boston. These are the kinds of opportunities that you can get. On to the winners. The theme for this one is, if not the divide, then what? So the first winner is Paul Roman Bertzos, 13, from Greece, who asks, how do cancer cells manage to bypass natural aging processes? Fantastic question. The next winner is Surindi Krishnan from India. And her question was, if the goal of any species is to produce more offspring, why are most of them restricted to genders that are only compatible with each other? Also a very good question. Thank you.
The final two questions with the theme, boundaries and balance of evolution. So the first question was, could impressive life events imprint on genes, and can they be passed down or cloned to the next generation? By Xinyi Liu, 12, from the US. So very interesting, sort of touching on epigenetics and all sorts. And then the last question, but not the least. So this is quite long, but if we follow it through, why are cells so limited in combating viruses which are non-living and cannot survive without cells? Could that showcase a relationship between the origins of the first viruses and the first living organisms? Could viruses have once been created by cells for multiple cell functions? By Konstantinos Konstantatos, 17, from Greece. So we'd like to congratulate everyone. Big round of applause for the whole 10 winners, 5 boys and 5 girls. And we'd like to leave you with this final quote by Richard Feynman. fall in love with some activity and do it. Nobody ever figures out what life is all about, and it doesn't matter. So I'm not sure if we can...
Wonderful. Thank you.
Thank you.
All of the organizers and all the speakers and all the winners come up for a group photo, please. And I think this probably is the end. I don't think we're going to do a panel. I'm leaving it up to the rest of you. I think it's quite late already. But please, everybody who participated and helped, thank you. Come up here for a little group photo, and I'll have people like Chris take pictures. And if anybody wants to say anything, who is going to be on the spot? Here you go. Sure. You can talk some. Come on.
So about what I was saying earlier about youth always asking questions. It is something that I've been told a lot from my aunt to my Uber driver. They've always been fixated on. Oh!
I'm sorry.
Congratulations.
We misspelled the name.
What's your name again?
Oh. Well, let's do this now. Can you announce her? Oh, there was another winner here.
Wait, what am I supposed to say?
Just say your name.
Come up. Everybody up.
My name is Xing Yi, and I'm 12.
Okay. I'm so sorry about this. We misspelled your name. That's terrible. Can you tell us what your question was?
Can memory be cloned and passed down to the next generation?
That is a great question. Thank you. So this is what they get, although they have to get it in the mail. And this is thanks to our sponsors. They get a medal designed by Shugwang, and they get a certificate that is, I believe, on the Swedish royal bond, and it is calligraphed by Liz Ball with their names that they will get in the mail. And they also get an iPad too. And thank you so much to the sponsors and everybody who helped organize this, and the organization, Debbie, and the speakers, and Bengt, and Magdalena, and Per, who is in Sweden. Thank you, Per. And all the people in New Zealand who are also helping, and all the audience. Thank you so very much.
Wow. Oh. Yes, eh?
Yeah,
yeah, you come with your father. Where's the mother?
Yeah.
Where's the mother?
Come on, quiet.
Hey, Chris, thank you. Yeah, there you go. Okay, good. The sponsors are up here too. Excellent. Come up here.
Thank you.
No, the mother of the daughter.
As a sponsor, as my daughter. Thank you.
Why don't you introduce yourself?
Oh, yes. The father was the winner.
Yeah.
Okay. Good.
Hey, boy. Look
away.
Yeah.
Take a picture.
Come here. Down here.
Good. All right. Fantastic. Wow. Never happened. Only the second time, 2 winners in the audience. And Aaron, you just left. Let's do the picture.
Thank you very much.
Congratulations.
I'm so sorry. We didn't realize you were in the audience. I'm so sorry, guys. Oh, we should have had you up here. Oh. Wonderful. Thank you so much for the question. You'll receive your prize in the mail.
I want to ask, what is your name?
My name is Latoya.
Well, if you have it.
Latoya Willis.
Can I use your quote that asking questions will always get you further?
Wait, where did I put the microphone?
With your name carved on the iPad.
Oh, where did I
Wow.
put the microphone?
Yeah.
So,
What's finished?
Okay.
Finished.
Thank you.
Gigi and the boy and you. Come on. Come here. I want to selfie.
And you're